Will I be able to share my screen?
Oh, yes. Someone, do you mind giving Sharena host permissions, please?
Yeah, I'll do that.
Co-host. And we should be live. Yep. So, let's turn directly. I'm gonna hit record to the cloud.
Excuse me, I think you have to make Sharena the host, the co-host.
Sorry?
I think only you can make Sharena.
Interesting. Sharena, do you see anything say co-host next to your name?
Nope. Oh, this is great. This is the point where I can start sharing my screen.
Yes. So right after finish speaking out loud your bio, I'll cue you and go ahead and share your slides.
Excellent.
Speaker intro. I'll take a picture of this intro part. Alrighty, looks like we're 4 minutes past the hour. Hi everyone, good morning. If you're on the west coast of the States, good afternoon or good evening or good night even. Welcome to Simply Neurocon 2021. We're super excited to have you all here with us today. Before we get started, just wanted to point out a few housekeeping things and notes to keep in mind. We do have a live captioning available here, so if you go to the bottom of your screen, you should see the option to enable or hide those captions or see the full transcript. Please keep yourself on mute. Everyone should be on mute by default. If you have any questions logistically wise, please try to send them to one of the hosts. You can see that everyone, could you actually mind, SN folks, could you mind adding SN to your name bio if you can rename yourself? So you can message me, Chinmay Valisu, see SN in my little headline there. Arvind has SN as headline, Intra or Sasha even. Please try to avoid messaging Ms. Rice or any of the speakers while they are presenting because it can be a little distracting. We will be opening up the chat when we have Q&A towards the end of each of these sessions. And you'll hear us reveal when it's time for the Q&A. A note that this meeting is being live streamed to the YouTube channel. If you go to just look up Simply Neuroscience on YouTube, you should be able to see it says live right at the top of the channel. And this is also being recorded. So if you would prefer not to have your likeness, basically your video, your face, and your voice in the recording, please keep yourself, with your mic turned off and your video turned off, and it should be all good to go. You should see, that it says live on YouTube on the upper left and recording on the upper left. Some sessions may or may not be recorded, so you can just get a general sense of what is based on that little note up there. If you have not already seen the email that our events team sent out last night from Neurocon at simplyneuroscience.org, it has some handy tidbits on including an invitation to the Simply Neuroscience event Slack, where we share upcoming opportunities like these. You can also be in touch with fellow attendees and Simply Neuroscience team members. I believe that is all the logistics, folks. Have I forgotten anything to mention? Nope. Alrighty, give me 1 second to get these slides going. Oh, wrong one. Great. So just a little brief overview. I've mentioned Simply Neuroscience a couple of times, and we wanted to give you a brief picture of who we are. We are the organization that is organizing this conference. And to summarize, we are an entirely student-led, volunteer-based nonprofit organization. We're primarily based in North America, I would say, but we have global reach and global impact with the community like yourselves. And our mission overall is to empower students like ourselves to get involved in neuroscience and psychology through a three-tiered approach of education, outreach, and awareness. So really trying to connect you with content, mentorship, events, opportunities, and whatnot, and inspiring you to put those into action within your own community. So with that, I'm going to pass it over to good friend Indra.
Hi everyone, welcome to Neurocon. So I'll just tell you a bit about what it is. Simply Neurocon is our annual global conference focused on exploring the diverse multidisciplinary branches in neuroscience and psychology. With prominent scientists, professors, entrepreneurs, and activists in the field, we are sure this event will provide you with new insights and inspirations for your personal brain journey. So this year we have some pretty exciting events. We'll be starting off shortly with our keynote speech, applying neuroscience to innovations outside of neuroscience. And then we also have some other interesting ones coming up, like why aspiring neuroscientists should study history. And we have a popular one that people requested about the gut-brain axis. And we also have an LGBTQIA+ queer neuro roundtable. So please do stay on for everything that interests you, and we hope you get to learn from this conference. I'll hand it over to Arvind now.
Yeah, welcome everyone again to Simply Neurocon. Thanks all for coming. So if you want to check out the event schedule, you can visit this link on our website or just copy this link into your browser, and you should be able to look at all of the upcoming events for the day. And we hope you can join us for as many as you're able to. So look forward to that. Okay, next we have a fun icebreaker for all of you. So since all of you are obviously from around the world, from so many different regions, just type in the chat what state or country you're coming into Neurocon from. Okay, we have Brazil, Turkey, Israel, Hungary, California, France, a lot of different US states, India, Estonia, Argentina. Nice, you have some unique ones. Yeah, so I see all of you guys are from so many different places. So thanks all for coming, some of you in the middle of the night even. And we hope you enjoy this whole event, and we hope we can make it worthwhile for all of you.
Awesome, thanks everyone. While we're here, I just want to very quickly acknowledge everyone who was very central to organizing this Neurocon. As you remember from last year, 2020 Neurocon was a really great start, and we're super excited to be on the team bringing you Neurocon 2021 this year. So I wanted to shout out Intra, if you want to wave for the crowd. Intra here is co-director of events. Our second co-director of events, Sydney, may be here a little bit later. It's quite early here in California. We have Priya, who I believe has her video turned off right now, but Priya is our amazing logistics lead. I don't know if Shreya is on the call right now. She is our wonderful design lead. We have Sasha, who probably, well, there you go, wave. Sasha is our chief operating officer. Arvind, who you heard from a little bit earlier, is our board president. And my name is Shemai. I'm the founder and CEO of Simply Neuroscience. And this event takes place with a whole village behind the helm. So there's many more folks you'll be seeing throughout the event. And with that being said, let's go ahead and get started with our keynote speaker, who I'm going to really briefly introduce to you. Just a really wonderful human being. Miss Sharena Rice, if you want to wave at the crowd. There she is. There she is. Sharena played with electronics as a child, wrote a documentary on human behavior in middle school, and eventually became an entrepreneurial scientist. She is co-founder of Info Inc., a computer vision company for pedestrian behavior prediction, and its DevOps spinoff, Squad Nest. She also serves as scientific advisor for Valence Vibrations, which transmits emotions from tone of voice to wristband haptics. In her dissertation research in the OMAR lab at the University of Michigan, she studies neural circuits involved in navigation, learning, memory, and sleep. And very exciting news is that she'll be completing her PhD in neuroscience this August, almost there. pretty crazy. Awesome. With that, Sharena, feel free to go ahead and share your screen. Get started.
Excellent. It's wonderful to be here and to be talking about how to apply neuroscience to innovations outside of neuroscience. The structure of this talk is, first I'll tell you a bit about my story in education and in life, then give you a brief intro to neurophysiology, because some people here, they're in middle school or they haven't studied neuroscience. Some people are senior leaders in their field. So it's great if we could be synced on the same page when it comes to some things about neurophysiology. Then I'll get into the question of why neuroscience inspired technology and some real world applications of this. When I was a kid, as Chinmayi said, I enjoyed playing with circuits, and I was always a step ahead in development, where adults, they made a lot of sense to me, and children, they really didn't. It was like I was a child trapped, well, an adult trapped in the body of a child and surrounded by other children in general. But then I became curious. Okay, why do people behave the way they do? And why do they do this sometimes to their detriment? Sometimes making bad decisions that don't lead to great outcomes. So then what I did was I picked up a pen and a notebook and I observed people in classes. I wrote down my observations about why people behaved the way they did and my ideas and what they were doing. And this eventually expanded to 45 typed single-spaced pages. I called this documentary The Theories of Normality. Then as an undergrad, I thought, okay, in college, I'm interested in these various different fields, biochemistry, philosophy, psychology. These are so interesting, so I'm going to take classes in them. And I felt pulled in 3 different directions. Okay, do I go with biochemistry? Do I go with philosophy? Do I go with psychology? But then I realized that all of these things are interesting to me because they're about how thought works and about how behavior works. And really, it comes full circuit where I'm back to pretty much being my middle school self. And I can combine them all now by being a neuroscientist. So then I came to the University of Michigan for a post-baccalaureate research education fellowship. I got to do things like work with this confocal microscope to look at neurons and to look at various different molecules that are involved in how neurons talk with each other. But neurons also talk with each other with electricity, and being able to use machines such as this one, where we can amplify from how a tiny little neuron is talking and be able to actually see that. It is quite a magical thing. Nowadays, for my thesis lab, I work in Omar Ahmed's lab, otherwise known as the Omar Lab at the University of Michigan. And we can see here that on the left, I'm looking up at a bunch of things that we've put together for implants. This involves making all kinds of things by hand, milling out plastics, spinning wires together, putting screws in, and these are all really super tiny parts because it's building implants for the rodent brain. And on the right, I'm here with the virtual reality setup that we have for mice in the lab. Mice in virtual reality is quite a crazy thing. Imagine this situation. There is a mouse on a spherical treadmill, and as the mouse is running on the ball, then the visual image on the screen is moving closer to it. So the mouse runs on the ball, it's navigating through this virtual maze, and we can use this to study things like how the brain processes speed and space. I also enjoy inventing solutions to problems, just because if there is a problem, then it's nice to just come up with something. One of the problems that I've had during the pandemic was the sense of it would be really great to have a pet. But the thing is that my apartment doesn't allow for pets. So what do? My answer was I took this plush unicorn pillow pet and wrapped it in a heating pad to create a proxy pet. And it's warm and cuddly. And it solves the problem that I wanted it to solve, and I don't have to feed it. I also made a lot of the art that I'm going to show in this. Here I am drawing some of the art, and then I converted it to a digital form in this nice black and white aesthetic. All right, in my thesis, I study spatial navigation, how we know where we are in space, how we know how fast we're moving through space, how we know where we're going next. So this is going to be something that comes up a lot in this talk, how speed, space, and sensory information and memory are integrated. The brain is an inner cartographer of sorts. We make mental representations of the outer world, and that's how we know how to find our way home from various places where we may live, or find our way from class to class if we are at school, or do various other things in finding our way around. All right, why is the nervous system a good inspiration for technology? There are lots of reasons for that, and I'll give you the brief intro to neurophysiology at this point. All right, what is neuroscience? Usually when people think neuroscience, then they're going to think of brains, and that is true. All right, the brain is composed of various cells. They're called neurons. Here's a picture of some neurons. And we can measure electricity from a neuron and see that it creates an action potential. So it is a little bit of electricity when it talks to other neurons. And if we have a bunch of neurons together, then we have tissues. Here are some pictures of brain slices. And brain slices, they're useful to look at because they allow us to see which parts of the brain are connected to each other some ways, and in some ways that MRIs might not be able to do. So for example, we can look at microcircuits of the brain by looking at slices. We could take a brain and inject it with some viruses. And then we can see, okay, if we injected the brain with some viruses, then where are the labels that these viruses express going? So here we have a picture where I have injected a brain with a virus that expresses a green fluorophore. So it's going to show up as green under a microscope. And then we have another fluorescent tag I've injected, which is going to show a red label in the brain under a microscope. All right, when I did this, then I found that, okay, this part of the brain, which is involved in head direction coding, so it has a lot of brain cells that tell us where our heads are facing in space. Well, they project specifically to some other regions. This is a region that my lab studies a lot called the retrosplenial cortex. And the retrosplenial cortex is an association cortex that takes in information from sensation and also from memory. And it is kind of like that bridge between how we know where we are in space and how we know where things are in our environment in relation to each other. So how I know where the bank is in relation to the theater is in my city. This is part of what this image in the middle of the screen does. There is also this red label. Okay, I injected this into a part of the brain that's involved in a lot of consciousness things, and it projects to certain layers of this part of the brain that's known to integrate lots and lots of things. So by looking at different circuits in the brain, then we can see, okay, how is this information aligned? How is it processed? And we can see also by increasing our sample size that we find the same results over and over. So this is indeed a more valid result than if it only happened once. All right, those are some tissues and some tracing experiments. But what about measuring electricity from tissues of the brain or from the brain? Okay, neurons, they fire action potentials, but also populations of neurons fire action potentials. And when they sync up, then we can see what's happening. All right, here I have put a probe into this brain with a lot of neurons around it. And you can kind of think of it this way. If you have a stadium full of people and people are talking, then you can catch the murmur of the crowd with the microphone. But if your microphone just happens to be dropped near someone, a certain individual, then you could pick up on exactly what that person is saying. But at the same time, you can hear the murmur of the crowd in general. So you can probably tell, okay, with this microphone in the stadium, what is going on in the football game? Is the crowd excited about something? Is it kind of a dull moment? We can see things like that with putting probes into brains as well. And with these probes, we can look at the sums of activity around the probe, and we can see brain waves. Synchronous activity is going to create a brain wave, otherwise known as an oscillation. Lots of neurons talking kind of in the same way at the same time. And if we zoom into a brain wave, if a probe site or a wire happens to be close to an individual neuron, then we're going to get things that look like this. We can see action potentials riding on the waves. So brainwaves, they're a measure of synchrony, and there are different kinds of brainwaves that happen at different frequencies and different levels of power. And these are useful for the brain communicating between different regions and between neurons. Gamma waves, they're associated with all kinds of behaviors that involve a lot of focus and attention. So for example, if you're running around the environment, then you're probably going to have gamma waves so that you can process all of this information very quickly. Theta waves, they come about also during navigation behaviors, and they're involved in learning and memory. And delta waves, they are slower oscillations that occur during sleep. Another way to think about how the brain processes information is that if you have a lot of people in a room, then, and they are all clapping, if they are all clapping in phase with each other, all clapping at the same time in the same way, then we're going to get something that looks more like a delta wave, where you get a strong sense of they're clapping in this particular way at this frequency. And it's going to be a strong information transfer. But if everyone was clapping randomly, then it may be more information that is more dispersed, but the power of the whole thing is not going to be quite as strong because people are clapping at different speeds. So it doesn't all sum up to one big sound wave. So that's kind of more like gamma rhythms and how those work. All right, neurophysiology involves brains, but wait, there's more. When it comes to neurophysiology, there's also everything that involves sensory information. So this is the eyes, this is the ears, this is muscles, this is movement. And sometimes it's social behavior as well. A lot of things that people do and things that animals do, they have a neurological underpinning to them. All right, why is the nervous system good inspiration for technology? For one thing, it senses signals. All kinds of streams of information are coming into us, but also they are able to be processed by us. The nervous system learns and remembers things. This is useful for making artificial intelligence. The nervous system performs computations. One example of computation is that some neurons, they keep track of head direction, which way we are facing in space. And they still keep track of head direction, even if we are in different positions. So my arms could be up, I could be standing on one leg, but as long as my head is facing in the same way, then my same head direction cells for that specific direction, they are going to be excited. It's kind of like having a neural analog of a compass. This is useful for navigation technology. Imagine if Google Maps did not show which direction we were facing. Then it would be a lot harder to use maps to navigate around. We might find ourselves going in the complete opposite direction of where we should actually go. Of course, we could orient ourselves by looking around and seeing, okay, what else is in the environment that is shown on the map? But it's much easier to do if the technology just tells us which direction we're facing. All right, the nervous system integrates signals, it responds to stimuli, it learns and remembers. Another example of this is place cells. Place cells, they exist in this part of the brain that's very useful for learning and memory. Maybe the most studied part of the brain, called the hippocampus. And if a person is walking around a room, say going from here to here to here, and we map out their entire trajectory as they continue wandering around the room, then we'll see that there is a certain population of neurons that becomes excited when they're in one place versus another place versus another place. So these various ensembles of neurons getting excited at once, they allow us to know where we are in places. And blind people, they have place cells too. So it's not just a visual thing. It's also a matter of being able to interpret other signals, such as movement signals that come from the motor system. These insights might help autonomous vehicles. For example, if one kind of sensor in the autonomous vehicle does not work, then maybe another can compensate. So for example, if a camera is blocked by really heavy rain, then maybe if the autonomous vehicle is equipped with LiDAR technology and creates point clouds that are not based just completely on camera data, then this may be able to help the autonomous vehicle navigate in severe weather. I have some examples to show in my robots that I've made. All right, navigation, it could happen by light. Here I am blocking some photosensors on this robot with my hand. And it's an afraid of the dark robot. So it will continue moving around. Well, it will move around according to where the shadow is. Similarly, I have made a whisker guided robot. And what happens here is that if the whisker hits a wall, then this completes a circuit in the robot and the robot pivots and continues moving onwards. All right, those are some robots, but what about the nervous system? Well, they're super duper efficient in terms of size, processing ability, and energy usage, where even the tiny brains of mice and rats, they're able to do all kinds of things. They respond to startling information. Rats and mice, they're very social creatures, and they're able to form bonds with other mice and rats. And they're able to make nests and learn tricks and all kinds of things, even though they're really small with super small brains. So if we can take some of these principles and put them into technologies, then perhaps they can work more efficiently using less energy to get better outcomes. Also, the nervous system is good inspiration for technology by making technology that is compatible with the human nervous system. An example of this is that 10% of people have alexithymia, which is that they have hardships identifying emotions. This may be emotions that they experience themselves, so they don't know how they are feeling. But also, when it comes to interacting with other people, it could be hardships interacting with them, partly because they don't understand how other people are feeling. Okay, 10% of people, that is quite a lot of people. So there's the question of, okay, what is the root of alexithymia? And we have a good hypothesis, which is that maybe this is a mind-body problem, which is, okay, well, people, they receive all of these signals, and every emotion, it comes with some sort of physical sensation in the body. So for example, if we're excited, then we will physically feel lighter than if we are having a terrible day, in which case our bodies may feel more heavy. And a separation between the mind and the body, where people may be feeling feelings, but they're not paying attention to how their body feels, that can create problems with processing emotions. And this can be both with a self and with other people. All right, every emotion has corresponding bodily sensations, from heartbeats being faster when we're excited to feeling this warm glow when we feel loved. And what one of the companies that I advise does, this is Valence Vibrations, is that we take emotions from tone of voice, process this through a machine learning algorithm, and then this creates, well, this sends the information over to a wristband. And the wristband, it's going to be able to create vibrations, various haptic feedbacks with different motors and different emotions corresponding to different vibrations. And perhaps this external signal will help people to sense things about emotions that they don't already feel. Okay, vibrations to express emotions emotionally. So for example, when it comes to anger, what if this wristband, it could just feel angry by having this intense burst, and that's exactly what it does. Or if it feels happy, then it will give this sense of a kind of happy tingle of vibrations. All right. The mind-body problem may be the root of alexithymia, but when it comes to relating to other people, there's also the sense of, okay, could there be situationally induced alexithymia, where people just do not understand each other in different situations? And the answer is probably yes. Sometimes this comes from visiting other countries and finding that you don't quite understand the culture. Or it could come from having different personalities from other people and people just not feeling the same way that you would because they just have a different personality and a different response to the situation. It could be also a matter of neurotypes, where some people, they are really, really sensitive. So, or they may emote in different ways than others, in which case, maybe other people don't quite understand their emotions as well, or they may find it hard to understand other people. So we're developing an emotional passport that will allow people to understand people across different, well, all kinds of humanities. All right, now there's the question of how to think like a neuroscientist. Okay, thinking like a neuroscientist, I've distilled this down to 3 parts. The first question is, what is the situation? The second question is, how does the nervous system handle the situation? And then the third thing is, okay, how do we synthesize the situation and the nervous system and apply this? What is the situation? Well, situations, they're everywhere. So there's the sense of, okay, it's good to follow curiosity. Situations can range from almost anything. Like, okay, there is a lizard that is breathing, or a dog can be trained to do a trick. But it's useful to follow curiosity so that you actually do have a direction in terms of where you're going as a scientist. Some people, they are curious about disease mechanisms, things that go wrong or things that are very peculiar about how the brain and behavior work. And then they follow those things. I personally like to look at things in everyday life just because I'm an easily amused person, and just things that everyone can relate to and things that are just typical everyday things. They're kind of magical in a sense, that the brain, this tissue, these bags of life talking to each other can do things like create our entire interpretation of the world. And also the brain named itself. It's the only organ that did that. All right, one situation that there is that most people, they stay balanced while they're walking around. How does the nervous system handle this situation? One way is that there is the vestibular inner ear. Okay, the vestibular inner ear is a part of the ear on the inside. And how it works is that it has these semicircular canals, and these are fluid dynamics. So you can imagine some water flowing through a hula hoop, and the hula hoop might be able to sense this. Well, that's kind of like how these semicircular canals work as sensors. Okay, well, we also have rocks in our heads. We're born with otoconia, which are these crystals of calcium carbonate, and they exist in the utricle and the saccule, which are also parts of the vestibular inner ear. So if we're moving through our environment, then that will allow us to have a sort of biological accelerometer. So we have this 3-axis gyroscope and this accelerometer as a biological system that helps us maintain our balance and to be able to figure out where we are in space, even if we have our eyes closed or even if we're blind. And these different signals from the vestibular inner ear, that goes over to the brain. All right, that's how a certain biological system works. But how can we use this? Some principles, they may have been used in some old measurement tools. And I've made a sassy robot that it's like a magic 8-ball, but it has 64 different sassy responses to it. And this uses a tilt sensor as well, an electronic tilt sensor, which uses kind of similar principles to the vestibular inner ear. But what about the problems of today that are more pressing. One of these problems is the problem of what happens when we switch from self-induced active modes of transportation to passive modes of transportation. So for example, if we are walking around versus if we are navigating a space in virtual reality. I remember that my first year of grad school, one of my friends had me play Portal, a video game on the computer. And I was playing Portal and moving through the portals and just feeling the sense of simulator sickness that got me just not wanting to play video games anymore. So could this kind of thing be alleviated? That is a huge problem for the virtual reality industry and a huge problem for the game industry in general. The sense of there's something that is lost when we switch from actively moving through an environment versus just having a visual scene. But also, you may notice that people, they don't become car sick when they're riding a bicycle, and they don't become car sick usually if they're driving the car. But if they're being driven around in a car and they're reading a book, then this creates a conflict. And the conflict is that the signals from the vestibular inner ear, they're going one way. Information from the visual system is going in another way, and this creates a sensory conflict. But how would nature more ideally handle the situation? How nature would ideally do this is that both of these things would be synced up somehow so that there isn't a conflict. All right, if we look a little bit deeper into the circuit, then there are neurons in an arc and there are various different brain regions that are involved in processing the visual system, well, visual information and vestibular information. And I'm not going to go very much into this, but understanding this biological circuit or other biological circuits, and then combining the external stimuli with the biological circuit, adjusting to gain a situational understanding that may help. So for example, the motion sickness example. Okay, what would happen if vision was aligned with the vestibular information? In that case, then motion sickness is alleviated. So if you look out the window instead of reading a book, then you may find your motion sickness fixed, at least for a moment. So what if we could use computer vision systems, for instance, or some sort of electrical stimulation to make sure that both of these things, they seem to be processing information in the same way. Understanding the science, it can make for the difference between just hacking things together on the surface and making deep technology. And well, one way to think about this is that some people, they have chronic pain, and this may be solved by pain medications or from physical therapy on the surface. But what if the root of the problem is really that they habitually have bad posture? And by having bad posture and being hunched over, then maybe their body is just not aligned to itself, and this creates things like back pain. In that case, then really the root of the problem is that this person has habitually terrible posture, and this may be solved on a deeper level by changing their habits so that they can have good posture. And then this solves the problem in the long term so that they don't have to take medicine. It solves problems in the long term because they don't have to go to physical therapy because they're already knowing how to move properly. So solving problems at the root rather than just at the surface, that is what we can get to by understanding neuroscience and being able to apply that to technologies. Still, it's useful to hack things together for creativity and for prototyping. You can imagine too, okay, there are artists, and artists, they may be very, very well trained in how to make art. But is it useful for people to be able to doodle things and to be able to sketch out ideas and to be able to test diagrams and things like that? Absolutely. And a lot of scientific advancements and technological advancements, they're stumbled upon by accident. So it is still useful to tinker around, even if the problem is many, many levels deeper than it may seem to be on the surface. All right, how else could neuromimicry work in developing technologies? We can look at different kinds of animals and how they solve problems. For example, there are dolphins. And dolphins, as they swim through the ocean, they never quite completely sleep. Half of their brain may be asleep, while the other half of their brain is awake and keeping them moving through. And this is important for dolphin survival. What if we were able to use this for other technologies, such as space exploration? For example, sending telescopes into space, like the Hubble telescope. Well, there's a problem for telescopes if they run out of energy. It would be really sad if a government spent millions of dollars to put a telescope out in space and then it just ran out of batteries or out of power while it's out there. But what if it could happen that if the telescope sensed that it's far away from solar power or far away from light sources where it could gain more energy, then it's able to turn off some of its systems, maybe doing some sort of electrical equivalent of shutting off half of its brain while it navigates to places of more life. That would be a good example of neuromimicry in technology. And different animals, they're good at doing different things. And different animals, they'll have different sensory systems. Bats, they may navigate more through echolocation. And these kinds of things, some sorts of perceptual analogs can be made in electronics. There's also the potential for combining computer vision with human vision to envision better outcomes for road safety. One example of this is how one of the companies that I've co-founded, Info Inc., builds models for pedestrian behavior prediction. Okay, what goes into predicting pedestrian behavior and what goes into how risky a person is in a situation? We can look at different things such as, okay, is the person interacting with the vehicle? Are they distracted on their phone? Are they riding a bicycle? Because people on bicycles, they're going to move through their environments differently than if they're just walking around. So can we create some sort of computer vision system that is able to process things kind of like how a human would? So being able to look at how fast a person is moving through environments and what they may be doing next. And the answer is yes, by looking at different risk factors and different things about, okay, how would I try to classify how a person is moving around and how risky they are? What things would I look at? And these ideas, they can help in building a model for them, because we could train a model, but it's also useful sometimes to ask in annotations of data in trying to make our initial ideas and be able to say, okay, this person is this person is doing this, and then eventually to take these notes about how people are behaving and put them into a computer vision system. Well, yes, it's useful to have these predefined beforehand. because risk is kind of a fuzzy situation. And so we use things like fuzzy logic. And so in combining all of these ideas about how human behavior works and how risky things are, then we're able to assign levels of risk. And those are denoted by these white bars on the sides of these bounding boxes. And then we can take these risks and then we can send a warning signal to the driver if a pedestrian is very much at risk. And the human driver is the person who makes the decision. This is a great thing because a huge barrier between where we are now and having fully autonomous vehicles is the sense of, okay, who is responsible if an autonomous vehicle hits someone and kills someone? What if an autonomous vehicle is, well, it navigates into the complete wrong way, in the wrong direction at the wrong time? Well, having a human in the loop and having a sense of responsibility is something that is quite human, where there is this sense of guilt if something bad happens. Whereas with a robot, well, what do? Do you sue the company or what? I guess that you can, but this is a huge issue when it comes to technology ethics in this field. So for now, keeping the human in the loop and the human making the decision, it is useful. They can choose to attend to the signal or they can choose not to, just as much as they can respond to that beep that most cars have, which tell, okay, do you have your seatbelt buckled or not? And you can choose to obey the law or not. But another company that I've co-founded is the DevOps spin-off of Info, which is Squad Nest. Can we take a bird's eye view of multiple information streams? And what we've done is we've created this smart city dashboard, which is that it senses all kinds of things about road traffic, how fast the pedestrians are moving around, how fast the vehicles are moving around, how many red light violations, and things like that. And this can be used for city planning, where there are many useful patterns of how all of these movements work that a human may not be able to notice or sense on their own if it wasn't for this kind of technology existing and measuring all of these metrics from computer vision systems. In the companies that I've co-founded and advised, it's interesting that we are not measuring neural data or what is considered neural data as typically defined, but it still makes for a better use experience, makes for better human factors and understanding humans in order to make for better outcomes. Thus, in summary, neuroscience combines many, many fields, and advancements in neuroscience and technologies, they're fueled by curiosity, by making connections, by applying knowledge. And neuroscience, it can be applied to many different kinds of technologies. As a shameless plug, I'm also on the job market for a full-time position, leading more tech development and expanding what humans and machines can do together. I'm also collectible at Clubhouse, Twitter, and Medium at Sharena Rice, or on LinkedIn. I'm happy to connect with anyone here. May the force be with you in going full circuit, whether it is considered neural or not. And I have this. All right, questions, comments, concerns?
Everyone, please feel free to drop them in the chat. If you're on the YouTube live stream, also feel free to drop in the chat and we can read them aloud to Mr. Rice. Thank you so much. That's a really wonderful presentation. right, we've got a question from YouTube. Roman asks, what are some technologies being developed in order to help with mental illness, such as anxiety disorder?
There are multiple technologies. For one thing, there have been advances in drug development. So for example, when it comes to psychedelics, these have been misunderstood for many, many years because of the so-called war on drugs. People thinking all drugs are bad, so let's just get rid of a lot of them and make some of them banned or illegal. Okay, but what if that is the very thing that could solve the problem? And sometimes it might be. So currently, psychedelics are being explored to treat lots of mental illnesses and depression. As for other things, okay, there are neuromodulation technologies looking at brain signals, and really they come from neurofeedback. What if you can solve attention disorders by having some sort of brainwave readout, but at the same time having that as you're doing a focused attention task, and then you're given some sort of feedback about how well you're attending to that, and then you're able to make some adjustments. Okay, maybe it's because of the music that I'm listening to that I am so distracted. Maybe if I change that, oh, my brainwave is operating somewhat differently now. Okay, well, you can do self-experimentation with that more and more as these technologies are developed and they come into common use. Some other technologies may involve better therapies. Therapies that just take people from the outside and do not really explore them on the inside and take into account, okay, how does the nervous system actually work when it comes to repeating patterns of sad thought loops or anxious thought loops. If we're able to modulate those circuits by thinking in certain ways, by having this sense of internal locus of control, otherwise known as the sense that you are in control of your own destiny, that is going to modify the brain as well. Really, anything that you do is going to modify the brain, which is kind of scary, but it's also very wonderful.
It's really fascinating to hear about. Thank you for sharing that. We are almost at the hour, so I'm going to go ahead and ask this question from Erin. Folks who have more questions, as Ms. Rice dropped her social media links before, you might reach out to her on those platforms as we are short of time for this specific event. But to wrap up, Erin has a question about the pandemic, because it's kind of on the front of all our minds. How has the pandemic changed how you work or the topics that you're interested in?
The pandemic, it has put me at home quite a lot more than I was before, where I can't go into lab quite as much to do things. So I've been analyzing data at home and reading a lot and not actually doing stuff in the lab quite as much as before. So a lot of things in neuroscience, they can be made through computational models and through looking at what is there rather than just collecting data continuously. Sometimes people procrastinate from analyzing their data by just doing more experiments.
Love that. Thank you so much. Alas, that is all the time that we have. Everyone, if you could join us in giving a big round of applause for Ms. Rice, whether physical or Zoom applause. Thank you so much.
Oh, this is great. It's digital applause. And maybe it's a gamma rhythm, and maybe it's a delta rhythm. Thank you so much.
Thank you. Everyone, like I mentioned, Ms. Rice shared her social media channels before. Even if you type her name into Google, you should be able to find her profiles and learn more. Awesome. Thank you. And with that, I'm going to pass it over to Intra to introduce our next guest speaker.
Sure, yeah. So next up, we have Dr. Richard Brown presenting a talk called Why Aspiring Neuroscientists Should Study History. Dr. Richard Brown was born in Victoria, Canada, and received a BSc Honours from the University of Victoria, an MA and PhD in Psychology and Physiology from Dalhousie University, and was a postdoctoral fellow in zoology at Oxford University. Since 1978, he has been a professor in psychology and neuroscience at Dalhousie University in Canada. His research has been on behavior of rats and mice, and over the years that has encompassed studies on sexual behavior, olfaction, social behavior, ultrasonic vocalizations, development, and aging. His lab is currently examining sensory, motor, cognitive, and affective behavior over the lifespan of mouse models of Alzheimer's disease and autism spectrum disorder. He has taught IBRO courses in neuroscience in Kenya, Nigeria, Uganda, and Cameroon since 2003, and has a long-standing interest in the history of neuroscience. He has published over 200 papers and 2 books, namely Social Odors in Mammals and an Introduction to Neuroendocrinology. So we welcome you, Dr. Brown, and I'll hand it over to you now.
Okay, can you see me? Can you see me waving? Can you hear me?
Yeah, we can.
Okay, well, I have to bring up my share screen, Kyle. I have my student Kyle here to help me with the high-tech stuff, which is everything.
You probably only need to press one
Yeah,
of the buttons at the bottom.
That one.
No, of the you're not
Whoops.
sharing your screen yet.
Okay, just a minute. I have to press a button somewhere, Kyle tells me.
Share screen.
Share screen. Okay, you ready? There.
And
Share. Okay. How are we? Can you see the screen? Why study the history of neuroscience?
Looks perfect.
Yes, we can.
You can see it. Okay. So a number of years ago, I became interested in the history of neuroscience. And it turned out the more I've studied the history of neuroscience, the more interesting everything has become to me. That's right. I'm going to get a drink of water. I'm so excited. My mouth is drying. Anyway, it's interesting how many people are interested in this topic. I was surprised that you discovered me and asked me to be in your conference. I was quite pleased. Since I'm in Nova Scotia, I'm wearing my sailing ship shirt. So this is a talk I started giving, 10 years ago. I'll just say that in our research, as you mentioned, we do many different projects. This is just half a dozen recent projects on MDGA mouse models of autism. Whoops, that wasn't planned. 5 times, every time I move something, it goes. The 5 times mouse model of Alzheimer's disease, synaptic theory of memory, olfactory memory in 5 times Alzheimer's mice, whisker exploration in 5 times mice, done in collaboration with Robin Grant at Manchester, England, and vision in mice done with people in the med school. So we have a lot of collaborations. Can you hear me all right? I get no feedback from you on this, okay, good, thanks. So that's our bread and butter research. It's hard to get grants to do history, so it's my Sunday afternoon project. And oddly enough, here we are, Sunday. Now what? It's not doing what I wanted to, Kyle. Why do you think that is?
Dr. Brown, maybe you could use your arrow keys to go right and left.
Yeah, I was trying to use my arrow key, but nothing happened. Every time I hit my mouse, something happened, but my arrow keys, it went dead on me. So after I'd given this presentation a few times, I wrote this paper, which is in Frontiers in Behavioral Neuroscience on history of neuroscience. And twice a year, I give this presentation at King's College London, and I have my history of neuroscience class at Dalhousie. So every time I give the talk, I change it a bit. And yesterday I thought I'd just edit it, and I spent the whole day making changes. So you have a brand new talk. One of my friends, Stan Finger, read my paper and pointed out that I never mentioned his society. He was annoyed at me. So I have to mention the International Society for the History of Neurosciences and the Journal of the History of Neurosciences, which I use a lot, and I have no idea why I forgot to put them in my paper. And so I have 8 parts of this presentation. There used to be 7, but yesterday I added another. What is history? Why study history? How does history affect you? The vanishing history, saving the history. I added this yesterday. How does neuroscience advance? A historical view of the future. The excitement of historical research, 12 examples, and neurohistory and neuroculture, art and literature of neuroscience. Just sec. Kyle, I think we're set. You can go. Kyle was here to help me in case anything went wrong at the beginning. Okay, so what is history? It's kind of an interesting question. I've always believed in the first one, the one on the left, the Elton 1967, history is a search for the objective truth about the past. And then I read E.H. Carr, history is a product of its own time, the interpretation of the past through the author's ideas and ideologies. And then I came across a very contrarian view, which I put at the bottom. History is not a record of the past, more or less faithful to the facts, but an invention or fiction of historians themselves. So it all depends what you want to believe. I still like to believe that history is like science. You dig out the data and then you organize it and interpret it and present it, but sometimes the interpretation interferes with the data. I keep getting little things on top of my screen and then all of a sudden nothing works. See, I don't know what goes on here. So I found this interesting book, Richard Evans in Defense of History, which he starts out with the history of history and historians and their facts. And I recommend that to you. And then I discovered in the Blackwell's bookstore in Oxford, History and Memory. And I thought, well, since we study memory, really this is how I like to think of history. History is memory with all of its flaws. Memory is a series of incidents connected in time. Memories are reconstructed, not imprinted. Memories are fragmented. 2 people remember the same event in different ways. Forgetting occurs and false memories occur. So that describes you and I and history. And then one day, many years ago, when I was at Oxford, Richard Dawkins was there, and I saw his first volume of his autobiography in the bookstore, and I bought it. And in it was this photograph on the right. And it said the Animal Behavior Research Group in 1970-71. But when I looked at that picture, I'm in it. And I was only there in 1976, 1977. So I dug through all my photos, and there's the photo at the top that's in the book, and there's my photo at the bottom. I'm the fat guy on the right. So I was there in 1976, 77. When exactly was that photo taken? I know where it was taken. I couldn't remember. So something keeps happening. So I was there, but where was it? What year was it? What month or day? What were we doing? Where were we? Was it a party, a punt race, a picnic? Who are all these people? Where are they now? Why can't I remember these things? I was there. So Marion Dawkins told me it was a group picnic in November of 1977. It looks like November because there's no leaves on the trees and everybody's got a sweater on. Anyway, so history is the facts as we remember them. Individual memory, your memory and my memory. When we're long gone, history is the facts as someone who was never there interprets them, a kind of social memory. And I put these references there of what I think are quite interesting books on the study of history. So why study the history of neuroscience? Since you're all students of neuroscience, you probably want to do research. And the previous speaker mentioned doing research and collecting data and the problems during the lockdown and the pandemic of actually getting into the lab. But how many are interested in history? You don't do you study neuroscience to study history? Anyone? Fatima, what about, I can see your face. You can speak, you're turned off, but you can wave. Do you study history in neuroscience, or are you just interested in collecting data? Oh, you're still turned off. Anyway, go ahead.
I'm just interested in the data currently.
Yeah, interested in research. And I don't think you're interested in history, but you are. Many of you are interested in history. Is history of neuroscience a waste of time, which people have told me, but I think they're wrong. You can learn a lot from history, and you use history in your research. I don't know what happens, but what do scientists do? Well, scientists do research and make new discoveries. That's why you're in the game. They design apparatus, they construct apparatus, they develop new methodologies, they develop new procedures and techniques, they test theories, they're involved in controversies, they write review papers, they write critiques, and they write grant proposals. So you think you're doing research, but really all you're doing is history. The introduction of your research paper gives the history of your problem. When you test the theory, you want to know who developed the theory and what data supports and rejects it. When you set up a lab, you have equipment and you have techniques. Well, somebody invented all that equipment and techniques, and who did it and why did they do it? When you get results, they're new today. As soon as you publish them, they're history. The discussion tells how your new findings fit into the history of the problem. Was your theory supported? If not, what do you do? And the references tell the reader where to find the history of your problem. So virtually every section of your paper is a history paper, believe it or not. I don't know what's happening here. So when I give this talk, many students don't realize there's many academic publications on how to write a paper, how to join history. Academic publishing, how to write a research paper so that it will be accepted in a high quality journal, a brief review of useful tips for publishing your scientific manuscript, and of course, that famous how to write a scientific masterpiece. And they come from a range of journals, the Annals of Neurology, Journal of Cardiovascular Pharmacology and Therapeutics, Journal of Clinical Investigation. So they all have tips on writing a paper. So every time that you do a research project, rely on previous studies, methodology, and theories about that project. Each project depends on the history of that project. And once it's published, it becomes history. So how does history affect you? Every moment of your day in school, history affects you. To show how even the names of brain areas have a history, I have my 3 questions. Some of you can open your little microphones and tell me what answer you think is right. So the first use of the term brain: A, the ancient Egyptians; B, Haller; C, Keel; D, Galen; E, Vesalius. What's your vote? Just shout out.
Folks, if you also want to drop your answers in the chat, feel free to.
Oh, well, I'll never see them there, but you can do that. Now
No worries,
Kyle's
Dr. Rand.
gone,
I can read them out loud.
Computer illiterate now that Kyle's gone.
No worries.
What
We
do you
can read
think?
them out loud so you can hear what responses.
Shout out a few. How many think it's A, ancient Egypt? I don't hear anything. B, Haller. C.
Some folks think it's D, A. A lot of A's coming in.
Okay. Well, sure enough, it's A. In the Smith papyrus, so-called because Edwin Smith found it, and it's been translated at University of Chicago. You can get a copy in the library. The Smith papyrus talks about how to treat head wounds and damage to the brain. I have 3 questions. Second question, who named the cerebellum? Galen, Aristotle, Vesalius, Willis, or Gunther? I'll let you collate a few replies. I always forget this one. What are you getting in your replies?
I think people are still thinking. There's silence.
The big quiz.
We have B, E, mostly B so far, a C, majority B and C, I would say there's a split.
Okay, well, guess what? They're right, Aristotle in De Partibus Animalium, and you can still get that from your library translated into English. Okay, final question in our little quiz. Who named the entorhinal area? Arnold, Haller, Brodmann, Arnold again in a different paper, or Swanson 2000? How are we coming?
B,
Sorry?
we have quite a range now. B, E, C, and D. We're kind of all over the board a bit.
Well, this has tricked you, hasn't it? Because the answer is C, Brodmann, 1909, in his book on localization in the cerebral cortex, translated not until 1994 into English by Lawrence Gary. Anyway, the purpose of this little quiz was to say that every time you even use a brain term, it's historical. There's a history behind it. And there's this amazing book by Larry Swanson called Neuroanatomical Terminology, where he goes through the history of every name of every part of the brain and who named it and when and its history and how its name changed. It's a fantastic book. We have it in the library. But when you look at who named the most bits of the brain, it's somebody you've never heard of named Burdak, Meckel, Arnold, Winslow, Haller, Baer, Galen at number 7, still in there from 192 AD, Cruvelaire, Vesalius from 1543, Haas and Walter. So some of the names have been around for a long time and others are newer, but you think every time if you're working on the hippocampus, well, who named it and why and where did it come from? Okay, part 2, how does history affect you? So when you're thinking of a master's project or a PhD project or a research project, what's the history of your project? What apparatus and procedures do you use? Who developed them? What are the main discoveries in your area? Who made them? How did they think of the research? That is, how did they come up with the idea? Was there a controversy? How does controversy influence your research? Is there a definitive answer to your research problem? Or does the answer depend on who did the research? Is the research a product of its time and author? And we'll talk about those in a minute. So what is history? Everything but tomorrow is history. So as I mentioned before, every publication is a historical document so that you become a historian when you publish your paper. If someone wants to replicate your experiment, how would they do it? What equipment and techniques should they use? What methodology? What is the stuff that never makes it into a paper? Once one of my students went to Boston to go to a lab who couldn't replicate our studies, and she watched how they handled the mice, and she said, oh, they just dropped them into the Morris water maze by their tail and they sunk. And of course, then they have a dive reflex. They sink and it looks like they're dead, but you pick them up and stroke them and they come alive. So there are lots of things that never make it into the paper. How the mice are housed, the day-night cycle, what food they get, all these little details. The raw data and statistical analyses, there's a big push now for people to keep their raw data in archives, which I'll mention. Letters, emails, lab notebooks, diaries, all the information about your experiment. How long should you keep this information? Well, I did my PhD in 1976, and people still write to me occasionally for the data, which is long before personal computers. So the data is all on sheets of paper in a folder in my lab. Where should you keep things? This has become a serious problem because I've carefully kept backup disks of every experiment I've done since the 1970s. Now you can't read any of those disks. People write you for data and it's on a floppy disk that you can't read because all the computers are out of date. So now there's a push to keep them in a cloud somewhere, but how long they'll stay there, I don't know. I always feel if you keep data on a piece of paper, you can still read the ancient Egyptian papyrus, but you can't read a floppy disk you made 10 years ago. What if someone accuses you of fraud? We'll come to that. So you need an archive of your research. You need a history of your own research project that if someone wants to replicate your study, they can inspect and look at it and see what exactly you did. So the problem of replication comes up since this guy, John Ioannidis, in 2005, wrote a paper called Why Most Published Research Findings Are False. And he critiqued all kinds of research design, experimental designs, definitions, analysis, everything else. He said, everybody's made a mistake and you can't believe anything. That, as you might imagine, set off a huge explosion in discussions of replicability. So here's a paper from PLOS Biology, Risk of Bias in Reports of In Vivo Research, a Focus for Improvement, how to improve the reliability of experiments. Reproducible research practices and transparency across the biomedical literature. Again, how do you demonstrate that you really did your experiment and you can replicate it and the data is real? And if someone did it in their lab in another part of the world, they'd get the same results. So what if your research cannot be replicated? Now, this happened to me many years ago. Someone published a paper on the left-hand side. The second conclusion from these data is there are no overall sex differences under any treatment. This is perhaps surprising in view of the claim by Brown. Oh, that would be me, that marking is more frequent in males. So I said, oh, I believe my data and they could not replicate it. So this happened. When was 1984, 94, 04, 14, 24. This is getting to be 40 years ago, isn't it? Oh God, it's twice your age, isn't it? Anyway, so I looked at their experiment and I looked at my experiment and I went through every little detail. And I found that they, Burke and Sadler, reared their rats in small all-male groups and they were sexually naive. And in my experiment, I had sexually experienced group-reared males. And so I did the experiment again with one group exactly like theirs and one group exactly like mine, and got their results with their group, my results with my group. So I solved the problem of why they couldn't replicate my study. Then I published it in 1991 in Animal Learning and Behavior. And so far, that's the only time that someone's written that they couldn't replicate my experiment, and I figured out why, but that's not always the case. What if you're accused of fraud? What if someone says they can't replicate your paper and they think you just sat in the pub drinking beer and made up the data and never went to the lab? So there is on the internet something called Retraction Watch. And Retraction Watch keeps track of all the papers that are retracted. So here's a journal, Molecular Endocrinology. A cell biologist who falsified Western blots has notched a second retraction, with one more expected after an investigation at Nanyang Technical University in Singapore. And you can see that the papers were retracted because they falsified data. So 3 retractions. So last year, I became interested in research in COVID-19 and the brain. And I looked on Retraction Watch, and in January, only 6 months ago, there were 36 COVID-19 papers retracted. Yesterday, there were 116 retracted. So look at number 76, noteworthy neurological manifestations associated with COVID-19 infection. That's what I was interested in. Published July 2020, retracted March 2021. Number 116, vitamin D supplementation could possibly improve clinical outcomes of patients infected with coronavirus 19. Posted 2020, taken down at an unknown date. Now you think, oh, that's terrible, but that wouldn't affect me. Well, retractions occur in neuroscience. This is from August 2020. P62 improves Alzheimer's-like pathology by increasing autophagy, published in Molecular Psychiatry, retracted in 2020, published in 2017, retracted in 2020. Retracted after an investigation by Arizona State University concluded that figures 1C and 4A had been manipulated and falsified by one or more of the authors. So beware. Beware, one, if you falsify data, someone will find it out. And beware, two, of citing papers with falsified data. So you do not want to see your name here. As of yesterday, this is the retraction watch leaderboard. It's like golf. Who has the most retractions? Here's our unofficial list. This guy, Fuji, has 183 papers retracted. It's hard to believe. Bolt has 155, Sato 105, and they have little comments on why they're all retracted. Iwamoto 81, Nazari 69, Stapel 58, down. This is just the top 10. They go on and on and on. So some of these people seem to be sitting in the pub drinking beer and inventing data at a rapid rate. So why does retraction matter to you? Well, number one, what if you're accused of fraud and have to retract your paper, but you're innocent? Number two, what if you cite a paper that's retracted? Should you retract your own paper? These have happened to my colleagues and me. So how do you respond if you're accused of fraud? This is a paper from 2010 by a guy named James Goldenring, who was accused of fraud. It's called Innocence and Due Diligence, Managing Unfounded Allegations of Scientific Misconduct. And just go to the underlined part in bold at the bottom. Scientists must safeguard their reputations by organizing and maintaining all original image files and data relevant to publications and grant proposals. So you need to keep your own archives, as I've said. Do you have an archive of your own publications? Well, as I said, we always kept an archive, but now we can't read it. People write and say, do you have the data for this paper? I say, yes, I've got it on this floppy disk, but can't read it. So the university has an archive, and now granting agencies are starting archives. Some of our data we did with the Jackson Labs in Bar Harbor, Maine, is on their Jack's Phenome Project archive, all the stuff we did with him. And so here's a case where my colleague, Vin Lolordo, was reanalyzing his own data, and he found he couldn't replicate his own data. And so he wrote a paper called Retraction of Ross and Lolordo Findings, and he redid his own experiments and found out where they were wrong. So if you're lucky, you can reanalyze your data and write your own retraction and explain the problems in the original paper. So you're kind of your own sheriff, so to speak. You've discovered your own errors and you fix them. That's the best of all possible worlds. And my other colleague, Ian Weaver, he wrote a review of a paper. So here on the left, with a photograph of Rudolf Uhr, is a commentary that they wrote on the paper in the middle, Childhood Treatment and Methylation of the Glucocorticoid Receptor. But that paper was retracted. Then they had to retract their paper because it was a commentary on that. So then people said, oh, look, you've got a retracted paper. You must have fudged your data. They said, no, we didn't fudge our data. We reviewed a paper and commented on a paper that had to be retracted. So when you look at the comments on the retracted paper, an investigation carried about by the University of Geneva concluded that one of the authors fabricated methylation data. A reanalysis of the DNA reveals no significant correlation. The original conclusions therefore no longer hold true, and we wish to retract the paper. So when you write a review of that paper, you have to retract your review. And what happened to us is we'd finished the paper and we were proofreading it for the journal, and I checked the references, and one of our references was retracted. So I had to go into the literature, to PubMed, Google Scholar and find another reference that said the same thing, or else we would have had to cut that reference out and cut that part of the paper out. So you need to check the references that you use and make sure they're not retracted. Now, it just keeps changing. So history affects all aspects of your research. The name of each part of the brain has a history. Your own research papers become historical documents. You need to keep a historical archive of the data for each paper so that it can be replicated and to answer accusations of fraud. And now there's starting to be many online databases, open access databases to store your archives. But I have shelves of data books that they're not anywhere but in my lab. So now we look at the vanishing history of neuroscience. Historical study is not the study of the past, but the study of the present traces of the past. What remains? If men have said, thought, done, or suffered anything of which nothing any longer exists, those things are as though they had never been. The crucial element is the present evidence, not the fact of past existence. And questions for whose answer no material exists are strictly non-questions. This comes from Elton's Practice of History, 1967. So things have to exist in order to be studied. So there's a nice paper written in 2010 on trepanation and Roman medicine, where they discuss the issue of how to study trepanation. The Roman Empire gave rise to an abundant and diverse range of source types, including skeletal remains, material culture, and detailed medical texts, each of which harbors the potential to contribute to our understanding of trepanation during this historical period. And there you see a brain, a skull with a hole in it. What needs to be saved to understand trepanation? First, the skulls, the osteoarchaeological evidence. Second, the tools, the material culture. And third, written text, sort of textbooks, how to do trepanations. Many things no longer remain. Bandages, ligatures, steel tools, all rot in the ground or rust away, so they're not going to be found. So there's the trepan skull of Chios. Trepan skulls provide rare evidence of the impact of medicine on a patient. The individual's history is unknown. You don't know who that patient was. You don't know whose skull that was or why they have a hole bored in it, but you see the skull and you see the hole. Secondly, you can find tools, ancient tools. As it says, even detailed written descriptions of surgical instruments cannot confirm exactly what they were like. Only finding them in graves or in places like Pompeii or in Egypt can you actually see what they look like. And then the third aspect is a written text. And here's Hippocrates, 400 BC, instructions on trepanning. Concerning trepanation, when necessity compels you to trepan a patient, here is what you must know. If you trepan, having undertaken the patient's care from the beginning, you should not excise the bone down to the membrane straight away, for it is not good for the membrane to be bare of bone and subject to damaging exposure for a long time. Otherwise, it may finally become macerated, and so on. It gives a complete how you, a young doctor in training, would begin to learn to trepan a patient. So the problem with written materials, and I'll tell you a story about that in a minute, the problem is that medical texts can add to our knowledge of features of the trepanation procedure as experienced by a patient that are not apparent from osteological samples of material culture alone. On the other hand, written texts have been described as the most important single source in understanding Roman medicine, but they can't be assumed to be free from problems of interpretation. So what you find in much historical literature is originally it was written in Greek, then it was translated into Arabic, then it was translated into Latin, then it was translated into French, then it was translated into English. And every time it's translated, things change. And I've been reading Pavlov. Look, I have my big book of Pavlov right here, my big book of Pavlov. I've been reading all about Pavlov. And the trouble is, every time you find a paper of Pavlov's, which was written either in Russian or German, if you have two translations, they're different words are used. And in reading Pavlov, I became interested in Russian literature, and I've been reading poems of Pushkin, and no two translations into English of poems of Pushkin are the same. You think, well, what did Pavlov actually write if you just keep reading translations that differ from each other? What did Pavlov really say? Only if you spoke and read Russian would you know, which I don't. So the vanishing history of neuroscience, what's vanishing? The present traces of the past, books, diaries, reprints, manuscripts, original data, equipment, methods and techniques, laboratory skills, and people, they're all vanishing. And so you need to come to the question of saving the history of neuroscience. So over Christmas, I read this book called Burning the Books. Gee, Christmas seems years ago, doesn't it? Well, over the lockdown and pandemic, I've been discovering all the books on my bookshelf. You know, you go to the bookstore and you buy 6 books and you read one and the rest sit on a shelf, and then you discover them in the pandemic. But well, one night years ago at Oxford, I had dinner with Richard Ovenden. And so I was keen to get his book. He's the librarian of the Bodleian Library. And he wrote this book on destruction of knowledge, destruction of books, either deliberately or by accident, and looks at political, religious, and cultural rationale for burning books. And just yesterday, when I was preparing this talk, it dawned on me that in the last year, fire has destroyed the oldest university museum in Brazil, at University of Minas Gerais, home to more than 260,000 works of folk art and ethnographic objects. And then in April this year, a few months ago, in Cape Town, South Africa, fire destroyed the University of Cape Town's special collections, including rare books, films, photographs, and records. So you think it's all safely stored in your archives, in your library, but you look at that poor fire destroys University of Cape Town library and realize none of that's ever going to come back unless it's in somebody's basement. So you have to beware of things like that. So why does it matter? As I said, the crucial element for studying history is the present evidence. No evidence, no history. Historical study relies on evidence of the past. History is memory. Loss of history, loss of evidence of the past means loss of memory. So where would you be without your memory? Loss of history, scientific Alzheimer's disease. And this is a serious problem. So historical research relies on present traces of the past. If there are no present traces, objects, tools, bodies, papers, the history is lost. It's important to save the present traces of neuroscience for historical research, and important to be able to match written text with equipment, methods, and procedures and physical evidence. Oh, things keep changing. So part 5, saving the history of neuroscience. How should we save the history of neuroscience, and what should be saved? Well, books in libraries, papers in archives, equipment in museums, raw data, lab books, letters, notebooks, histological tissues, anatomical specimens. And where do you find this stuff? Well, quite often it's in a room in a university, and some administrator who's not a scientist comes along and says, what's all this junk doing in here? Let's throw it all out and use this room for something important, like a secretary or an administrator. And so a lot of stuff just gets thrown out as useless old junk. If you look behind me, you see I've saved most of that stuff. So why not a history of neuroscience museum, which I tried to have opened in London for a long time, but they ignored me. So we started a virtual museum. So many libraries discard old or seldom used books. Look, I have Hilgard and Marquis, Conditioning and Learning, first edition, 1940, bought for 34 cents from a library Deaxation. You know, you can't find these books anymore, the first edition. So these are books I love that I downloaded. The Universities of Ancient Greece, written in 1910. The Genuine Works of Hippocrates, you can figure out the date. And The Dancing Mouse by Robert Yerkes, one of the first studies on genetic mutant mice, 1907. So they're online, you can download them. The University Archives has many things. The Dalhousie University Archives and Special Collections actually has a collection of ancient medical texts that were collected from the British Army and British Navy physicians. And many universities, libraries have archives and special collections, but they're kind of not listed. You have to find them, you have to ferret them out. Museums, this is a guide from the British Museum in London, the Museum of the Mind, Art and Memory in World Culture, is to remind you that museums are memory palaces. They remember the past. All this controversy on museums saving things that well, they would have been destroyed if they'd been left. Also, people vanish. They retire, they die, they vanish. You forget about them. So it's important to save their autobiographical memories of neuroscience. And Larry Squire started years ago the history of neuroscience and autobiography. I think there's 11 or 12 volumes now, maybe more. So we decided to start an online museum of neuroscience at Oxford years ago. There's the website, History of Medicine and Science, Medical Science at Oxford University. And it has a number of components. It has slides, it has objects, and it has lectures. And so it began with Sherrington's box, which was going to be thrown out, and my colleague Zoltan Molnar saved it. It has microscope slides belonging to Sir Charles Sherrington that he had, that was at Oxford since he retired in 1936, 21 drawers of slides that he kept from the 1890s up to the 1930s, and then others were added by his students and other people. They're used for teaching. So the earliest slide is from 1898 and the newest from 1960. That seems old to you, I suppose, but so it began with that. How can we save that and how can we share it with people? And so there's a typical slide drawer, drawer 10. And you see the note at the bottom, 9 slides sent to Sherrington by Ruffini in 1898. They're the ones with a solid black border and red borders. Anyway, so we scanned all these slides into a computer, and we use a program called Zoomify. And if actually you go to this program, it's like putting the slides under a microscope. So slide SHB 103004 is a Ruffini ending that we think Ruffini cut from his own finger. And there it is zoomed in on the left at the bottom and zoomed in even more on the right at the bottom. And I'll tell you, if you look at 20 different anatomy textbooks, you'll never find a slide that's that nice of a Ruffini ending. There's also equipment, and we used a 3D camera and photographed all the old equipment in the physiology and anatomy department at Oxford. And you can see there it says drag to spin. You can spin the pictures around and look at them at 3D. That is a model of eye muscles for moving the eye. And we have a series of lectures, a special symposium on understanding the cortex, all the history of medical sciences seminars at Oxford. Some I gave, some Zoltan gave. A number of these people have died. Marianne Forlenz's died since then, Ray Guillory's died. So we have them in perpetuity, even though they've passed away. I'm still around. This year, Zoltan organized the Willis 400th birthday symposium of Willis, Circle of Willis, and had a number of lectures, and we were supposed to have a big event in Oxford, but guess what? All online. And we started, in my class here at Dalhousie, we started a brain museum project. And the idea was that in order to study history, you need the objects, but in order to study the objects, you need to find them. So where do you find neuroscience objects? We said, probably in a neuroscience museum. So where are these museums? And we decided to start with Europe. So we made a website, which is gone now. I couldn't find it the other day. We kept it for many years. A Brain Museum Tour of Europe, and you click on a country, such as England, and it would give you some of the brain museums, King's College, University College, Queen Square Library. I'm supposed to give my history lectures in that Gord Museum of Pathology in King's College, but lately I've been doing them online. Anyway, the European Neuroscience Society, FENS, has taken over our European Brain Museum project, and now it's a global brain museum project, which is online in a partnership with the International Brain Research Organization. And if you go to their website, just type in Global Brain Museum in Google, it tells you we're all over the world where there's brain museums and different types of museums, anatomical models, biographical objects, instruments, etc. So our European Brain Museum project lives on. And it dawned on me late yesterday afternoon, just before I went home for supper, that I'm talking to people in India. So I should mention history of neuroscience in India. And I have a lecture on India and China. But most of the stuff that I found is anatomy, history of teaching anatomy in India, anatomy in ancient India, surgical instruments and surgery in ancient India. I found this paper from 1913, a short review of the history of ancient Hindu medicine, and a more recent one, neurosurgical training in India. So, some of you guys out there should work on a history of neuroscience in India or wherever you are. So how to make history relevant to students, integrate historical topics into neuroscience teaching and research using websites, artifacts, biographies, and classic books, introduce history section into neuroscience programs, and initiate history of neuroscience tours. This is my dream retirement job if I ever retire. So I now teach the history section at King's College London to their neuroscience students. Usually I go there once or twice a year, but as we said, it's all by internet these days. There are a number of books written in the last 10 or 20 years on history of neuroscience. The one in the upper left, Origins of Neuroscience by Stanley Finger, I swear by. It's like an encyclopedia. The other, History of the Brain from the Stone Age Surgery to Modern Neuroscience, Neuroscience, a Historical Introduction, and Minds Behind the Brain. They're all fascinating books. So the history of neuroscience needs to be saved. Museums, archives, and libraries need to keep objects of neuroscience. There should be a history of neuroscience museum. I think it should be in London, but we'll try. History of neuroscience websites can be used for teaching and research, and we can engage in history of neuroscience tours. This picture is from our first meeting on preserving the history of neuroscience we had in Paris on 23rd of May, 2017, and we wrote a paper from this meeting called Neuroscience Without Borders, Preserving the History of Neuroscience in the European Journal of Neuroscience. And so that talks about the problems in saving the history of neuroscience I mentioned. Maybe we should have the museum in Paris. Anyway, I added this yesterday because I've been working on this lately. How does neuroscience advance? A historical view of the future. So one argument is that neuroscience advances by new ideas, and another argument by new technology. And there's these two papers as examples. Some would argue that new tools and techniques are fundamental for new discoveries in neuroscience. That's Bickel. Others would argue that conceptual change is what drives advances in neuroscience. That's Parker. So that's the first thing I've been thinking about lately. The second is, suppose you have a new idea, everybody will tell you you're crazy and no one will believe you. So how does a new idea get accepted? And this guy, Charles Gross, in 2009, wrote a very interesting paper called 3 Before Their Time, neuroscientists whose ideas were ignored by their contemporaries. And these include Emanuel Swedenborg, Claude Bernard, and Joseph Altman, he mentions there. Another book I have at home that I'm very fond of is called Controversies in Science. It's Great Feuds in Medicine, 10 of the liveliest disputes ever. And two that I use in my classes and think about are Galvani versus Volta on animal electricity and Golgi versus Ramon Cajal on the neuron doctrine. I had Cajal's book here. I took it home, I guess. So all of a sudden, everything you read in your textbook becomes interesting because you say, was it part of a controversy? How did it come to be believed? And I'm writing a paper now on William McDougall, who was sort of chastised for writing a social psychology textbook when he says all human behavior was instincts. However, buried in the literature between 1898 and 1901 was his theory of how neurons communicated. He said they must communicate with some chemical, and he proposed one of the first theories of chemical neurotransmitters he called neuron. He said it might, of course, he said, I think that for the present it might be best conceived as a fluid, and I propose that this fluid shall be called neuron. It might, of course, be called the nervous fluid, or nervous energy, or animal spirits, or ether, but the name I suggest is preferable because it implies nothing beyond the fact that something has to be named. And of course, neuron turned out to be acetylcholine, which was not identified until 1914. And so I'm looking at all these old papers now on why it was completely ignored. You can hardly find a reference to any of these papers. But the miracle of the internet is you can look them all up and download them. So his neuron theory, he developed a theory of inhibition based on work by Sir Charles Sherrington in 1903 of how inhibition would occur. And it's very interesting because he said, well, you could have inhibitory nerves, but that would mean you'd have to have two sets of nerves, excitatory and inhibitory. And who would ever believe that? My theory is there's neuron, which is excitatory. And when you turn neuron off, when it stops flowing, it's inhibitory. And he had all these pictures. And it wasn't until 1950 that GABA was identified as an inhibitory neurotransmitter. So I say he backed the wrong horse, but he didn't. It was 50 years before someone else backed the right horse. He talked about this in 1903. So part 7 is sort of autobiographical. It's my adventures in the history of neuroscience. And this is a picture of me at the Royal Society in London on a day when they had a display of all kinds of historical artifacts. And you can see a little tiny microscope there, which was Hooke's original microscope, which he used to write his book Micrographia in 1665. And so it is, why do historical research? Why study history? It's interesting, it's illuminating, enlightening. You meet interesting people, you hear good stories, you make sense of your own research, and you get to travel to great places. But more and more, I'm realizing that if you don't know the history, nothing ever makes sense to me anymore without knowing the history. So how do you do historical research? The internet, PubMed, Web of Science, Google, libraries, archives, museums, basements and attics. You know, some professor retires and takes all his stuff home and it ends up in the attic and he dies and his children don't know what to do with it, or grandchildren. Interviews with scientists, interviews with students, colleagues and friends and relatives of scientists, and travel to the original places. It blows me away, I'm going to tell you this. So I give you 12 examples of my travels in neuroscience tourism. So the first was I was at a meeting in Thessaloniki, Greece, and I had my lunch in the museum across the street from the meeting. And the museums in Greece sell you exact replicas of their exhibits. So I bought an exact replica of Greco-Roman surgical tools from the Archaeological Museum. And the one I bought was broken. The glass was broken, which I said I wanted because I could take them out and show them to students. So there are all these tools that are exact replicas of tools used by Greeks and Romans 2,000 years ago. And some of them have etchings on them. And one has an etching of a snake, and one is in the handle in the shape of a mouse's head. And so you can, when you talk about ancient surgery, you can see, and students whose parents are physicians, they say, oh, my mother or my father uses exactly these tools today. The second was we went to a meeting in Milan, and so I thought I'd give a poster on Leonardo da Vinci. And we ended up having a Leonardo da Vinci tour of Italy. We went to the town of Vinci where he was born. We went to the house where Leonardo was born, supposedly, the Leonardo Library and Exhibit in Milan, the Leonardo Museum in Florence, Italy, and the Biblioteca Nazionale in Milan, Italy, where they had an exhibit of historical books. So we had the Leonardo da Vinci tour of Italy. You might think of Leonardo as an artist, but he was a scientist and a biologist, a physiologist before his time. Leonardo's notebooks, he never published his notebooks. He was working with a physician to do drawings of the human body and brain. And the physician died, and the notebooks became the property of Leonardo's assistant. They found their way to the King of France, and then they found their way to the Queen of England. So I had the adventure of, after passing many security tests, going into the bowels of Windsor Castle to the Queen's print room and seeing the original prints of Leonardo da Vinci. This one is from about 1480, looking at the ventricles of the brain. And I said, I'd like to have these prints. And they said, sign these papers and we'll mail copies to you. So you can't see my office, but I will just tilt up. You see on the back wall of my office up there, 2 Leonardo prints, and there's other ones up here that I bought as copies from the Queen's collection in Windsor Castle. So I have my own Leonardo museum here, if you ever come to visit. Thomas Willis, a famous surgeon in Oxford that my colleague Zoltan Molnar has written an article about. He wrote this pathology of the brain and nervous diseases. He wrote a number of books, and something keeps happening. There's this amazing book called Soul Made Flesh about Thomas Willis and the mapping of the mind during the time of the English Civil War in the 1600s. I'm going to read this because I love this. Every building in Oxford has an internal signature of smells. The incense burning in the churches, the roasted beans in the coffee house on High Street, the foul reek of prisons. But the strangest smells in all of Oxford can be found on Merton Street in a medieval 2-story house known as Beam Hall. Its odors are almost unbearable, a reeking blend of turpentine and the warm decaying flesh of dissected dogs and sheep, along with the aroma of a nobleman's decapitated and freshly cracked open head. The room where his body is being dissected is something between a laboratory and a butcher's shop. On a long table lies the corpse, surrounded by a crowd of natural philosophers. The owner of Beam Hall, Thomas Willis, leads the dissection. So I went to Oxford and they said, oh, our guest house is full, but the only place you can stay is Beam Hall. I was so ecstatic, I danced around. They thought I was crazy. Anyway, you see me with my friend Zoltan, and the lit room is my room in Beam Hall. It's still there. And then at Corpus Christi College, they showed me their original copy of Willis's book, which was actually in a vault. And you see the key they had to use to get into the vault. So I'm there with my book on Thomas Willis, staying in Thomas Willis's Beam Hall. That was exciting. I've already told you a bit about Sharrington and Sharrington's box. We did this about 2008. We discovered Sharrington's box of slides, and I worked in the Sharrington building and the Sharrington library, and there's Sharrington's desk and a picture of Sharrington somewhere on my back wall. Oh, you can't see it, it's too small. I have a picture of Sharrington. Anyway, we wrote a paper on Sharrington, Zoltan and I, as a result of this. 2010 it was published. And then the anatomy building at Oxford is called the LeGros Clark building. And I must say, I'd never heard of LeGros Clark. but he was very famous in his day in the 30s, 40s, and 50s as an anatomist. He studied the thalamus, he studied the hypothalamus, he studied the visual pathways. And then he became we also found a box of all Legros-Clark's slides that he used for teaching and research at Oxford and saved that. And the beauty of it is he made cards that showed the slide and described it clearly of what it was and why he used it in the magnification. And it didn't say which papers they're published in, but we found a few of those, but he used them for teaching. He also wrote textbooks in comparative primatology and physical anthropology. He was interested in evolution. And so studying evolution by studying anatomy. And then you've probably never heard of the Piltdown forgery. So in England in the early teens, they discovered a skeleton and it did not fit into any logical evolutionary sequence. And I remember as a student in the 1960s taking an anthropology course, and they had all this evolutionary theory, and over here they had Piltdown Man. And it turned out Piltdown Man was a forgery. People just took a bunch of bones and buried them in a gravel pit. And Legros-Clark was involved in analyzing the jaws and deciding the teeth had been from a primate, not from an ancient human. So this 1953, the solution of the Piltdown Man forgery was very exciting. Then a bizarre thing happened. We were in Oxford and my wife said, did you know that Legros-Clark had two grandsons, one of whom teaches at Cambridge? And so I phoned him up and we went to visit Cambridge. And he said, oh, you know that I have a whole pile of letters that my grandfather wrote to people in South Africa. Legros-Clark was known to be one of the first people who believed that evolution occurred in Africa and not in the Middle East. And people poo-pooed him, but all from the 1940s and 50s, 30s, all during the Second World War, he was getting correspondence from people in South Africa, and they had drawings and measurements of brains. This is from 1940 from Pretoria. So I have copies of all these, and I'm trying to get them organized and write an article about them and get them put into a museum. They're now in somebody's attic in Cambridge, his grandson's attic. They were all saved. So Donald Hebb was my entry into neuroscience research. Donald Hebb is famous for the Hebb synapse and Hebb cell assembly. He came from Nova Scotia. He was a student at Dalhousie. He worked most of his life at McGill. And I had owned as an undergraduate the green book on the left, The Organization of Behavior, but it was a paperback that was falling apart and I couldn't buy a new one. So I organized with Peter Milner to get it published. So there's my Organization of Behavior, and I wrote the introduction with Peter Milner. So I'm very proud of that. 2002, it's almost 20 years now. Anyway, Hebb was born in Chester, Nova Scotia, and at the top is a picture of him in a boat with his mother. He was a blonde-haired kid. And I used to say that I would take you, if you came to visit to Chester, I would take you and show you the house he was born. But a couple of years ago, I went there and it's all been demolished. So I can show you this picture. That's all. I can take you there for a beer. So we actually crashed his 80th birthday party. There's me in 1984 and Donald Hebb and my colleague Ray Klein, whose daughter there is now a mother. So we used to go there. My wife used to like to visit him. We'd go there like visiting your grandparents on Sunday afternoon. I wished I'd gone more often. Anyway, he wrote autobiographical articles, most of which were not published. I have copies from his family. And I'll read you this because it's rather interesting. An account of my life written for my daughters and grandchildren began in 1980. I used to be a psychologist. Once upon a time, I was a psychologist up to my ears in this stuff, psychology that is, with lectures and examinations and meetings and research. I was a professor for 25 years at a distinguished university, and now I'm quite detached in my own mind. I know quite well what I did and thought and wrote, but there's a feeling of strangeness about it, as if when I write about my life, it was someone else I'm talking about. Between me and my students, we made some important discoveries, exciting ones, some of them, and I had the pleasure of teaching some very bright and enterprising students and learning from them too. Now there is a sort of unreality about that time. Now I'm back in Nova Scotia, near the village in which I grew up, Chester, where my father and mother were country doctors. His mother was the third woman to get an MD from Dalhousie University. I live on a little farm where I work in the summer, cutting grass and bushes and planting pine trees and sugar maples. In the winter, splitting kindling and cutting hardwood for the kitchen stove, along with the inevitable repair jobs. My life is like a continuation of my life as a boy, cutting kindling and so on, as if the academic interval had never taken place. Well, that's not quite true, because he kept writing articles and books, up until he died in 1985. But it gives you a picture of someone, a famous psychologist, looking at their life. I did a lot of work on HEB, and during the war, people got Defense Research Board grants, and their research was all classified secret, as was this, and I had to go to the Defense Research Board and have things declassified. So not only do you have to look in archives, you have to look in secret government archives where things are classified and get them declassified. You see the fine print, declassified December 2005 at my request. Apparently they don't declassify them till someone requests them, but you can't find them till they're declassified. I knew it existed because I had HEB's copy. I've written many papers on HEB. You can look them up in PubMed. Most recent paper I wrote, I tried to follow every step he made for 17 years in organizing his thoughts on synaptic change and HEB synapses. And then I realized that I'd been working on papers on HEB for 17 years. A few years ago, I was invited to St. Petersburg, Russia, to give lectures in the Pavlov Institute of Physiology. And we were taken for tea in Pavlov's apartment museum. So that's me having tea in Pavlov's dining room with all of Pavlov's stuff around. They use this it's a private museum, and you can go in by appointment with the curators and Pavlov's family. It was near Easter, and so we have Easter eggs, Russian Easter eggs. And so our trip to Russia resulted in I wrote a paper kind of like, what did you do on your summer vacation? What did you do on your trip to Russia? We went to the 100th anniversary of the Russian Pavlov Physiological Society. And you see figure 1 on the upper left, that's the Pavlov Institute of Physiology. The upper right, Pavlov turned his summer cottage into a research center called Tushi. The lower left is Pavlov's living room with some of his art collection. And the lower right is one of Pavlov's laboratories in the institute they've kept as a museum. And so this is Pavlov's original laboratory and the apparatus he used to collect and measure saliva from classical conditioning experiments with his dogs. I visited them all. It gave me an appreciation of Pavlov, and I've been reading about Pavlov, and Pavlov says he was inspired by Sechenov. So I've been reading Sechenov, Reflexes of the Brain. And then it turned out that Ivan Turgenev, in his book Fathers and Sons, used Sechenov as a model for one of the characters, Barazov. So I've been reading Russian literature and poetry and I'm writing my paper called From Pushkin to Pavlov, Arts, Culture, Politics, and Science in Russia in the 19th Century. It is fascinating. And many professors taught in night schools for workmen, and since women didn't go to university, they taught in women's colleges as well. And Sechenov in the middle there is famous for studying reflexes with frogs, and so you see him there with his frogs hanging on a little balance. I occasionally give lectures in Sweden, and when I was in Sweden a few years ago, I was taken to the Medical Historical Library of the Karolinska Institute by my friend Abdul Mohammed, you see there with the cap on. And I asked the director about the earliest photographs, and he found me a French journal of medical photography. I asked them if they had any old books by Avicenna or Galen, and they took me to the old book room, and this is, they said, if you look in the upper right, they said, yes, we have Galen in Greek, Galen in Latin, Galen in Arabic. Which one would you like? And so there, look, they got 2 Galens there and an Avicenna sitting on their shelves, a book that are very hard to find. But of course, this is where I failed to study Latin and Greek. You go there and they're beautiful, but you can't read them because they're in Greek and Latin. You need someone to translate. In Uppsala, it has a famous university in Sweden, and they took me to the medical museum there, the whole history of medicine in Sweden. And I went to the university museum. And if you look at the upper left, you see a building, and they actually cut a hole in the roof and installed that cupola in order to have light coming into the medical demonstration room, which is on the right. Everybody stood and watched the dissection, which of course they could only do in the winter because in the summer the body's rotted. And you see anatomical textbooks used in Sweden in the 1600s in that medical teaching room. Oh, this is Venice. You know, I stayed a week living in this museum in Venice. I had my own rooms in the library museum. So it's the Scuola Grande di San Marco, Venice. It's an old monastery, which is now a hospital. And in the upper left, you see the building at the back. And the upper right, you see the museum room. And the lower left, you see my good friend Lorenzo LaRusso with a book that's as big as he is in the library. I spent a week in that library. And actually, Lorenzo just the other day sent me our manuscript that we're writing from that. And you see even Thomas Willis there. They had a special room of books written before 1600. You can't believe. There are all these tourists out there and they don't even know what's in there. I sent some in to look at the museum, but many of them thought I was crazy, I think, being so excited about an old museum. In France, when we had our meeting in France on preserving the history of neuroscience, a lady was there saying, well, she's the director of a museum, but it's closed. And all the museum is in shelves in the basement in storage until they rebuild the museum. And so she let us in after hours. And we went, and they had the bottles with the brains of Lelong and Laborde studied by Broca. And all those labels are written by Broca. And he published his famous papers on language centers of the brain based on those 2 brains. And we're writing a paper now on why it's important to save things like brains, because in 2007, some scientists took the brains and they scanned them with a scanner and MR imaging. And they said, well, Broca made a number of mistakes. He didn't cut up the brains. He just looked at the surface. And if you scan them, the equivalent of cutting them up, you can see different types of damage, some which was not reported by Broca. And this is one of the brains, Laborde, and this is the other brain, Lelong, that they scanned and then identified all the types of damage in a brain that's 200 years old, sitting in a jar in a museum that's closed that you can't get into, but I could because I met the director. So one of my passions lately is to identify the first photograph of the brain. And it's important because before that, people looked through microscopes and drew them by hand, but then people argued that they drew things that weren't there, missed things that were there, and that if they used photography, it would be 100% accurate. And I show this because one of the earliest scientific photographs were taken around 1860 by Charles Ludwig Dodson. So this picture, George Rolleston seated examining a skull, Charles Robertson standing center, Vernon Harcourt seated, and Haywood Smith examining the skeleton of a stonefish. Now, the important thing is the photograph is taken by Charles Ludwig Dodson, who is a professor of mathematics at Christ Church College, Cambridge. But he also had another life. He wrote books under the name of Lewis Carroll. So the first photographs taken at Oxford of scientific objects were taken by Charles Dodson, who also wrote Lewis Carroll. So a quiz question would be, what's the relationship between photography and Alice in Wonderland? So this is Rolleston's first photographs, but I think the photographs were taken by Dodson. On the affinities of the brain of the orang-utang, Natural History Review, 1861. So I was supposed to go to Oxford and go to the Natural History Museum. You see, it's from the Oxford Museum of Natural History where they're stored, and I communicated with the librarian, but I haven't been able to get there since the lockdown to look at the original photographs of the brain. And they were taken because of the evolutionary debate of whether the brains of humans and primates had the same parts or not. So the last section of my talk is the rise of neuroculture. And I have a lecture on neuroscience and art. So I'm going to take my shoe off. I know it's crazy, but I'm crazy. And so my old shoes, a friend of mine's an artist, I got him to paint neurons on my shoes. Just because I was tired of all the break. I was going to throw them out, but I decided to get them repaired and have neurons painted on them. So we talk occasionally about collaborating on neuroscience and art. The first collaboration is my old shoes. Anyway, there are all these neuroculture. This is a paper from 2009 on neuroculture in film, literature, television, visual arts, etc. And this paper from 2010 is a sort of philosophical paper on, are we living in a neuroculture? Everything is neuronovels, neuroeconomics, neuroethics, neuroanthropology, neuroaesthetics, neurolaw, neurophilosophy. And the brain is everywhere. And so I looked at some journals, and here's the need to better integrate neuroscience and neuroethics, a decade of neuroeconomics, scientific practice and neurophilosophy, current limits of neurolaw. So it's not that this is just in kind of novels, but it's in the real world in scientific literature. Well, I'm kind of interested in art and neuroscience because if you look at the upper left, every time you look at a drawing in a textbook, somebody's taken that brain picture and labeled it so you can learn the names of things. So one aspect of the art of neuroscience is just academic drawings in textbooks. The lower left, you see the Rembrandt painting of the anatomy lesson of Dr. Tulp, very famous painting of physicians giving public anatomical dissections in the 1600s in Holland. And so there's a whole art of neuroscience. And there's plastic models, especially in Italy. They began making plastic models of anatomical features for students to study and to understand. And one of my students discovered this guy. You must go to his website. It blows you away. He makes etchings in gold of the brain. And he's called Greg Dunn, The Art of Neuroscience. I've never met him, and I could never afford his works. I should get a poster, but isn't that hippocampus the most beautiful hippocampus you've ever seen? So I do like neuro novels, and there's a whole world of the neuro novel. Ian McEwan, Jonathan Lethem, Mark Haddon, Richard Powers, Ian McEwan again, Riva Galchen, all kinds of novels of mind and brain issues. I've got most of those. And when I found this book recently, just published a few years ago, The Elusive Brain, Literary Experiments in the Age of Neuroscience. And it's kind of interesting. They talk about neuro novels, but they also talk about brain memoirs, people who've written books about their own neurological damage. And they also include neurocomics. I thought we'd given up comics when I was a kid, but neurocomics. And so I realized I had a whole house full of neuro literature. And these are 3 brain books that I have. Tillman Spengler's Lenin's Brain. I thought it was a novel, then I thought it was a true story, then it turned out to be a novel about what happened to Lenin's brain after he died. Driving Mr. Albert is about the doctor who removed Albert Einstein's brain after he died and drove it to California in the back of his car, which it now resides at McMaster University in Hamilton, Ontario. And then Hemingway's brain. Unfortunately, Hemingway committed suicide by shooting himself in the head, so no one ever studied his brain, but they went through all the records of all his life and various types of brain damage he had. And he was in 2 airplane accidents in 2 days in Africa. One, they thought he had died. Anyway, it's an interesting sort of forensic anthropological study of all the brain damage of Hemingway and what may have led him to commit suicide. So even I found coming soon, it's the fall of 2021, a special issue of the European Journal of American Studies, a whole issue on the American neuro novel. I looked it up yesterday, but it's not out yet. Anyway, so we've come to the end of my talk. It's planned for an hour and a half at King's College, and they asked for study questions. So why is it important to save these type of historical objects? Equipment, books, brain slices, laboratories, and brains themselves. So that's the end of my talk for you on understanding history and why it might be important. Over to you.
Thank you, Dr. Brown. That was truly amazing. Everyone was sounding off in the chat saying we should build a brain museum in the United States if there isn't one already.
Well, there are various brain museums in the States. There's a big one in Washington, D.C., the Army Brain Museum, but they're all kind of hidden away and not easy to find, you know, but they're there. There's a fantastic medical museum in Philadelphia, the Mutter Museum. I try and search them out. University of Iowa Medical Museum is quite interesting. So I've seen a lot of them. Wherever I go, I go to find the local medical museum. Well, I gave you lots of examples, but it's sort of, once you've sat in Pavlov's dining room, look at my coffee cup, as well as neurons on my shoes. I have my Pavlov coffee cup that I got in Pavlov's apartment museum, sitting in Pavlov's dining room. You know, I've become a big fan of Pavlov. I have dozens of Pavlov books in here. Look, right at my feet, conditioned reflexes. So I'm trying to understand Pavlov and trying to understand these people, because, you know, you just don't, like Cahal. I've been reading Cahal, and Cahal wanted to be an artist, and his father made him be a physician, and he combined both art and the brain. And created all these amazing drawings. Been to the Cahal Museum. Anyway, it's the problem with being 22 is you're 50 years too young. You need to go and travel and see all these places and learn these things. Because when I started out as a graduate student, I didn't know anything. I thought I was a clever kid, but I didn't know anything. Even when I started teaching, I didn't know anything. The reason I wrote textbooks is because I was madly trying to put my notes together for my teaching. And I thought, oh, well, I might as well publish a book on this. And now the history stuff, but I realize that if I teach a class in learning and memory to my students, many of them have never been in a lab where they study learning and memory. I talk about Pavlov's lab as if they've all seen it, but how many of you have seen Pavlov's lab? 0. So I figure they should see these things, even if it's my photographs of my trip to Russia, and look at these things. And when we go, I had some students who went to Europe for the summer, and they got to Paris and they said, Dr. Brown, what's the museums you told us to see in Paris? We want to go and see them. So they tried to go and see some of the museums in Paris I told them about. So I've inspired a few of them, I think.
That's really wonderful. And I'm glad, thank you so much for sharing those photos too. It felt like you took us on a mini field trip with you.
Yeah, it's to realize that when you go to places, you can look up the medical museums or brain museums or interesting museums. And you know the problem, you go to the museum and you walk around and there's a security guard or there's someone there and you never talk to them. I talk to all the security guards and all the people there and ask them about all kinds of things. And ask dumb questions, and sometimes they take you in the back room and show you stuff they don't have on display. And I always try and buy the books. You can see I have mountains of books. I buy every book from everywhere because I'm reading about Pushkin, and I realize I went to the Pushkin Museum in St. Petersburg. So when I'm reading about Pushkin, I can look at my little book and look at the Pushkin's house museum. All these sort of things. And they come back, years ago when we went to St. Petersburg, we went to the Summer Palace, so I could find that book. Yeah, that's why I have such a mess in here. I never throw anything away. Anyway, it's great fun. So are these students here in your little seminar from all over the world? I'm assuming you're in India, but you're in the States, are you?
I'm in the States, yes. I think Intra, my colleague, was in touch with you. She's currently in India, but we have students coming from all over.
Oh yeah, I reminisced with her about my trips to India, to Mysore, and I found my book on what's his name, the king of Mysore who fought off the British in 3 wars. Oh, anyway. So I reminisce about travels. Whenever I meet someone from a place I've been, I have to talk to them about it.
That's really wonderful.
I don't know if you have questions. I mean, I've gone my hour and a half. Somebody might have questions, but you'll have to field them because Kyle's gone home and I have no idea how to look at most of these things on the computer.
No worries at all. I can definitely read them out loud. Anyone, if you have any questions, please feel free to drop them in the chat. Anyone on the YouTube live stream as well, feel free to drop those on the chat over there.
I was going to ask Intra how she found me to ask me to do this. You never know who's your stuff.
Intra, I think, is having some Wi-Fi difficulties, so I'm not sure.
Yeah, I'm back. But yeah, I think it was another team member that came by your profile. So I'm not entirely sure.
I think I might have stumbled across your Why Study History paper.
Yeah. Well, that's good. You never know who's reading your papers, do you? You throw them out there and you never know what's going to happen. Well, that's good.
They're out there. We're catching them.
Did you get any questions or anything?
Let's see. Leonardo has a question. How do you feel about the progression of neurotechnology in recent years?
Sorry, the progression of neurotechnology. I didn't hear the last words.
In recent years, how do you feel about neurotechnology progression?
It's a catch-22, isn't it? I call it the hula hoop phenomenon. That is, when I was a kid, there was the hula hoop and everybody had to have a hula hoop. And there are all these papers on the dangers of hula hoops and were hula hoops a bad thing or a good thing? And then the hula hoop craze is over and you're on to the next phase. So it's fads in neuroscience. As soon as someone invents a new technique, you have to use it for everything. And first of all, no one believes it. Then everybody wants to use it and it's the best thing since sliced bread. Then people critique it and then you evaluate what it's good for and what it's not good for. And it becomes part of it's like a microscope or a scalpel or anything else, just part of the armamentarium of doing neuroscience research. So they go through phases and there are always going to be new techniques. I mean, imagine if Golgi had never invented the Golgi stain. Cajal could never look at neurons and find out they had synapses between them. And so that depended on stains and microscopes. And the old microscopes of Leeuwenhoek and Robert Hooke weren't powerful enough to detect neuron endings. They looked like the neurons were all connected. We couldn't see the difference. So better and better microscopes made better and better neuroscience, better stains made better neuroscience. And now you've got these confocal microscopes, you've got imaging techniques where you put in a dye and you can see the brain cells light up when they fire. But the questions don't really change. You can read stuff from the 1600s about Descartes talking about animal spirits and just change the name to chemical neurotransmitters and they're talking about the same thing. I think some of the newest advances are in the glial cells, long ignored. All of a sudden, people find out that glial cells might be important. And in the immune system, interaction of the immune system with the brain and things like that, and different kinds of questions people are asking. And then finding the tools to identify the cells and to look at the molecular biology. And for a long time, I thought epigenetics was the solution to everything. I still think epigenetics is important, of tying genetic change to evolutionary to environmental stimulation and the interaction of environment and genes in evolution. So I think endless new techniques will come about, but the questions don't seem to change that much. I mean, you get mRNA and things like that, questions, and the technology to treat the brain. But still, people born with Down syndrome or autism, or people get Alzheimer's disease, and all this research, how much has it done to help them? You got some drugs, got some treatment, but they still have Alzheimer's disease or Down syndrome or autism, right? So there must be, it's not a magic potion. There must be something that we're all overlooking. And I sometimes say to students, what if everything we know is wrong? How would you start again trying to understand, say, Alzheimer's disease? And maybe I gave a talk once called, Is Alzheimer's a Disease of the Spleen? Because I realized that people took the brains out of Alzheimer's mice and patients and threw the body away, but the brain and the body communicate. And so maybe the Alzheimer's is involved in the guts or the spleen or the liver or the kidney. And there's all these nerves and chemicals communicating between the brain and the guts, which are often ignored. Now that people are on gut bacteria and things like that. But, you got to think outside the box. So somewhere there's a solution to Alzheimer's disease, and it may be hiding in plain sight, or it may be so obscure that you need new techniques to discover it.
It's a really interesting perspective, just different ways at looking at the same things.
Yeah. So I was impressed by the fact that so many, I call you all kids, you're all 20 years old. Are you 20 years old, 22? They're interested in history.
About, I would say 13 to 25 or so, maybe around that age.
25. That's fascinating to me that you're all so interested to spend your Sunday morning like this. So did you have anything else?
Celine here actually has a question. She is zooming in from Argentina, and she's asking, what advice would you give to young people who want to pursue a career in neuroscience? And maybe even drawing from your presentation today, how can we as young scientists incorporate more historical thinking into our work?
So I'm going to go and get something to show you, because I can't remember the title. So my friend Don Pfaff is involved in this book. Can you see it? Neuroscience in the 21st Century. And it's 5 volumes. And somewhere in the end of one volume, I wrote a paper called 7 Steps to Setting Up a Neuroscience Program in a Developing Country. Well, I wrote that about 2003 when I started to teach in Africa, and it's been published in a Nigerian journal. And we've just done a new edition of it for the 3rd edition of this book. But the things I put, it's called 7 Steps. And I can go to my own notes. And it involves things like setting up an undergraduate neuroscience program, setting up a master's PhD program in neuroscience, having integrated research facilities, ethics committees, national and international neuroscience student societies, national societies, and then organizing student societies and public outreach. So, people can email me. I can send them copies of the old version of this chapter. The new version is still in press. But it's on the fact that the students can take their education into their own hands, so to speak. At each university, they can start a student neuroscience society and things like this. I was blown away when I looked at your website. I thought, is this another one of these dumb requests out of nowhere that, so I'll look up their website, and there you were. I was so impressed with your website that you're doing all this stuff. So it strikes me you're off in the right way, and you can collaborate with IBRO, the International Brain Research Organization. They have a lot of student organizations with FENS, the Society for Neuroscience in the States has chapters, and you sort of slowly build. And You know, if students are in a university and there's no neuroscience program, but there's biology and chemistry and physics and anthropology and computer science and psychology, they can say, well, can we take one course from each of these disciplines and make a neuroscience program? That's what we did. So I'm trying to do in Africa, and we're starting slowly to put it together. But a lot of the old professors, they don't want to change. You know, they want to say, oh, we always did it this way. We're always going to do it this way. But if the students make a big fuss and a big noise and want to start their own program, you can do just what you've done. Ask people like me to give a lecture on some sort of neuroscience to your student society. They'll all be pleased and kind of chuffed to be asked, you know, by a student. A student actually wants me to give a lecture, not giving an excuse why they can't come or why can't they do their essay. I have students Sunday, and I've got at least 4 students working in the lab testing mice today. My students seem to be working away and not leaving the lab. I do pay them, of course, but they're getting their work done. So the student societies can be the leaders in some universities. And I wonder where this person is in Argentina. You know, I spent the gala New Year's Eve 2000 in Buenos Aires. So I have fond memories of Argentina. You know, it doesn't, it takes just a small group of students who get together and say, well, why don't we start these things? And who can we ask? What professors or what people can we ask? And then it starts. You know, we started our neuroscience program in 1989. Everyone thought we were crazy. But now it's been going, what's that, almost 30 years, 89, 99, 09, 19. It's more than 30 years. And I've been teaching in Africa since 2003, building up neuroscience programs. So it's really, if it's student-led, it really has a lot of clout.
Power to the students from the ground level up.
Power to the students, yes.
Yeah. Well, thank you so much. We're actually a little bit over time, so we do have to wrap
Yeah,
up.
I realize that I've gone on too long. It's my problem in life.
No worries at all.
I'm so excited to talk to people. You know, I taught my class for the first time last year to students that I'd never met. And then one day I was walking across the parking lot. 2 girls came running up to me and said, are you Dr. Brown? Yes, we're in your class. I never met them. You know, it drives you crazy. Anyway, thanks so much for inviting me. Ensure you're there. I'm waving to you. You have letters behind you, ADBURA. What does that spell out?
Oh, that's my sister's bed behind me. Her name's Madhura.
Ah, who's to know? You never know what's on the wall behind people. You see my office full of books and papers. Anyway, thank you so much for contacting me. It's been fun. I hope you learned something. You can always write to me.
Thank you, Dr. Brown. I'll drop your website link and contact information in the chat so students can reach out.
Okay, that's super. We'll sign off now, will we?
Absolutely. Thank you so much. Have a wonderful Sunday.
Oh, yes. It's football today, European Cup. Aren't we all watching the European Cup? Will Portugal beat whoever they're playing today? Who is it? Portugal versus... Oh, come on, someone must know. Portugal versus France. I forget who's playing today, but it's Portugal this afternoon. I want to watch.
See, we might take your recommendation and I'll tune in.
Okay, see you. I look
you,
forward to
Dr.
hearing
Brown.
from some of you. Bye-bye.
Before you go,
Thank
Dr. Brown, I wanted to take a picture of everyone. So if we could just take a minute for that.
Smile. Smile into the camera.
Everyone turn your cameras on and smile. And should you want to give us a countdown?
Yeah, sure. 3, 2, 1. I'll just do a quick video as well for the Instagram. So if everyone can just wave.
Good Arvin there. He's got his tie on. He's all dressed up. Where are you sitting, Arvin? Are you in India?
I'm in New Jersey, in the US.
And you've worn your tie to the meeting.
Yeah.
How great is that?
Okay, that's good. Thanks, everyone.
Okay, bye-bye.
Thank you so
Thanks,
much, Dr. Brown.
Dr. Brown.
And I will pull up the slides for the brain break intro slide. Cynthia, over to you. I believe you should have co-host permissions.
Yep. So hi, everyone. We're now going to be starting our brain break. Ria, do you want to go over all of the rules?
Yeah, so just a couple of housekeeping rules that we're going to play hangman. So Inshira will write down a certain number of blanks on the Zoom whiteboard, and you guys just have to guess letters in the chat box. And the correct letters will be filled, and the hangman will be drawn in part for every wrong letter. So please put all letters in the chat, and please stay on mute. Just keep in mind that this brain break is totally optional and is fun. And it's just a way to take break from the event. So there are no prizes. But we hope you have fun. And after you guys guess the letter, I will just say the definition and write it in the chat. So you guys have some neuroscience facts with you.
So everyone, quick note that if you would like to take this time to hop off of Zoom for a second, because we have been on for a while, get some water, feel free to. We will be back with Dr. Z's speaker presentation in about 17 minutes or so. So up to you if you want to hang out or take a break.
Can everyone see my whiteboard? Okay. So our first word has 1, 2, so 10 letters.
So the letters that we have right now in the chat are A, E, and I.
Okay, I'll put those down.
Okay.
Any more?
And then O, U.
So that's the hangman. Both those letters were wrong.
N, R, and S. So we have a guess of the word in the chat. Someone guessed adrenaline.
Yep, that's correct.
Okay, and we will also be putting the meanings of the words in the chat. Yeah, thank you, Ria.
Okay, so the next word actually has 2 words. So we have 5 letters and then a space and then 6 letters.
So we have A,
Okay, no A.
E,
Right, we have an E,
I.
no I.
Okay. O,
Okay, we have some O's.
R. Someone guessed sleep apnea as the word.
Nope, that's wrong.
Okay, the next letter we have is S,
That's also wrong.
L,
Nope.
N.
N?
Yeah.
Okay, no.
And then we have a few people guessing motor cortex as the word.
Yep, that's correct. Okay, so our next word is 15 letters long. So it's just one word, but I had to put it in 2 lines because I didn't have space.
So we have O.
Okay.
Then we have AEI.
Okay. No A. Oh wait, there is an A, sorry.
Yeah, there's A. I was just gonna say.
A. Did you say AEI?
Yeah. Then we have U. Okay, and then we have T. Okay, now I'm still waiting for more letters.
Okay.
Oh, I think Stephanie asked how much time we have left. So we were like a little bit behind on time, but the brain wake will end at around 1 p.m. EST. So about like 10 more minutes. Okay, so some of the next letters that we have are N. Then we have R. Then we have L. Then we have M.
M.
Mm-hmm. Okay, then we have H and P. And then we have a few people guessing that the word is neuroplasticity.
Yep. Okay, so the next word has 10 letters.
N. E. R. S. O. T. V. A. I. P. And now we have a few people guessing that the word is perception.
Yep, that's correct. Okay, so our next word has 9 letters.
We have someone guessing E. N. R. I. Then we have O. Then we have S. Then we have A. And then we have a few people guessing receptors.
Yep, that's correct. Okay, so the next one is a long one. It's 2 words, 10 letters, and then 7 letters.
So we have someone guessing E. A.
Oh, wait, give me.
Then we have R. And then we have someone guessing that the word is Parkinson's disease.
Yep, that's right. Okay, I'm not
Good
gonna write
job.
it down.
Yeah.
Okay, so next one is 9 letters and then 4 letters, 2 words again.
Okay, so we have someone guessing E. Then we have R. Then we have I. S. D. P. H. N. Then we have someone guessing Wernicke's area.
Yep, that's right. I think we'll do one more and then stop. It's just one 11-letter word. I didn't have space, so I had to put it down.
Okay, so we have E. Then we have A. Then we have N.
1 second.
Then we have P. S. I. E. We have someone asking if this is one word, and it is. We just didn't have enough space to put it in like one line, so there's a few spaces below. Then we have R. Then we have T. Then we have someone guessing L. G. We have someone guessing angiography.
Yep, that's right. Okay, I think that's all the time we have today, but thanks everyone for playing. This was really fun.
Yes, thank you all for joining. And we just wanted to let you guys know that the definitions that we found were from the Dana Foundation. So Ria, can you just send the website to that in the chat? Okay, so it's in the chat in case you guys are wondering.
Awesome, thanks guys. Next up, we have Dr. Z, who's going to be presenting. I'm not sure if they've hopped onto the Zoom call yet. So we might hang out a couple minutes, wait for them to hop on. How are y'all's weekends going so far? Any fun plans? I know it's summer vacation for half the world, but not for the other half. So who's in school right now versus on summer break? Anastasia's in school. London, Irvin, Carpenter, Fatima all are on summer break. Yeah, if you're working, there's no summer break. Vishnu's in school. That's true, Davis. It's like you're in school, but not school. So got a nice blend going there. Weekend is your break, indeed. Thanks for joining us on the weekend. So they're asking any summer plans for folks who are on break? Yeah, 4th of July is coming up pretty soon. Thanks for your patience, folks. We're just making sure that I'm just in touch with our speaker. In the meantime, I can put some music on, and you can also take a quick Zoom break. Those are always fun. I see my
If
favorite.
we want to do a couple more rounds of hangman while we wait, that's fine too, because we have some more words.
Oh, yes, let's do that then.
Okay. Siddhi, are you still here?
Yeah.
Okay, cool.
I think Ria had to go, but I can like send the meetings and things in the chat. Let me just open up the document.
Okay, sure. Okay, so the next word has 9 letters.
Okay, so we have someone guessing A. Then we have I. Then we have E. We have O. We have U. N.
Sorry, N.
Yeah. Then we have Y. We can give you guys a hint. It's a hormone. Okay, someone guessed serotonin.
Nope.
Okay, then we have someone guessing melatonin.
Yeah, that's right.
Okay, I will send the meaning in the chat.
Okay, our next word also has 9 letters.
Okay, so the first guess we have is E. Then we have I.
Nope.
A. O. N. S, T, L, this word is a little bit difficult, so we can give you guys a hint. It has something to do with like the sense of smell. F, and then we have a few people guessing olfactory.
Yep, that's right. And I think our speaker is here, so thank you all for playing.
Okay, great. I will just send the meeting and then we can move on.
Awesome. Thanks, folks. I'm going to go ahead and share my screen real quick to introduce Dr. Z. 1 second. Bio. Alrighty, so get excited because we're going to be learning about neuroengineering. Dr. Z is a neuroengineer with equal interests in creating accessible science, outreach, and education with a neuroscience focus, as well as exploring and describing the neural processing of animals, specifically mosquitoes and flying snakes. Outside of science, Dr. Z's diverse interests range from cooking and bread making to woodworking, from musical performance to graphic design, and various athletic endeavors. Dr. Z holds a BS in Biological Sciences, Bachelor's in Sciences in Biological Sciences from Cornell University, received her degree in 2007, and then later received her PhD in Neurobiology and Behavior from the University of Washington, Seattle in 2015. Thank you so much for joining us today. I'm going to go ahead and pass it over to you, Dr. Z.
Okay, good. I couldn't unmute before. Hello, everyone. Good morning. Sorry for my delay. I keep sending your emails to spam, even though I marked it as not spam several times. So I apologize for that, but I'm really excited to talk to you guys today. I'm going to talk about a little bit about my work, my research, and about where I see the field of neuroscience going, given my, oh boy, how long have I been doing neuroscience now? Oh gosh. We're going on around 15 years of research. So I've been in the field for a while, but I'd rather just use my talk to explain exactly what I have going on. So just give a few minutes here to get everything together. And I don't have the best of service out here. I live way out in the middle of the woods, so I don't have very good internet service. So I will be turning off my camera for the talk because it seems to improve the quality of the slides, which I think is a little bit more important than my face. So let's hop on over. I'll share my screen. Oh, hold on 1 second. Let's come over here. And let's do a couple of little things. Okay. All right. So thank you again for having me today. I'm really, really excited to talk to you guys about neuroengineering. I'm a firm believer that neuroengineering is the very future of neuroscience. I don't think that we'll be able to ask very many questions from here on out, or have been able to ask any questions without incorporating some considerable degree of engineering in our neuroscience prospects. So I'm Dr. Z. I am a neuroengineer. I have been doing neuroengineering research for the past 11 years now. As you guys heard in my introduction, I have a bachelor's from Cornell in biological sciences with a focus on neuroscience, and I have a PhD in neurobiology and behavior from the University of Washington. I've done a lot of research since then, but I've landed as the STEM outreach director for a creative tech institute called the Atlas Institute at CU Boulder. And there I'm developing neuroengineering kits to help prepare our next generation of thinkers for the inevitable future. I also really love snakes. Here's a picture of me with my ball python. And this is relevant because I'll be talking a little bit about my snake research today. All right, so many of you, if you're in this room, you're probably maybe you have an answer, but you're likely wondering what the heck is neuroengineering? Am I just making up words? Yes and no. Neuroengineering is the merging of neuroscience, device development, computation, and mathematics in order to explore, understand, repair, or augment neurological processes and systems. So anytime we're making a device that interferes or intersects with our nervous system, we are actually engaging in neuroengineering. So yes, this means that neuroengineering has existed long before the term neuroengineering was coined. Neuroengineering at its heart is multidisciplinary, as is neuroscience. We're just taking a little bit more of a practiced and organized way of looking at how engineering is applied into neuroscience. So there's a number of different fields that coincide to create neuroengineering. For example, when we add electrical engineering and computer science to neuroscience, we think about developments such as novel recording devices, like this transcranial magnetic stimulator that you see here. With this TMS, we're able to turn on or turn off large regions of the brain, which has a lot of therapeutic value. If we look at intersections of computer science and biomechanics, we start to think about things like brain-computer interfaces, where we take recordings of the brain, either external with these EEGs shown here, or internal with electrodes, and we send these signals to computer programs or devices. So I'll talk a little bit about these guys today. When we add in material engineering, then we start to think about new designs and interfaces for medicines. For example, there's a direct contact patch, which can be applied to open wounds and directly stops the sensation of pain. Not only do you have to think about the material that would have to work with the elasticity and movement of the skin, you also have to think about the neurological implications of stopping signaling from free end nerves in your arms, for example, or your extremities. And then if we think about nanochemistry, then we get into the really cool futuristic stuff like neural tissue printing. So here we're able to make complex tissues by aligning different groups of cells together and actually producing them. We just make the cells and put them together to create particular types of tissue. Neuroengineering is a rapidly expanding field, and there's 3 areas that I can see that are contributing considerably to what neuroengineering looks like and will be. First, there's neuroscience. Neuroscience is largely dictating the kinds of questions that we need. So basically outlining the demands for the engineering tools that we need to answer our questions. Then there's biomedical engineering, which is providing the devices and the outlets for us to communicate and record the brain and the nervous system. And then, curiously, we also have big tech that's been contributing to neuroengineering in a number of different ways, and we're going to talk about each of these today. I'm going to start off by talking about neuroscience and using my research as a model for how neuroscience is lending to the progression of neuroengineering. Then I'm going to give examples of how we're seeing neuroengineering used today in the field in biomedical engineering. And then we'll talk about where we see this field going by looking at the influences that big tech has on our lifestyle and our well-being. So I'm actually one of the world's first neuroengineers. When I was working at the University of Washington, I was doing neuroengineering without even knowing it. I did my PhD research on mosquitoes and how they use warm air to track host prey like humans. To do this, I had to do a couple of different things. Mosquitoes don't like to fly in wind. If you are living in tropical areas like where I grew up, you kept a fan on your bed because that kept the mosquitoes from getting too close to you. So it makes it really hard to use a wind tunnel where we can look at flight easily and repetitively. So I had to design a low-speed wind tunnel that allowed for laminar or easily repeatable flows to happen in this wind tunnel. I would release the mosquitoes downwind and they would fly upwind toward one or of 2 of the heaters, one of which was on or both were off. These had to happen at very low speeds, somewhere between 0.2 and 0.5 meters per second. It's incredibly difficult to create a wind tunnel at those speeds, I just have to say. I tracked the flights of these mosquitoes using cameras, and I programmed my own computer vision software to track the mosquito flights. About a year after I defended, all of these computer vision tools came out that would have probably cut about a year off of my research time. I also looked at their trajectories, their flights, and looked at their kinematics, the kind of speeds, accelerations, turns, things like that, that they exhibited during these flights. And I used that to create a computational model that mimicked how those mosquitoes would fly through the wind tunnel. This way, having a model, I was able to look kind of inside the black box a little bit and start to make some guesses as to their decision-making strategies as they're navigating in this world. What we found was that mosquitoes don't really have a prescribed way of flying. They don't make really clear turns. They don't really go directly upwind. They kind of meander and bounce all over the place. Their flight was actually best approximated by something called a random walk, where every step and rotation for in between movements is randomly chosen. This creates a path that looks a little bit like the one that you see here on the right, which you can imagine a mosquito flying a path like that, right? So these guys fly in this erratic and hard to predict flight, and we think that this is a trade-off. Yes, it would be a lot faster to fly directly upwind, but direct flight is easily predictable. So if your host prey spotted you, you would probably be dead. So these guys make a trade-off by sacrificing the efficiency of their flight to avoid detection. All of this work was done in conjunction with the world's first institute for neuroengineering called UWIN. This stands for the University of Washington Institute of Neuroscience. UWIN is still functioning today, and a lot of their research is pretty cool. I'm actually going to talk a little bit about some of the more modern, more recent work that's come out of there in this talk. So I'm gonna pause my screen sharing here and just chat with you for a second. So I wanted to talk to you guys a little bit about my interests. So I'm really fascinated. Once I started programming with my grad work, I got really interested in how we can actually provide these tools a lot more quickly and cheaply. A lot of the programs that we use for animal tracking or to record behaviors or to sort spikes if you're recording from neurons are owned by companies and really, really expensive and tend to not work exactly the way you want. With this generation of the maker boom and everybody doing it themselves, a lot of DIY stuff, it made a lot of tools become accessible. So I started to look at what tools were in the maker world that could come into the scientific world. And one of the big ones that I stumbled across was Unity 3D. Unity 3D is a platform for designing 3D and VR games and experiences. Shown here is the Book of the Dead, which is a hyper-realistic game. This is actually a screenshot from the game. It's not a photo. And if you look at the gameplay, which I highly encourage you to do, I have a video set. I can send you guys the link, but you're not going to be able to see a very good quality if I were to try to share it here. So I'll make sure I drop that link in the chat afterward. But it's hyper realistic, and there's wind that blows through, there's lens flare from the sun, and the players are encouraged to just walk around and explore this very, very detailed world. So you can imagine having a detailed world like this catered to different organisms and how they move through the environment could be a pretty powerful tool for research, especially in animal behavior. And Unity 3D has great potential for exactly that. It's free for beginner users and educational use. For the scale at which we would use it for research, we don't have to pay for a license. If you're new to Unity 3D, there is an extensive library of tutorials that are accessible and free. And the best part, the thing that cinches it, is that Unity 3D, the objects in it, and all of the animation, controls, etc., can be programmed through independent computer programming, which we call scripting. This allows us to incorporate precise positions, behaviors, and velocities, things like that, into our code, which we need in order to capture the precision and accuracy that we want for science. So Unity 3D has a great amount of potential, and I'm now going to show you how I did an entire suite of experiments using a gaming platform. So I study flying snakes. These are snakes in the genus Chrysopelia, and they're found in the Malay archipelago, although their range extends up into the southern tips of India and China as well. They're pretty small and they're pretty shy, and they're actually the fastest snakes recorded on land. When they glide, they hit speeds of up to 11 meters per second. The second fastest snake is the black mamba, which clocks in at a mere 9.5 meters per second. So these guys need to see a lot. And if you take a look at them, they have these gigantic eyeballs. When you think of snakes, you don't really think of eyes, right? You think of long bodies. You don't really think of, oh, they got these big frog eyes sticking out of their heads. So I was really curious as to what they were able to see. To ask this question, I conducted inverse telemetry using Unity 3D, and I'll explain what inverse telemetry is as I walk through the process. So here's a live snake, which I took to turn into a 3D digital model by scanning their heads using a laser scanner. Here is the resulting model, which is accurate down to approximately a micron. I took that head and I placed it in a 3D world, and I fit their eyes with spheres so I can get the precise position of their eyes in the world. I then used a laser that moved all around every single point on the sphere around the head, and I pointed it at the eyes. So the laser is always trying to see the eyes as it moves through the world. Everywhere that the laser was cast is shown in blue. So we're showing a little bit of an array of lasers to imitate how light moves through the environment, natural light, sunlight moves through the environment. And any place where the laser successfully hit the surface of the eye, we call that a line of sight. And we can determine then where we see binocular areas like we're seeing here, monocular areas, which is where one eye can see, and blind areas as well. And we can calculate this for the entire 3D sphere. So let's take a look at what that data ultimately looks like. So here on the left, we're looking at that sphere that I had mentioned, comparing males and females. These guys have a pretty strong sexual dimorphism where the females are considerably larger in head and body than the males, although their eyes are roughly the same size. So what we see in the sphere is the position of their snout, which is projected onto the surface at these blue triangles. This is just to help you orient to where their head is facing inside of the sphere. Everything in red shows their binocular region, and everything in black shows their blind region, so places that the lasers did not contact the eyes. All the other vertices that are unmarked indicate monocular or single eye areas. If we take a horizontal slice through the sphere, we're able to look at where the amount of overlap is in their binocular regions and their blind spots as well. Because of this sexual dimorphism, the difference in the size of their heads, the females have a considerably smaller binocular region, and the males have a quite larger binocular region, but their blind spots are roughly the same size. If we take a vertical slice through this sphere, we're able to see that the females have an upturned field of vision, so they can see what's above them a lot more readily than they can see what's below them, whereas the males have a much wider range of view. This is probably an artifact of evolution, as the females tend to be larger and more sedentary, and the males tend to be more active and more likely to glide. So these guys, the females are going to be looking more for predators, while the males are going to be looking around their environment a whole lot more. We also had suspicions that these guys were in the middle of the food chain. They are predators as well as they are prey. And the size of their binocular region, first, the fact that they have a binocular region suggests that they're predators, but the fact that it's small suggests that they are prey as well. So by looking at certain features of their visual system, we're able to confirm little known information about their natural life, what they do day to day, and how they live in their environment. I didn't want to finish there. I wanted to continue to use this gaming platform to see what we can examine in visually mediated behaviors. So using virtual arenas or virtual reality to explore these visually mediated behaviors is not new. This is actually work that was done by a postdoc in my lab at University of Washington, where he took a hawk moth, which is a really big moth, and tethered it to this device that can detect how much torque or rotation the moth was exerting onto this post, and then use that information to update a world that was projected onto a screen. So we could take a quick look at this video right now and see what it looks like. So here we see our tethered moth flying, and as it moves around, there are trees, and it's trying to avoid these bright trees. It's eventually going to fly out of the forest right now, And it doesn't like that there's not a lot of visual information, so it turns around and goes back into the forest. So just by looking at 15 seconds of a simulation, we're able to glean a lot more information than we would if we were doing something called open loop, where we just present the data or the stimulus to the animal and don't update the stimulus based on how the animal responds. By looking at this, just 15 seconds, we're able to discern that hawk moths like to keep a lot of visual information around them. They don't like the lower visual information of being outside of the forest. So I wanted to do a similar thing with flying snakes. We know quite a bit about birds and bats, but we don't know that much about flying snakes. And virtual arenas are really powerful tools because we can potentially make naturalistic scenes like we saw in the Book of Dead, the Unity game that I showed before, and we can capture lifelike speeds. For example, flying snakes see really high speeds, about 120 hertz. We see at about 50 hertz. So they're seeing about 1 to 2 times faster than we see, but I can create a stimulus that works for them based on what I'm able to do and control when I make these virtual setups. And finally, we can make these things closed loop, where the organism is controlling what's happening in the scene. So if the organism moves to the right, the scene will also move to the right accordingly. This allows us to ask, how do animals explore different scenes, and what visual events are preferred? To give you an example of what I mean by visual events, I'm going to talk about some research that was done in 2010 by Mike Reiser and Michael Dickinson. So here they worked with Drosophila or fruit flies and created 2 attractive stimuli. One is a stripe, which is, when it's just a stripe on its own, Drosophila don't like it because it basically to them looks like a giant predator. So the stripe, but the stripe with movement when they're flying is a very different thing, because now it could be used as a navigatory tool. For example, trees appear to fruit flies as dark vertical stripes. So they had 2 stimuli. They had this open rotation where it was expanding from one point. So they would have LEDs that would turn on and rotate around to the back in both directions. And they had the same animation, but with a dark bar imposed over it. What they saw was, as they increased the speed of this expansion, this rotation that was happening around, they would go from an aversive behavior where they're moving away from the stripe, shown here in these 2 arcs that you see. So this is where the stripe is, and this is where the center of this is this point, right? This is the back up here, is kind of backwards, and this is the front. So this is where the stripe is. So they're moving away from the stripe when the speeds are low, but as you increase the speeds of this rotation around, and it's going to feel to the fruit fly like it's moving. And once it starts to feel sufficiently like it's moving, it changes its behavior and flips 180 from flying away from the stripe to flying toward the stripe. And they see that this tendency to fly toward the stripe with the movement of the expansion around it wasn't affected by contrast levels. So if you had something that was black and white, or you had 2 versions of gray, it didn't really make an impact. Mostly what you cared about was how fast this was moving. In other words, if the animal is moving and you have this expansion, these lights that are moving around the side of the head, then stripes are avoided until you hit a particular speed. But once you have that expansion, the stripes are focused upon. So we can see from these types of immersive animal controlled or closed loop experiments that visual events can change in preference based on the state. So if you're standing still and you're a fruit fly, vertical stripes are scary, but if you're moving or have the sensation that you're moving, vertical stripes are helpful. So, okay, how does this work in reptiles? Again, these guys are pretty rare in the US, and they're rare in research in general. So we don't know very much about their visual world, what they can see, how fast they can see, how fine they can see, let alone if virtual reality works for them. So I built an immersive virtual visual arena to help these flying snakes explore virtual scenes. The arena has a couple of key components. First, there was an air table, which is like a beefed up air hockey table that minimized the amount of friction that the snakes had on the surface. This allowed them to undulate in place so they could locomote but not move forward. And then I put translucent vinyl around all of the walls onto which I back projected an image which came from projectors. I then tracked the animal using a Leap motion controller. This is a fairly affordable motion controller. They run about $80 and they have 2 cameras in it, which allows me to triangulate distance and calculate speed with built-in code. I then tracked a particular marker on these snakes so I can get very precise data about where they are in the arena as they're looking at the scene. These snakes are pretty small. The size of their head is probably about the size of my pinky. And they really don't like things on their head. So I had to come up with a device that was going to be small and lightweight enough that we could track it, but big enough that the camera could see it and it wouldn't disturb the behavior. So I would follow the shape of this asymmetrical tracking object where one dimension looks different from the other dimension. And I used something called computer vision template matching to find where this was and the orientation. And I used size scale to determine how high up or low the snake was in the arena. I then sent this position data into Unity 3D to send the scene out to the virtual reality camera, which was then projected onto these projectors. I gave the animals a number of scenes. The most important thing that I showed them was a replication of a physical experiment that I had done in something called an optomotor drum. An optomotor drum has vertical stripes on the inside and it rotates around. And as you induce this rotational view, it causes a reflex called nystagmus. And you can look for the strength of that reflex as a measure of how well the animal is able to see the scene around them. So in order to determine if reptiles were viable candidates for VR, I first had to make sure that they would respond to the digital stimulus the same way that they would respond to an analog stimulus. I also put them in a forest where I could adjust the amount of fog, which would allow me to explore how much depth they needed to avoid collision with these trees, much like what we saw with the hawk moth earlier. I had 2 other scenes that I didn't really get to experiment with in detail, but these were pretty fun explorations all the same. The first is the infinite tunnel, which is an ever-growing pipe. The camera is placed inside of the pipe and moves along the center line of the pipe. So it feels like to the snake that it's moving through a giant tunnel. With this information, I can get input about how they change their posture in response to upcoming turns. I also made what I call the warp field, which is the scientific term for that is an optic flow field, which is looking at how dots move coherently through a scene, which is how many sighted animals determine movement and calculate speed. So let's take a look at how these animals performed inside this immersive arena. I ended up having to use an acrylic, visually clear acrylic container because these guys are excellent climbers and movers, and our Airtable really didn't help keep them in place very much. So what you're going to see here is the snake looking at the scene around them, and the scene's going to update as the snake moves its head around. Particularly, I want you to pay attention to when the snake has its head over here, because it's going to engage in that visual reflex that I had mentioned, nystagmus, which works to keep the visual scene fixed in place on the retina and allows the visual system to gain as much information about the scene as it possibly can. So if this animal is doing nystagmus for the purpose of gaining as much information as possible, we should expect that this rotating scene would be held still while they're engaging in that behavior. So you're going to see this rotation along the outside. I do want to give the warning that if you are light sensitive, this might be difficult for you to watch. So I'll let you know when I play the video and when it's stopped. You might want to close your eyes for it though. So here's the snake. Oops. All right, so here we see the rotation along the outside. It's very curious. When snakes are excited, they'll tongue flick a lot, so it's very engaged in its scene. And now it's going to do those few little nystagmus movements and hold the scene steady. It happens pretty quick, so I'll show it a couple of times. So it moves over to the opposite side. And there we have the nystagmus, and the scene is being held still. One last time without any narration so you can see. Cool. So by using an immersive arena, we were able to find that these snakes were able to respond to visual scenes, visual scenes, and that nystagmus works in the way that we anticipated. So we can confirm that we can not only replicate the behaviors, but their functions as well using virtual reality. We're also able to glean a whole lot more about their visual system by doing these series of experiments. On the whole, we know now that flying snakes have high speed vision, so they can see very high rates, but they have very low resolution, so they can't really make out a lot of fine detail. To do this, to make these measurements, we looked at the number of vertical stripes that could fit into a single degree of the visual field. We call this a cycle per degree. To give you a sense of scale, if you take your thumb and you extend it arm's width, that's about 3 degrees. The width of your thumb is about 3 degrees of your visual field. So we're looking at how many stripes can fit into 1/3 of the width of your thumb held at arm's distance. We gave the snakes a number of different regimes: hard, where the stripes were very, very narrow; medium, where the stripes were wider; and easy, where the stripes were very wide. Then we calculated what their cycle per degree was, which varies based on distance. If you're far away, you fit more cycles per degree. If you're close up, you fit fewer cycles per degree. And we're looking at how these guys are going to perform. How well do they match the rotation of the drum with the rotation of their head as we go from an easy spatial frequency to a hard spatial frequency? And what we see are these very broad curves. So we look at this orange curve here. There's a nice subtle green curve, a red curve there that are all going. So we're looking at different rotational angular velocities of the drum, which is also impacting how they're processing their scene. And we're seeing that we have these very broad curves. Not exactly what we were anticipating, and I'll show you what we were looking for in a moment. We looked at where we saw the best performance happen across all of these experiments, and they happen at about 2.9 cycles per degree. This is the equivalent of 2,200. In other words, if flying snakes were humans, they would be legally blind. We did the same thing, same measurements with temporal frequency, going from easy or slow to hard, fast. And we again see the same kind of sets of curves where we start to see a little bit of a fall off around 100 hertz. What's not shown in this graph is that the tendency for the nystagmus events, these reflexive events, was lower at these high frequencies. So while they are doing fairly well, we want to target this dashed line here. They're doing pretty well, but it happened very rarely. And again, if we looked at where they did the best performance, they operate at about 46 hertz, which is about where our vision starts to fail. So they're doing their best work at our maximal work, at our maximal speeds. What we had expected to see were these nice, big, sharp tuning curves. We wanted to see this nice roll off. We wanted to see these nice, clear arches. Instead, we saw these big, broad arches with a very slow roll off. And it really stumped us for about 18 months until we realized that what we were seeing is broad spatial and temporal tuning. In other words, these animals have a very dynamic lifestyle. They live in dark cavities, but then they go out into bright sunny canopies. They're sedentary for much of the day, but then they fly and glide and hit these speeds of 11 meters per second. And their visual system is adapted for this exact lifestyle. By having broad spatial tuning and broad temporal tuning, they're able to change what they're able to process in a large number of circumstances versus having a very sharp tuning where their vision would only work in a number of very constrained circumstances. But better than me explaining this to you, the benefit of using these platforms is that I have a much easier time showing you. So I made a simulation of how these snakes can see based on the data that I collected in these experiments. In this simulation, this is the interface for Unity 3D, and this is the game that we're going to see closely in the next minute or so. I took several cameras and I stacked them on top of each other, and I placed them in such a way that they replicated the field of vision that we saw from that inverse telemetry. So I used their field of vision measurements to determine accurately where the camera position would go. I'm now going to play this simulation, and you're going to see a snake moving through a forest, and there will be an inset that shows you the top-down view in a way that looks a little bit more familiar to us humans. I also want to point out that these snakes have a very wide field of view. They can see about 270 degrees around their head, and the screen is only going to be able to show us 180 degrees. So it's going to be a little bit warped and funky, but that's how translation works. So here we have a snake moving through this forest, and you can see that it's really hard to translate this inset, which looks more natural to us, to this very wavy and smeared and fuzzy scene. I use the spatial acuity to determine how clear these scenes would be. So if you look at a tree that's close, it actually has a fair amount of resolution, but once they get far away, that resolution is lost. Because I'm able to reproduce the frame rate at which they're able to see, we are able to capture some of the elements of how they're able to see. So we don't see a lot of motion blur or animation blur. The movements are quite crisp and precise. It's just the objects are blurry. So this is how flying snakes are able to see. It might be confusing at first to think, how can an animal navigate the world with such blurry vision? But if you're relying on motion as much as these animals are, it gets a lot easier to do. So this is a quick overview of how neuroscience influences neuroengineering. I wanted to study these animals that are rarely studied, and I wanted to look at how they used visual behaviors, how they controlled these visually mediated behaviors. In order to ask these complex and nuanced questions, I needed the right tools to help me do it. So I started to look toward gaming tech in order to achieve these goals. I'm not the only neuroscientist to do this, and I'm certainly not the only neuroengineer to do this. And to prove my point, I'm going to talk about now some examples in the field that we see predominantly in biomedical engineering. So one of my favorite and most exciting aspects of neuroengineering is the brain-computer interface. The brain-computer interface was originally designed to improve performance of prosthetics and as a cure for spinal injury. So when we have a prosthetic or have the need for a prosthetic, the remainder of the limb has nerves and muscles in it. And these nerves are still getting the descending signals from the brain and spinal cord as if the rest of the hand was going to be there. In this case, this is an amputated arm. The problem with trying to create a brain-computer interface with a prosthetic limb, a remaining limb like this, is that the nerve signals are really small and weak. And compared to the muscle signals, which are very loud and overtaking. So it's very difficult to have external devices detect the nerve signaling. And a lot of the prosthetics that are around today use the muscle signaling instead. This limits the range of motion that prosthetics have, and it limits the feedback. So right now, even with the brain-computer interfaces, we don't have a lot of sensation going back up to the brain and spinal cord. We only have motor control coming down from the brain and spinal cord. But with the advances of artificial intelligence, this electronic to neurological communication is much, much easier. So there's some research that's being done at the University of Michigan, which is helping improve the quality of prosthetics through using brain-computer interfaces.
We've now seen, to my knowledge, the largest voltage recorded from a nerve compared to all previous results. That makes these signals big enough that we can record them and interpret them for controlling a prosthetic.
Back into my mind the thought of, well, if I had something like this, I could actually outwork without risking hurting myself.
So all of these prosthetics that you'll see in this video are all being controlled by nervous signaling in very much the same way that they would have controlled their biological hand.
I think it's a good step into the future. It's a good way to move forward for not only me, but for other people.
So these prosthetics are actually interacting with nerves in the arm, but there are other prosthetics that interact with signals in the brain. So we'll talk about that now when we think about spinal cord injury. Before I get there, I want to do a quick aside to talk about artificial skin that's being developed in Japan and Korea. So here we have an artificial skin that's placed on a tube. And if we look at the computer readout, you not only get the intensity of the force, you also get the rough direction of the force as well. In Korea, they've taken this technology and extended it into material science by thinking about a self-healing material that can coat surfaces easily. This material not only gives you location information, so if I touch the tip of a finger, it can give me the signal at the tip of a finger, much like how this artificial skin does, but it's also self-healing. So the polymers will realign and reattach in such a way that the location information is not jeopardized. So if you are cutting, let's say you're cooking and you cut your finger, this prosthetic skin has the potential of healing itself and still continue sending feedback through your prosthetic limb. So you can imagine that the future of prosthetics is really exciting, mostly because of these advances in neuroengineering. I do want to make the quick aside to talk about 3D printing, which is one of the main tools that has been coming up a lot in neuroengineering. So by 3D printing prosthetics, we're boosting accessibility to who can have these tools. These are doctor and physicist design plans that can be printed at home if you have a 3D printer, which makes the prosthetics more affordable. The BCI prosthetics that I showed before range between $50,000 and $80,000 after insurance. So it's really difficult to get, but at-home 3D printed prosthetic costs on the order of $100 to $300, which is still not a great price, but far more accessible than previous prosthetics. This is particularly useful for children who need to have prosthetics that grow with them. So now you're able to produce 3D prosthetics as needed as the child grows. And finally, we can normalize prosthetics by incorporating and supporting stylish and aesthetic designs, such as this wireframe 3D printed leg and this super sci-fi futuristic hand. Elon Musk has started a company called Neuralink, which aims to popularize brain-computer and streamline BCI devices. So here's a BCI device that will get implanted into a brain. They're currently doing research on non-human primates, but they are thinking of expanding to humans in the next 10 years. They're working on automated implants, so trying to create a robotic, largely robotic implant procedure, which will minimize the amount of damage and recovery time for implanting such devices. They're also working on one of the hugest setbacks in neuroscience to date, which is having long-term implants. We have an immune response in our nervous system, which basically covers up and crowds out the surface of implants. So they're working on designs to help us get past the 4 to 6 month mark that we have now and more into 1 year to 1.5 years. These devices are accessible via Wi-Fi, so you're able to communicate and control them and program them quite easily. So all of this, with the popularization of such devices, I can say confidently that in our lifetime, we're going to see cyborgs more and more often. We're also going to see a lot of BCI used with spinal injury repair. And this is a really great example of applied animal research. So here is some work done by the Moritz Lab at the University of Washington, where they trained a rat to do a grasping task, and then they chemically cut off the nerve by injecting a chemical that silences the nerve, and they can't move their arm. They implanted a brain-computer interface device that works with the muscle on the arm, and it simply sends the exact same signals from the brain that would go to the nerve directly to the muscle. So there's not as much programming and figuring out as with a prosthetic limb. You're simply trying to bridge a physical gap in the system. They were able to regain their task by using the BCI. And this was done in about 2016 or so, and we've already seen it applied to humans today. So this is research that has happened in 2021, where there's a person who has a brain implant, which then interfaces with these electrodes on their arm, and they're actually able to regain mental control of their paralyzed limb. There's also a lot of prospects between nanotechnology and neuroscience. You might have heard a little bit about this with a lot of the conspiracy theories swirling around COVID-19, but we don't have self-assembling nanotechnology, so there's no way that we could have things that are turned on and assembled by Wi-Fi just yet. In about 10 years, you could come back to me with that problem and I'll give it a little bit more pause, but right now, nanotechnology isn't quite there yet. There are a lot of exciting prospects for where this merging can actually go. For example, at the Atlas Institute, where I work at CU Boulder, Carson Bruns is working on something called tech tattoos. These are functional tattoos. For example, here is a tattoo that's shown on some skin, which is white in visible light, but once exposed to UV, as done with this flashlight, it turns a bright blue. Any part of the skin that is protected will not react. And so these tech tattoos allow function to provide function to our tattoos. Currently, this lab has produced two types of tattoos, one that responds to UV and one that responds to heat. And they're currently working on a tattoo that is conductive, so they can tattoo circuits into our skin. This opens up what we can do with our skin and signaling by expanding what we can communicate with ourselves. So let's say you have diabetes and you can measure the amount of insulin and blood sugar that you have at a skin level using one of these circuits that were tattooed into your skin. Now you have an element of biofeedback and get some early warning signs to avoid more damaging scenarios. So we talked about how neuroscience is motivating neuroengineering, and I've given you a couple of examples about how biomedical engineering has been progressing neuroengineering as well. But now let's talk about the future of biotech, specifically a future of neuroengineering, specifically by what is given to us by big tech. Whenever we think about technology, we're really just imitating a lot of sensory or motor functions. For example, there's speech to text, computer vision, and all sorts of decision making that mimics how humans do these same functions. When we think about speech to text, we've accidentally found some alignment. So deep learning algorithms, when they set up on their own to learn human speech, inadvertently develop the same processes as humans. If we had neuroscientists in this development team, they would have come up with this procedure far earlier and probably have a more streamlined and faster, better working speech to text. We would also be less likely to see the biases that we see in speech to text today. There's strong racial and gender biases due to lack of diversity in development teams. So because a lot of white male people are developing this technology using white male samples, we're getting biased tools as well. If it was neurologically designed, we would get around these biases in a major way. For example, if we look at how well women's speech is recognized, about 47% of women's speech is detected accurately by Google speech to text versus 60% in men. The highest scores in men are around 95%, whereas the highest scores with women is about 75%. When we look at the rate of errors across race, we see that white people have fewer errors with speech to text than Black people. Again, if we have these problems with speech to text and the solutions exist in comparative neuroscience, there is a way that we have learned functionally to detect language that supersedes race and gender. So if we're able to mimic that in our design, we're able to get more widely accessible speech to text tools. The same thing goes for computer vision. Here on the right is Daimler's self-driving truck. Any vehicle that's self-driving must require computer vision that has to classify objects by sight. So the car has to know whether they're seeing a human, a car, a lane, the edge of a road, etc. It has to predict speeds and acceleration to avoid a crash. So if you've ever driven in a car that has the automatic alert that you're going to run into the back of a car, that's done by detecting the amount of acceleration in a visual scene. And then finally, there's the control of lights by detecting light levels. Currently, this computer vision is done by analyzing the entire scene, an entire high resolution image. The way some companies have gotten around this demand is to use IR, which is going to have far less information than RGB color. But neuroengineers, such as people in UWin at the University of Washington, have explored how they can use classification using low and well-placed spatial sensors. So if we're trying to distinguish cats from dogs or cats from dogs from people, we actually don't have to look at the entire image. We can look at key points, such as these high contrast areas around the eyes and places that are where we see a lot of change, such as the shape of the ears and the shape of the nose if we're looking at people. So if we were to incorporate, for example, this low sensor strategy, we would be able to make computer vision the kind that that's used in self-driving vehicles. We would be able to make it cheaper, faster, and more biomimetic, which means we're getting, again, away from biases. Tesla, for example, has self-driving cars that have a difficult time recognizing and identifying dark-skinned humans versus light-skinned humans. If we were using a more biomimetic approach, we would be able to detect a human regardless of the color of their skin. Finally, there's this trend toward biological integration. If we think about the history of all of our common devices, a radio, a television, and a telephone, we could see that the trend for how these devices have changed over the years is one that's approaching biological integration. For example, the radio, the television, and the telephone all started out with approximately one per household. When people engaged with it, they were physically far away. Then we started to make these devices portable and wearable, such as with the Sony Walkman shown here. And eventually, they became nearly integrated into our bodies, as we see with earbuds today. The same thing happened with televisions, where they became smaller and more portable. And with the invention of computers, computers started to take over a lot of the functions that televisions had. So we were having this large screen that we use for, was once in one room and then one per room, and then something that was wearable. And we see the same thing happening with phones, where there was one per house, then one per room, and then one per body. The evolution of mobile phones has overlapped with the evolution of televisions and computers, where now we walk around with wearable devices that give us all of the same information and functions as all three of these devices, which once lived far away from our bodies. And the impacts of these devices being near our bodies nearly 24/7 is measurable. And unfortunately, in a lot of these cases, damaging. For example, blue light is known to very extremely disrupt sleep, which has caused an epidemic of insomnia and loss of sleep quality due to people falling asleep with their phones in their room. This exposure to blue light is much more pronounced than the exposure to green light that we mostly see from natural light during the day. Blue light disrupts your circadian rhythm twice as readily as green light. And having this disruption leads to memory loss, difficulties in learning, and because sleep is the main way that we flush our nervous system out from neurotoxins, we have a difficult time getting rid of our neurotoxins. This leads to chemical imbalances, which then affect our mood, causing depression, and our gut, causing issues with digestion and nutrient absorption. And we also see long-term impacts on our eyes as well. We also see impacts on posture from looking down on phones. There's an increase of dowager's humps that have been formed by looking down and causing this spinal misalignment. We also see issues around security and health regulation with telemetry. So these are things like Apple Watches and Fitbits. They tiptoe into the gray area around medical information. And it's very difficult for doctors to get to use this information, which could be very beneficial for our collective health. We see early hearing loss due to headphones and earbuds, partially because of the tendency to increase buildup in our ear canals with the wearing of earphones, but also because people have a tendency of listening to music at volumes that are damaging to their ears. For example, here we're looking at loudness of sounds that are dangerous to us. So we really want to listen to sounds that are less than 90 decibels. But if we are subject to listening to sounds this loud, we want to shorten the amount of time that we listen to it. So here we're seeing the safe levels that we can listen to sounds at various increasingly difficult, damaging levels. And if we look at These are Swedish teenagers that were interviewed. On average, they listen to their music at 94 decibels for over 4 hours. So they're incurring damage to their ears by just their everyday use using earbuds. So we see these impacts, these biological impacts, and this is now becoming more of a talking point in big tech. There's a lot of room for physiology and neurobiology in these sorts of technological designs. So you can see yourself potentially as a neuroscientist working for companies like Google, Facebook, and other app designers as well. The benefit is if we see more biology, specifically neuroscience, considered in the development of these designs, we now see a very, very bright, shining future. For example, the phone and telemetry devices like Apple Watches and Fitbits can be used to improve sleep patterns and healthy behaviors by signaling to us when we're engaging in damaging or unhealthy behaviors. And there's a push toward personalized medicine through the use and commonality of these telemetry devices. Like a lot of people have smartwatches, a lot of people have Fitbits. This has led to working with fluid measuring microchips, such as this microfluidics chip that's being designed at the Atlas Institute right now. So these chips are being used to detect fluids, any bodily fluid or any running fluid, in order to do at-home tests and diagnoses. We could also consider sound design, that the sound itself, you don't have to change the product, you could change the sound, the types of sound that are being produced in a way that it protects hearing, which then can lend itself to create smarter hearing aid design and make a more equitable world. The best part about all of this, as I've shown you in a couple of examples before, is that when you consider these natural tendencies, when you start to incorporate biology and neuroscience into these designs, you get more compatibility with diverse bodies and people. We're seeing more and more different types of people using these devices than what was initially thought of or intended by the very non-diverse groups that are inventing them. So now, as we're seeing more and more people use these designs, if we consider biological design, we're inherently approaching more universal and accessible designs. And we're seeing a lot of this happening already. VR and phones are developing eye tracking tech. This is already in the market. For example, the Pico Neo virtual reality headset has a ring of cameras that look back at you while you look at it. And the primary reason why they want to do this is that our eyes are constantly moving all over a scene. We actually build a composite of our scene by performing these very rapid movements, about 3 per second. They're called saccades. So if you were to look at a picture as we see here, we're going to spend a lot of time in these high contrast areas, like around the eyes, around the nose, and on the lips. And this actually helps us identify people. If we have eye tracking in VR and with phones as well, we can improve our user experience and create more natural interactions. And we can increase security since our eye movements are unique to the person. Unfortunately, not all of the motives for getting eye tracking are positive. A lot of marketing companies are trying to get eye tracking data to help enhance their advertising strategies. Right now, whenever you're scrolling on an app, if you're on Instagram or Twitter or Facebook, it's tracking how much you're scrolling and where you stop. So if you hover over an ad, you're going to see more of those ads. They want to take that technology and enhance it to use eye tracking. So they're going to look at where you are looking on a screen and send you more ads and materials based on what you're looking at. Because a lot of this marketing already is and will continue to happen without proper consent and informing of the user, a lot of these changes are going to happen without us knowing about it. And we're back to where we are before, where we're seeing that these wearable devices are going to have impacts and we're not considering what they are. So we really need a lot more neuroscientists that are involved in the development of technology, especially as we get more and more integrated with our tech. In general, there's no neuroscience that's happening now or will happen in the future that exists without substantial engineering. Neuroengineering, again, is just applying the discipline and rigor of engineering more intentionally to the practice of neuroscience, which is already inherently an engineering-focused field. We look at a lot of different things that come into play. There's far more fields than I showed here, but we can see how all of these intersect within neuroscience and how we need to use all of these things to answer the complex and nuanced questions that we're asking today. So thinking about the future, I have been developing these neuroengineering kits for kids. These are STEM experiment kits for students 9 through 13, where they'll build a device and test it and use that device to explore or understand how their nervous system works. For example, in this demonstration that I have here, this is a static electric detector. In this kit, students are learning about how there are things that happen in our world outside of our sensory perception, things we can't see, smell, touch, or hear. With it, we can use neuroengineering to expand our sensory ability by creating a device that translates some of these phenomena that happen outside of our sensory experience into our sensory experience by turning it into light coding, for example. These tools are built for independent exploration. So after they do the experiment, they're given a couple of guidelines so students can start to explore and ask questions for themselves and see how neuroengineering naturally and organically fits into their life. Not only will they use this to explore new phenomena, but they'll use it to understand how their brains work. For example, this is a Chladni plate. Here you have a vibrating plate on top of a speaker, and you place small particles which will align in areas where the wavelengths or the waveforms are really, really strong. We use this same phenomenon to determine how we hear pitch in our ear. Inside the cochlea is a basilar membrane, which is wide on one side and narrow on the other. When we hear sound, the vibration will resonate or have a higher amplitude in certain places and hit hair cells. The specific hair cells that are hit are how we encode pitch. So lower sounds are heard at the base where it's wider, and higher sounds are heard at the top where it's narrower. So they'll be making a model of this and using it to understand how they're able to hear different frequencies of sound. So that's the future, but what about you all? How can you pursue neuroengineering? Just think multidisciplinary. Think about the multidisciplinary fields such as neuroscience, biomechanics and biomedical engineering, applied mathematics and applied physics, material engineering, computer science, or human-computer interfaces. Human-computer interfacing is actually quite close to neuroscience and has a lot of crossover of a lot of labs and intellectuals. And I also just want to let you guys know that you can create the education of your dreams. Just because you major in biology or just because you're getting a degree in engineering doesn't mean that you can't take other classes that will fill out the information that you want to learn. So you could be a biology major and take a couple engineering classes. You can double major in neuroscience and human-computer interactions. You can join maker spaces and become familiar with a lot of the tools that are being used in neuroengineering, such as 3D printers, CNC routers, and laser cutters. And I encourage you to stay strong in your math skills. A lot of these tools that we're seeing today, like the brain-computer interface, AI, anything where we're looking at spike sorting, if you're doing internal recording of neurons, they're all just using sophisticated mathematical algorithms. So the stronger you are in math, the better you'll have access to a lot of these tools and be able to change them for your express purpose. I mentioned a lot of research institutions here that might be of use to you guys to follow up on. I work at the Atlas Institute, which is an interdisciplinary institute. We work with a lot of neuroscientists, engineers, nanotechnologists, chemists, biochemists, all to create new solutions that are socially minded. So trying to solve a lot of the social issues that are currently going on. And that's at the University of Colorado Boulder. There's also, I showed you work at University of Washington. The prosthetics were designed at University of Michigan, and the two skin materials were designed at the Japan Institute of Science and Technology and Korea Institute of Material Science, respectively. Neuralink is a new company and they are hiring like crazy. So if you have a degree, a bachelor's degree in neuroscience or biology or engineering, you might want to think about that. And they also have internships as well for people who are younger. So you might want to check out Neuralink if you're thinking about getting a little bit more research experience. There are a number of neuroengineering institutions in the US as well. Duke, Johns Hopkins, Virginia Tech, MIT, and Caltech all have express neuroengineering institutions. Virginia Tech is couched in the biomedical engineering department. So there's a lot of universities that are thinking actively about neuroengineering that you can think of when you're thinking about advancing your education. So I'd like to thank you for your time and attention. I particularly would like to thank the Soha Lab for their help with my flying snake research and my funding sources, NSF and the RIOC grant. I'm always active and happy to keep in touch with you guys. I'm active on Instagram and Twitter, so if you have questions, feel free to tweet me or you can send me an email. And if you'd like to learn more about what I do, you could check out my website, curiousdrz.com. And if you're a programmer yourself and you're curious about the code that I built to do all of the experiments that I talked about, you could check out my GitHub, which you can get here from the QR code. And I'll stop at this point to answer any questions. Thank you guys so much.
Thanks so much, Dr. Z. It was really fascinating to kind of get that foray through multiple avenues and talk about what's out there, all the integrations, and even like gaps in what's out there, disparities.
Yeah.
So thank you so much for that really wonderful overview.
Awesome. I know I went a bit over time, but I'm happy to answer any questions, or if you tweet me, I'll answer them sometime today.
Awesome. Yeah, it seems like we are a little bit short on time, but folks, if you have questions, please reach out to Dr. Z. Super awesome. And if we have a lot of questions coming up later, we can even maybe send them in a group email to you. So
yeah, that would be great.
All those questions answered.
Awesome.
Great. Thank you. Thank you so much. So
Thank
wonderful.
you. This is really great. Have a great day, everybody.
Thank you. You too.
Thank you.
All right. And I'm going to pass it over to Megan and Arvin to introduce Dr. Gamble-George.
Yeah, so our next event is also very exciting. We have Dr. Joyonna Gamble-George, who will be presenting on a topic called Understanding the Brain from Cells to Behavior in Neurodegenerative Diseases and Anxiety Disorders. And before they start, I have a bit of a bio for you guys to give a bit of a background on their research as well. So Dr. Joyonna Gamble-George, PhD, is a former early career policy ambassador and alumna of the Neuroscience College Program for the Society for Neuroscience. Through the AAAS Science and Technology Policy Fellowship at the National Institutes of Health, she has served as an expert science advisor for doctors, psychologists, nurses, and other scientists all across the United States that conduct implementation science research on cardiopulmonary diseases and mental health in vulnerable populations, such as people living with HIV. She holds a bachelor's in science in biochemistry and biology with honors in mathematics from Xavier University in Louisiana, a Master of Health Administration from the University of South Florida College of Public Health, and a PhD in neuroscience from Vanderbilt University. She completed postdoctoral training at the University of Florida McKnight Brain Institute, where her research focused on the effects of methamphetamine on bidirectional neuron microglia communication in the human brain and animal models of HIV infection. Her other research efforts concerned potential drug therapies for Alzheimer's disease, the molecular and synaptic consequences of drug abuse and addiction, endocannabinoid signaling in affective disorder pathology using mouse models, and the biopsychosocial factors that contribute to substance addiction and HIV risky behaviors in people. Given her considerable accomplishments, Dr. Gamble-George was selected for the AAS If Then Ambassador Program, which brings together women in a variety of STEM careers to serve as high-profile role models for middle school girls and young women. Dr. Gamble-George has received notable recognition and many awards for her strong dedication to mentoring the next generation of scientists. So we're very excited to introduce Dr. Gamble-George, and you can have the floor.
Okay. Hi, everybody. So I'm going to start sharing my screen. So can you see the presentation?
Yes, we can.
Okay, great. So today I'm going to, sorry about that, let me go back. So today I'm going to be speaking with you about sort of my career trajectory to become a neuroscientist. So I'm just going to give you a snapshot of my journey as a STEM professional and just how my life experiences, especially those involving my research work in brain science, helped me to see how I could really make a difference in society, especially from the aspect of, improving the medical field as well as the healthcare industry. So I was raised in rural Alabama on my grandparents' farm. And as a young girl, my grandparents really instilled in me the importance of acquiring a quality education or knowledge just to make better informed decisions about life. But they also showed me the importance of community service and how it really could help me as an individual to help me build my character, find a new passion, but also like promote personal growth and a sense of accomplishment while also doing good for people in the community. But the other interesting thing is that during my upbringing in rural Alabama, I witnessed a lack of medical care and equipment and the need for more primary care physicians and specialists in disadvantaged populations due to poor social determinants of health. And residents often had to drive like 30 miles or more just to visit a physician for a wide range of ailments, from the common flu to like severe complications from diabetes and cardiovascular disease. So my grandparents were among these individuals where their own city could not provide for meeting and serving their healthcare needs. And this experience and the values that my grandparents instilled in me really compelled me to believe that I could alleviate these impediments to patient care if I pursued a career that could really shape the medical field or healthcare industry. So I majored in the biological sciences as an undergraduate at the University of Louisiana. And then I later attended the University of South Florida College of Public Health to obtain a master's degree in health administration. I was at that point in my life, I really wasn't sure if I wanted to be a neuroscientist. I was going back and forth between becoming a medical doctor or a neuroscientist. But during graduate school, I got to participate in a program that was sponsored by the American Hospital Association, where I served as a health system specialist in a VA hospital. And in this role, I was exposed to health services research and had to come up with solutions to improving poor patient-provider relationships due to understaffing of medical personnel and preventing delays in patients being admitted to medical units like the emergency room. And so this program, it really just gave me firsthand experience to improving the healthcare industry, as I had dreamed of during my childhood in rural Alabama. However, I still felt that need to do more, especially in terms of understanding how poor social determinants of health can impact an individual health outcomes. So I went on a mission trip and volunteered with medical, physical therapy, public health, and nursing students and the U.S. Peace Corps in El Yaya, Dominican Republic. And the goal of the volunteer experience was to implement outreach and sustainable health projects for underserved communities in the Dominican Republic. So I got a chance to build ventilated pit latrines, and these are pictures of me actually building the pit latrine. And I also provided free anti-parasitic medication in a clinic and health education workshops on like hygiene, water sanitation, maternal and infant health. We also talked about body mechanics and ergonomics. And we did this to the people that were living in this impoverished remote village in El Yaya. And the residents in this village, they had limited access to education, healthcare, and electricity. They also encountered a lot of daily issues with hygiene and sanitation due to a lack of clean running water, which was sometimes substituted by rainwater, and they used that for bathing, cooking, and even drinking. So the residents, as a result, they endured a lot of vaccine preventable and vector-borne diseases. So just getting the thank yous for the residents and the warm smiles and affectionate hugs, it really made me realize the importance of connecting community-based and patient-directed organizations to medically underserved communities. Because these organizations, what they can do is they can really help deliver comprehensive, high quality primary care healthcare services to the most vulnerable populations. That could be like people that are homeless, it could be like combat veterans. And what they could do is help to integrate these services to provide better access to them in areas where there's like economic, geographic, or even cultural barriers that limit people's access to affordable healthcare services. So to gain more exposure to health disparities and inequities, and how I could actually implement projects to combat these public health concerns, I served on an interdisciplinary team of health professionals at a community center to develop and implement community-based projects in a rural underserved community in Laurel, Florida. And there I identified many barriers to healthcare in this community, including, but not limited to, like financial insecurity, issues with health illiteracy, problems with accessing certain healthcare services. And one of the solutions to addressing these barriers was that I helped with implementing a health education intervention at the community center. So for preteens and teens, I provided instruction to about 40 teenagers that were aged 12 to 16 during a teen empowerment program on various chronic diseases like cardiovascular disease, diabetes. We also talked about HIV, AIDS, and dental health. And then I also helped to develop and implement a healthy nutrition program for up to 100 camp youth that are aged 5 to 12. So this is just an example of the health education curriculum that I actually implemented for preteens. And I did something very similar for the teenagers as well. So with this, we invited like different guest speakers just to talk about the different chronic diseases like diabetes and cardiovascular disease, and also how to prevent these diseases from developing. And then we also did a lot of interactive activities with the preteens and the teenagers just to promote their health, such as teaching them how to check their heart rate using their wrists. So the projects I mentioned earlier, a lot of them focus on health disparities. But during that time when I was doing them, I was also volunteering at a VA hospital with doing like different fun activities with veterans that actually suffered from post-traumatic stress disorder, or what is also known as PTSD. So a lot of these veterans, they had poor management of their medical condition. And as a result, I sort of developed this curiosity about what could be done from a scientific perspective to really understand post-traumatic stress disorder, or PTSD, and find a possible solution for that disorder. So this volunteer experience really led me to pursue a PhD in neuroscience at Vanderbilt University, where I got to study the endocannabinoid system. So you're probably wondering, what are endocannabinoids? So endocannabinoids are also known as endogenous cannabinoids, and they are lipid-derived molecules that activate endogenous cannabinoid receptors in the brain. And the receptor that I will be mainly focusing on for this talk is the cannabinoid type 1 receptor. But there's two well-studied endocannabinoids, that's anandamide, or AEA, and 2-arachidonyl glycerol, or 2-AG. I'll be focusing on this talk on anandamide, or AEA. So endocannabinoid signaling can really modulate or regulate your anxiety-like behaviors. And this is important since anxiety disorders are the most common mental illness in the US as well as worldwide. And millions of people in any given year are affected by these disorders. So when you activate or block the cannabinoid type 1 receptor, an animal can exhibit a variety of behavioral characteristics. So activating the cannabinoid type 1 receptor with like an agonist, such as THC, can actually induce this reduction in anxiety behaviors. But if you block that same receptor with an antagonist, such as rimonabant, you will actually have an opposite effect on anxiety. There should be an increase in anxiety in animals. So in the lab that I was in during my PhD at Vanderbilt, one of our main findings was that blocking the CB1 receptors actually elicited these behavioral responses that were linked to anxiety, only when the animal was subjected to a stressful external stimuli. And in this case, it was like a novel case setting because I was working with rodents. So with mice specifically, when they're very anxious, they gravitate more to dark areas. So if you put them in a well-lit area, they make them very anxious. So a lot of this relates to a theory that endocannabinoids, they're produced or synthesized, or at least the cannabinoid receptor is modulated only under stressful conditions. So such like synthesis or modulation really occurs on demand, or when there is like a physiological need to dampen the stress response. So in this lab I was in during my PhD, next we wanted to see the effects of targeting molecules in endocannabinoid signaling on anxiety-like behaviors that were upstream of the cannabinoid type 1 receptor. So the molecule that we targeted was an enzyme that actually breaks down the endocannabinoid anandamide, or AEA, and that enzyme is called cyclooxygenase 2, or COX-2. So for my PhD dissertation, my hypothesis was that if you inhibit this enzyme known as cyclooxygenase 2, or COX-2, it will lead to a reduction in stress-induced anxiety-like behaviors in mice. And that really occurs, well, at least I thought at that time, by increasing anandamide levels in the brain. And we thought that the mechanism was actually the signaling that was going on with anandamide was occurring in excitatory neuron, also referred to as glutamatergic neurons. So in this lab during my PhD, we had previously shown, in collaboration with another lab called the Marnet lab, that COX-2 was an important modulator of endocannabinoid signaling in the brain. So COX-2 is well known for producing prostaglandins from arachidonic acid. However, we developed a new drug that we call substrate selective COX inhibitors, or I might refer to as SSCI, in collaboration with this lab. And the one that I'll be focusing on is called LM4131. So we created LM4131, which is a substrate selective COX inhibitor. And what it does is that it prevents the breakdown of endocannabinoids, but it does not inhibit the oxidation or breakdown of arachidonic acid like other selective COX-2 inhibitors. So COX-2, in this case, is a really important regulator of anandamide, or AEA. And that is why we were studying it as a target for drugs that can potentially affect anxiety-like behavior in animals and potentially in humans. So the behavioral test that I use for most of my experiments to really try to understand the effects of this drug on anxiety-like behavior is called the novelty-induced hypophagia test. So this behavioral test is also referred to as novelty-induced food suppression, and it spans over 8 days. So it begins with an acclimation period in which the animal is habituated to being singly housed in the Allentown cage. And then for about 3 days, the animal is trained to feed from a vanilla shake in the same home cage settings, but it's under red light illumination. And under this type of illumination, it helps the animals to reduce stress, because the animals, at least mice, they gravitate more to dark areas to feel safe, as opposed to well-lit areas where they feel very anxious and unsafe. So next, we actually gave the animals an intraperitoneal injection, or an IP injection, and that's an injection in the abdomen of the drug, which was our substrate selective COX inhibitor. And we gave it 2 hours prior to testing, because that was the time point in which the drug actually worked. And during testing, we observed the animal for feeding latency, which is the amount of time that it takes the animal to take its first taste of a vanilla shake. And we also observed for the amount of the shake that was consumed by the animal, and that was equivalent to their motivation levels. So they consumed a lot, they were very motivated. If they didn't consume as much, they were not so motivated. So these observations were taken when the animals were in the novel cage setting. So this is when they're in a very bright light setting. So that's how the novel cage setting is a little bit different from the home cage setting, is that the novel cage is under bright light conditions, where the home cage is nice. It's much more dark. So the theory is that when you administer a COX-2 inhibitor, in regards, or according to my hypothesis, to an animal, their anandamide levels actually should increase in their brain, since you're preventing the breakdown of anandamide. So we saw this in the brain of the mice that we injected the COX inhibitor with. So that's the diagram at the top far left, where we did mass spectrometry, looking at changes in brain anandamide levels under no stress conditions. And then we also measured feeding latency. using the NIST, which is the behavioral test I just talked about earlier, paradigm, and we saw no changes in fetal latency in the novel case setting. So this lets you know that really under no stress conditions, even though the drug is working, it's increasing and anonymizing the brain, the animals, it doesn't really affect the animals when they're not as stressed out. So then we repeated the experiments after the mice were subjected to foot shock stress, and we gave the injection 2 hours before performing this behavioral test called NIST, and then 24 hours after the foot shock stress. And once again, we did find that the COX inhibitor increased anandamide levels in the brain, so we know that the drug is working. However, only when the mice were subjected to foot shock stress that the COX inhibitor was able to reduce anxiety by reducing fetal latency in the mice, such that the fetal latency was very analogous to control settings or no stress settings. So then we wanted to see if the drug could work at different time points, and we found that our drug actually did work at different time points. So it completely prevented the anxiety-like behaviors in the animals at each time point that is shown in the figure. But we found that at the 8-hour time point was where we actually saw the maximum effect. So pretty much at this time point, we decided to do all of our experiments at that time point because that was the max effect we were getting with the drug for preventing anxiety. So then we repeated the experiments, still using that same behavioral test called NIST, just to see how our substrate selective COX-2 inhibitor actually worked in comparison with other COX-2 inhibitor. So what we found is that our drug called LM4131 actually performed a little bit better in reducing anxiety in the animals as in comparison to other COX-2 inhibitors like LMX celecoxib. We also realized that with our previous result, once we increased the number of animals we're using for our experiments, we actually could see some effects under no stress conditions. But they're very not as significant as under stress conditions. So then most of these experiments I've been talking about, they're more so geared towards seeing if a drug can treat someone with anxiety after they've been exposed to stress. But the question is now, what if you can give somebody a drug before they're exposed to something stressful in the environment? Will it still reduce their anxiety levels? So then we wanted to see if our substrate selective COX inhibitor could actually prevent stress-induced anxiety. So we gave the drug before we exposed the mice to foot shock stress. And then we performed our test again. And what we found is that the drug was not effective. So it's not good for preventing stress-induced anxiety. It's more so preventing, treating anxiety after you've been exposed to a stressful encounter of some. So next, we wanted to see if we could see any sex differences. So a lot of our experiments were just performed in male animals. So what we found is that our substrate selective COX inhibitor does indeed work in both male and female mice, but it seemed like it worked a little bit more effective in the male mice as opposed to the female mice. So then we also perform another behavioral test to determine if the hippocampus, which is a brain region that plays a role in memory. So we wanted to see if the hippocampus was actually involved in the anxiolytic effects we were seeing in the mice. So we examined the effects of our substrate selective COX-2 inhibitor, LM4131, on contextual fear extinction. So contextual fear memory involves a neural circuit that involves the hippocampus, also other brain regions such as the prefrontal cortex and different parts of the amygdala. And what you can see is that our substrate selective COX-2 inhibitor actually enhances contextual fear extinction. So this definitely says that the hippocampus is involved in these effects that we're seeing with the mice. So then we wanted to verify our results with in terms of the drug reducing anxiety-like behavior in the mice and a different behavioral test that can also test for anxiety. So the test is called the elevated plus maze, and this is a maze in the form of a plus sign. And it really takes advantage of the fact that mice tend to gravitate toward dark areas to feel safe, and they stay away from well-lit areas where they feel unsafe and anxious. So if you have an animal you place them in an elevated plus maze, you put them in the center of the maze, animal that is very anxious is going to run very quickly into the closed, very dark arms. So they're going to spend a long period of time before they ever venture out back to the center maze to look into the open arms, which are very well lit, to try to just explore that environment. So you might see an animal that is anxious have a decreased number of entries into the open arms. They might have increased number of closed arm entries, or an increase in the amount of time that it takes them to actually make their first entrance into the open arm, and a decrease in the closed arm latency. So we had a very similar setup where we would give the drug 2 hours before we expose them to foot shock stress, and we will wait up to 8 hours and then test them in the elevated plus maze. So as you can see, when we tested the mice in the elevated plus maze, once again, we did see a reduction in stress-induced anxiety-like behavior in the mice. As you can see that the mice that were treated with our substrate selective COX inhibitor, they had more entries into the open arms. So they were going into the very well-lit area where usually mice will feel unsafe. So they felt very safe and adventurous and just went into those areas. Also, the amount of time that they spent there they spent a very long time, and then they took a very short time just to go there first, as to going first to the closed arm. So this lets you know that our drug, indeed, it does work to reduce anxiety. Now we confirmed it with another behavioral test. So then we wanted to really understand more of connecting which brain regions, because I talked earlier about the hippocampus, but we also wanted to understand how is the amygdala actually involved with the drug effects that we're seeing in our mice. So why is the amygdala so important to endocannabinoid signaling? For one, it plays a role in emotional processing, since it assigns emotional significance and produces behavioral responses to external stimuli. Also, there are many connections to other brain regions that are involved in emotional responses linked to stress. One of those brain regions is the hypothalamus. In addition, the central nucleus of the amygdala is the major output brain region of the amygdala and plays a role in controlling fear responses. So studies have shown that lesions to the central nucleus of the amygdala can block fear condition responses. So the amygdala is composed of 3 subdivisions: the lateral nucleus, the basolateral nucleus, and the central nucleus, as shown in the diagram to the left. So over the next 2 slides, I will be discussing the effects of endocannabinoid signaling in the central nucleus and amygdala on synaptic depression using electrophysiology, which is a really cool technique where you actually get to record the activity of neurons. So we tried to really answer a question of how does endocannabinoid signaling modulate synaptic function using electrophysiology, in which we took these coronal mouse brain slices and we measured excitatory neuronal responses at the population level. And we did this by placing a stimulating and a recording electrode in the central nucleus of the amygdala, and then we measured changes in excitatory postsynaptic potentials. So on the right is a graph of our findings. So on the x-axis is time in minutes, and on the y-axis is the percentage of excitatory postsynaptic potential. So for the first 20 minutes, we measured our baseline when the brain slices were submerged in artificial cerebrospinal fluid. And then we washed on a cannabinoid type 1 receptor agonist called CP55940, which brought about a reduction in the excitatory responses at synapses in the central nucleus of the amygdala. And this effect was actually reversed by a cannabinoid type 1 receptor agonist. We considered as a result, we can, it was actually reversed by, a cannabinoid type 1 receptor antagonist, rimonabant. So since rimonabant was able to reverse the effects of the cannabinoid type 1 receptor agonist, we considered that these synapses were actually functional, which means they were working properly, at least under basal conditions. So then we wanted to connect a lot of the findings that we were getting with the behavior with electrophysiology. So you can actually perform simultaneously electrophysiology as well as animal behavior in a living animal using what is called in vivo electrophysiology. So this is where you put in electrodes in the brain region of interest, and then you'll place the animal while you're recording electrical activity in these neurons. You put them in a maze. So we actually use the elevated plus maze to test for anxiety in the mice. And these are mice, all of them were exposed to stress. And then 8 hours later, we looked at the amount of time they were spending in the open versus the closed arms. And you can see in the bottom diagram to the left, you can see that the mice that were treated with the substrate selective COX inhibitor, LM4131, seem to spending a lot of time in both the closed arms as well as the open arms. So these mice are definitely venturing out to areas where mice would actually consider unsafe, that are very well lit, as opposed to the control animals that are stressed out. They're spending most of their time in the closed arms. And what we see going on in terms of the activity of neurons, and we looked at excitatory neurons. So with the firing activity, as the mice, at least the mice that are treated with our substrate selective COX-2 inhibitor, after they've been exposed to stress, they're basically exiting the closed arm very quickly. But as they're doing this and they're exploring the open arms, the firing activity of those excitatory neurons is not as high or is doing as much as in the control animals. So what you see is a reduction in firing of excitatory cells in the amygdala. So after my PhD, I completed a postdoc fellowship at the University of Florida. And at UF, it sort of changed gears. Instead of still focusing on anxiety disorders, I started getting into HIV disorders and how they're related to neuroscience. So I was looking at HIV-associated neurocognitive disorders or HIV-associated dementia. So people living with HIV, they can develop a spectrum of cognitive, motor, and mood problems collectively that is called HIV-associated neurocognitive disorder, or HAN. And a lot of the symptoms include like difficulties with attention, concentration, and memory. And sometimes they can even have a loss of motivation, irritability, depression, and slow movements. So as a postdoc, we really wanted to understand from a cellular and molecular standpoint, what is really going on in people that have HIV in terms of their drug-seeking behavior. So these are people who might actually acquire HIV because they could be an injection drug user. And then their HIV actually it infects the cells in the brain. And in this case, or this study that I was doing as my postdoc, we were looking at how HIV actually affected microglia. So microglia is another cell that can find in the brain, but there actually are macrophages that get rid of cellular debris in your brain. So we had this theory that, okay, someone is using a dirty needle, they become infected with HIV, their microglia becomes infected, and now it leads to this low dopamine state. So we think that the reason for that is that there's an increase in a protein called HIV-1 TAT, and that protein is involved in the replication of HIV-1 virus. And then that caused all these like mechanisms or signaling processes where it really affects or causes a reduction in dopamine homeostasis in your brain. And then we believe that even with this hypodopamine state, so you can have somebody that has HIV-associated neurocognitive disorder or HIV-associated dementia, even if they're on antiretroviral therapy, we think that because of this state, that if they encounter something in their environment that can cause them stress, it's like a cue to go back again and seek out drugs to misuse or abuse, that will increase their drug seeking behavior. And once we understand the mechanism, what we want to do further down the line is really understand how to prevent or treat this. So before I get into some of the research that I did as a postdoc, we had to really understand that there's a lot of brain regions that are involved in drug-seeking behavior. Prefrontal cortex, the nucleus accumbens, or even different parts that make but our focus was actually on the midbrain. So we wanted to look at these mechanisms in the VTA or ventral tegmental area, as well as the substantia nigra pars compacta area. So one of our first questions we were trying to really understand, and we use a transgenic mouse model. And this mouse model, if you actually inject them with doxycycline, they produce HIV-1 TAT. And I had talked to you earlier about HIV-1 TAT is a protein that plays a role in replication of HIV. So we wanted to understand interactions between microglia and dopamine neurons. The figure in panel B in green are the dopamine neurons. So these are neurons that actually produce the rate-limiting enzyme tyrosine hydroxylase. And that leads to the production of dopamine. And the red are the microglia. So we want to understand the interactions in two brain regions in the midbrain, which I mentioned earlier, the ventral tegmental area and the substantia nigra pars compacta. And what we found is that the interactions between these two different cell groups are pretty much the same. However, it seems like in the ventral tegmental area, the interactions are much greater as opposed to the substantia nigra. And it just didn't matter whether we had control animals that didn't produce HIV-1 TAT, or we had the animals which are called WT-DOX, it stands for wild type doxycycline, that actually produce HIV-1 TAT. So once we knew that there was more interaction between the dopamine neurons and microglia in that particular brain region, we wanted to understand, like, why is that so? What is really going on? So we tried to understand how the HIV-1 TAT protein actually affects the expression of tyrosine hydroxylase. So we perform immunohistochemistry, which is a procedure where you take brain slices and you actually place antibodies that will target your protein of interest, and in this case, it's tyrosine hydroxylase. And the antibody will be linked to a fluorescent probe where you can actually visualize the protein within your brain slices. And specifically, we're visualizing tyrosine hydroxylase within dopamine neurons. So that's what you see in panel E to your right. So what we found is that the HIV-1 TAT, it actually decreases the protein tyrosine hydroxylase in the substantia nigra, but it does not do it in the ventral tegmental area. So it's doing this at the protein level. So then we're like, okay, so what about the mRNA level? If it's affecting it at the protein level, could it also be affecting the mRNA for tyrosine hydroxylase? So interesting thing, it did not. We didn't find any changes at all. And this is just an example of what's going on in substantia nigra pars compacta. We also found similar results where there's no changes in the ventral tegmental area. So it seems like the changes are only occurring at the protein level, and it could be that the HIV-1 TAT is actually killing off the cells, at least the dopaminergic neurons, as well as the microglia. And that's why you might not have as many interactions in a particular brain region as opposed to another. So once we did those experiments, our next question was, okay, we know what's going on. The changes are at the protein level. So what about the activity of these dopamine neurons in the midbrain? Is HIV-1 TAT doing something to it? Is it increasing it, or is it decreasing the activity? So in panel A, that's actually a picture of a neuron actually expressing tyrosine hydroxylase. And we perform ex vivo electrophysiology, but these are in cells, and we did what is called patch clamp. So you actually take an electrode and you're patching it onto a cell to record that individual cell electrical activity. And what we do is we actually applied HIV-1 TAT to the patch electrode. So it will go into the cell, and then we will see HIV-1 TAT's effect on electrical activity on that neuron. And what we found is that HIV-1 TAT actually reduces firing activity of the midbrain dopaminergic neurons. So we know now that at least in the midbrain, there might be a reduction of interactions between dopaminergic neurons and microglia that are exposed to HIV-1 TAT. It occurs at the protein level, but it's also affecting the activity, it's reducing it. So the dopaminergic neurons are not as active. So where do I go from now? A lot of the work I did was in anxiety and depression, and then I switched gears and I went into HIV-related neurocognitive disorders. But all of these experiences I've had now allows me to take it a little bit step further and do research where I can understand interactions between different factors. So a lot of research I did was always focused on the biological aspects and maybe behavioral aspects. But sometimes when it comes to understanding like brain disorders like anxiety and depression, there's other factors that come into play. People's lifestyle, what they're exposed to in their environment, genetic factors. So now I'm doing research where I'm trying to understand how those interactions really contribute to those brain disorders with the hope that I can monitor them and then predict them before they cause any adverse clinical outcomes in people. And one of the projects that I'm working on is really trying to understand interactions between individual genes in people and people that have been exposed to early childhood adversity, and how that leads to these brain changes that are at a regional global scale that eventually will translate out into anxiety-like behaviors. So that is currently what I'm doing now. So before I end my talk, I like to provide tips for young scientists and those that are interested in the STEM field. And my first tip is really be unique and think outside of the box. So when I was in high school, my art teacher, we nicknamed her Froggy because she loved frogs. And she would always tell us to try to be different and unique when it came to creating a piece of art. So this advice has been quite helpful in my research because sometimes you have to think outside of the box when it comes to understanding how biological processes in the human brain and the rest of our bodies work, especially when you want to develop a cure to treat them when they do not work properly. The second tip is know, claim, and own your worth. So the thing is, don't settle for less in life and know what you deserve in life, especially when it's regards to your STEM career. Do not give away your power or inner joy to others, but aim for your dreams in STEM, despite the odds that might be stacked up against you. And lastly, you always want to be a living, breathing example of excellence in STEM. Because you never know, future generations that aspire to pursue a STEM education and a career will one day look up to you for guidance and view you as a role model or potential mentor. My third tip is don't let negativity or people's actions or words deter you from your dreams. So when you let people's actions or events that you endure in life defer your dreams, you are denying yourself the chance to see whether or not your dreams become a reality and how they can help you in your pursuit to become a STEM professional. Always strive to live the life you want to live, to pursue the dreams and career path you want to achieve, and to never give up on you and your lifelong aspirations, regardless of the hardships you might face in life. So a quote that I think sums this up is, life is like a camera. Focus on what's important, capture the good times, develop from the negatives. And if things don't work out, just take another shot. My last tip, live your dreams. I think I would tell myself, if I can go back in time, to not let fear, intimidation, stress, or uncertainty really determine the steps I took during my pursuit to become a STEM professional. You have to learn to take risks and explore the endless opportunities that await you on your STEM journey. According to Harriet Tubman, she was an Underground Railroad conductor. She was an American abolitionist and an activist in the women's suffrage movement. Her quote is, "Every dream begins with a dreamer. Always remember you have within you the strength, the patience, and the passion to reach for the stars, to change the world. Even if your dreams are to become an extraordinary scientist, technologist, engineer, or mathematician." That's the end of my presentation. If you need to contact me for any advice or help or have any other questions, that's my website as well as my email address. So what questions do you all have?
Yeah, thank you so much, Dr. George. It was so nice hearing about your experiences and your research work as well. So if any of the audience members have questions, feel free to type them in the chat and I can ask them to Dr. George. Any questions? Anyone? Okay, I guess I'll ask a few myself then.
Okay.
So what are the most promising research areas you think are in the future of neuroscience? What specific areas do you recommend us as young future neuroscientists going into in the future?
I think right now, neurotechnology is a big one in neuroengineering, especially if you're able to use like data science or artificial intelligence to make predictions about brain disorders. I think that is really big right now. Also, people are trying to find other ways of treating anxiety and depressive disorders, other brain disorders, instead of just using pharmacology or drugs, because sometimes drugs don't always work in everybody. We're all unique. So another way I've seen is that people are now getting into creating devices that can provide electrical stimulation to certain brain regions. So one is TMS, where they actually use these magnetic fields to electrically stimulate different brain regions. And that can help people with different brain disorders.
Yeah, that's all really fascinating. So we have a question in the chat. So Pranav asks, what are the technologies that one would need to be good at to be a neuroscientist or a neuroengineer?
I think for right now, I think it's, besides just like the traditional education that they have, where you just learn like the basic background information about neuroscience, now people are definitely trying to train students in computer science that relates to neuroscience. I think that's, if you get a background in data science, computer science, that will help you in both fields these days, because a lot of fields are now pushing to more technology-based things that will help with really understanding brain disorders a little bit better. Besides that, in terms of engineering, if there are neuroengineering fields, definitely go into, or you can get into like electrical engineering and maybe find someone, a PI that's looking at the working in both disciplines with neuroscience and electrical engineering to bring the two together. Then I've heard of mechanotronics, where they do mechanical engineering with electrical engineering, and that's actually where you're able to build the devices. So that's another field. Then I think the other thing is, like I had mentioned how I'm now starting to get into like other factors like social, environmental factors. I think that's very important now, because when it comes to dealing like, at least with mental disorders, we can't always try to understand it at a cellular molecular level. You really have to understand it from a more holistic, systematic level. So if you can get into neuroepidemiology, that's the field, or something that's an interface between neuroscience and the social and behavioral sciences, I think that will definitely give you definitely an edge, or a more competitive edge, as opposed to everybody else, because people are not really bringing all the different fields together. So definitely be in fields that are more interdisciplinary, transdisciplinary, and multidisciplinary, that really interface with neuroscience.
Yeah, all really great advice and very fascinating talk also. So yeah, thank you so much, Dr. Gamble George. And before you go, Sasha, if you're here, do you want to take a photo of everyone? And
Yes, of course. Thank you so much for that incredible talk. If anybody feels comfortable, please feel free to turn on your camera. We're going to take a quick photo and we can post on our social media platforms if you're comfortable. I'm going to count down from 3 and then take a screenshot. I'll just give a second for everybody to turn on their camera who would like to. Awesome. The more the merrier. Let me go ahead and invite some more or let people in. Okay, awesome. I'm going to count down from 3 and then I'm going to take the picture. Okay, 3, 2, 1. Okay, let me just see. It looks good. Okay, I think that's good. Arvid, you want to take another one just in case?
I'll take another one.
Okay, let's just get one more. Sorry, my computer is being very slow.
Okay, 3, 2, 1. Okay. Yeah, thank you, Dr. Gamble George, again. Really excellent talk and really amazing all the work you've done. So thank you.
Thank you so much for just giving me the opportunity to present to everyone.
Yeah, nice having you here. Now I'll hand it over to Gurnur and Megan to introduce our next speaker.
Okay, I'm just going to share my screen.
Do you have sharing permissions?
Yeah, I'm just making sure I get like the right slideshow shared. I'm not sure if everyone can see this.
Yeah, we can see.
I can see it.
Okay, so our next speaker is Dr. Yvonne Nyavor, who is a scientist and microbiome enthusiast. So originally from Ghana in West Africa, Yvonne holds a PhD in neuroscience from the University of Idaho and currently works as a microbiome principal scientist for a biotech company. In the lab, she's passionate about understanding how bacteria in our bodies interacts with our brains and the entire nervous system. And outside the lab, she splits her time between consulting, coaching, science communication, and scientific mentoring activities. She enjoys collecting houseplants, cooking, listening to good music, reading, engaging in various community initiatives, and spending time with her husband and friends.
Thanks, Gunnar. That was a really nice intro. It was really good to see you all, and I really appreciate this invitation to speak. So I'm just going to go ahead and share my screen where I have my presentation, which hopefully shouldn't be too long. I want to leave time for discussion and questions, but I do have a lot of slides to get through. So anyway, I hope you like my picture. I picked it specially for you all. This is a picture that has never seen the light of day anywhere. And I picked it because I was like, oh, I need a fun picture. So there's my fun photo. I hope you like it. So just as Gunnar said, my name is Yvonne, and I am a neuroscientist and a microbiologist as well. So that is why I call myself a microbial neuroscientist. I'm sure you've never heard that phrase, but I hope by the end of my talk, you're going to be able to understand why I use microbial neuroscientist, because it's a fairly unique field. So before I begin, I just want to share a little bit of my story with you all. And I don't know if you can see this. I'm just going to start from the beginning. and that way you can see everything really nicely. So my talk is titled Gut Feelings, How Many Brains Do You Really Have? At the end of this, I hope you be convinced you don't have just one brain. And I come from Ghana in West Africa. So to do my PhD, I flew 7,200 miles all the way to the very exotic, not really, remote town of Moscow, Idaho, which is all the way in the Northwest. And so if you're from the Seattle area or the Portland, Oregon area, or from Idaho, give me a shout out. Technically an Idaho gal now. I loved it there. And I'm currently presenting from there, where we have a crazy heat wave, if you believe that. It's usually cold most of the year, but right now it's just really hot. But when I came to the U of I came to do a PhD in neuroscience, where I focused a lot of my study on figuring out how tiny little bacteria that live in our guts interact with our body's nervous system, including our brain as well. So I'm going to share some very little of that work with you, but really I hope to present more about a broader picture of the gut-brain axis, as we call it, and explain what that is to you, so that at the end you have a basic introduction to the concept. So when I was at the U of I was involved in a lot of different things. I started a TEDx event, which is like, if you like TED Talks, it's basically TED Talks. And I would coach a bunch of people. And that's kind of how I got my interest in science communication, because I realized a lot of scientists aren't really very good at talking about science. And we like using big words that confuse everybody, including ourselves. So if I do that, please tell me, and I'll try not to. I try not to use big words when I present, but that's kind of where I got my start in science communication, got a bunch of training, and then started coaching people doing that. And I love it. So Ghana, where I'm from, we love food, delicious food, and we grow a lot of different types of tropical vegetables and stuff. And so that's why I have this little pictures in here. We love chili peppers. We have maybe 100 different varieties that we use for cooking. And I love it. I love my Ghanaian food so much. It's super healthy. And since my talk actually does have some diet components, I thought you might like to see this meal, which is eggs and avocados and ripe plantains and just all mashed together with peanuts. It's really, really good if I ever make it for you. I'm a gardener as well, so I grow a lot of veggies here, but I mostly collect houseplants now. So instead of collecting jewelry or fashionable items, I just like plants. So I collect plants from around the world. And also while I was at the University of Idaho, I did a bunch of work improving the graduate program at the university, doing professional development initiative work and all of that. But the most important takeaway from this slide is I come from a country of just about 30 million people, but we speak over 250 languages. If you forget everything else about me, just remember that out of those 250, I speak a grand total of 2. So, that's impressive. It's less than 10%. Anyway, I did my PhD at the Balemba Lab at the University of Idaho. This is Dr. Balemba, right? I don't know if you can see my pointer. And he is our PI. He was a pretty awesome mentor. So this is going to be my first tip for anyone who is considering going into science, is find an awesome mentor. And I did. I was super lucky to find someone that really believed in me, that believed that I was smart and good enough, right, to do anything I wanted. And I wanted to do something that was really difficult. So if I didn't have someone behind me who had my back and was like, you got this, you can do this. And I was surrounded by awesome faculty who all, when I doubted myself, encouraged me. So that would be my first tip for anyone who wants to go into science, which I think is all of you. Just think about that. And I had the opportunity to work with a bunch of really smart people, all smarter than me, and they all helped me to do this work. So this is a crew. This was our photo from a few years ago. And then when I finished, I went on to the Washington State University, where I'm still working as a postdoc, and I basically study poop from kindergarten children I study the gut microbiome. And so I will go into what the gut microbiome is, and that will make a lot more sense. But before I started working at WSU, I worked at Axial Biotherapeutics in Boston. And so that's why I'm in Boston. And then I moved from Boston to Boston Analytical, where I currently work as a principal scientist. And in that role, I do a lot of meetings, a lot of Zoom, a lot of talking to people, a lot of presentations, but also a lot of science, super cool science, a lot of lab work, and also just helping people who are trying to make drugs based on the research that they're doing, helping them figure out what they need to do to get that drug to us, the public. So it's pretty cool work. I love my job a lot. All right, so moving right along, I'm going to start the actual science portion of this presentation after spending all of, 10 minutes talking about what you're not interested in. I'm kidding. I like to joke, as you might tell. So if it gets a little bit over the top, please feel free to tell me stop. All right, so let's get right into it. I'm sure you've all heard gut feelings before. Like, I have a gut feeling that it's going to rain tomorrow. Or I have a gut feeling that this person is a pretty cool person that I need to hang out with or get to know more. What does that actually really mean, right? So this phrase actually is hundreds of years old. It comes all the way, it's based all the way in the Bible, if you believe that. It has its origins from the Bible, where people in that time believed that your decision making was actually in the center of your body, so your gut, right? So that's why they started using that phrase, gut feelings, and it's kind of stuck around now, even though we kind of use it to refer to something different. But really, this cartoon is a really nice, accurate depiction of what gut feelings is. So your gut actually does have feelings, and it does have its own little brain. And I'm going to convince you of that. So a lot of us have this concept that we have this one brain that is up here that gives instructions, right? It just controls everything else that is below. And that is why it's seated up here to be able to control everything. It's like a king, a queen. It gives orders. Everyone must follow and obey. No, it doesn't actually work like that. When we look at the actual input of and output of energy into and out of the brain, what we notice is that 90% of information is coming into the brain and only 10% is actually going out. And so really that begins to challenge our mindset that we have this brain that's just giving instructions all the time. It's actually only doing that 10% of the time. So where's the 90% coming from? That's the question, right? It's coming from everywhere, but a lot of it is also coming from your gut. Okay, so your gut actually has its own little brain. It's actually called a little brain, and it's called the enteric nervous system. The enteric nervous system was discovered about 150 years ago by these super smart people who are long dead. Sadly, I would have liked to meet them because they're always super smart, but I can't pronounce their names, so we're just gonna leave it like that. Auerbach and Messner discovered neurons in the intestines about 150 years ago. And so that's when people began to think, wait, why are there neurons there? We thought neurons were always supposed to be up here. What are they doing down there? And that's when people started to actually think about it. But then about 50 years later, right, about 100 years ago, in pretty cool experiments, Bayliss and Starling, these were scientists who were studying a dog. They cut a dog open, sadly. I think the dog was sick and they wanted to figure out what was wrong with it. But then they noticed this really cool thing happening that they then called the law of the intestine, right? And that was basically that if the dog swallowed something, food, water, whatever, they noticed that there was this really coordinated movement of the thing from the top of the intestine all the way to the bottom. So I'm gonna play that again, right? So there's contraction right up above the ball, and then relaxation down there, and that creates a pressure gradient that pushes that ball all the way down. right? And it was the first time anyone had ever seen that. Now we know that this is peristalsis, and it's very, very normal and very common, and that's how our body digests food. But then it was like super cool. So then they were like, something must be controlling this very coordinated movement. And then they thought back and were like, wait, neurons were discovered 50 years ago in the gut. That must be part of what they do. They must be telling these muscles that are right up here to contract and these ones to relax, because it can't just happen in a vacuum. Instructions have to come from somewhere, and the brain is way too far away. And at the time, there was really no evidence that there were brain neuronal nerve endings in the gut. And so they were like, well, it has to be the neurons that they found. And so that's when they called it the local nervous mechanism, referring to the enteric nervous system. All right. So in our lab, we have fun studying the enteric nervous system a lot. And we do this with little intestinal segments. And this one is from a guinea pig. We use mice as well. We use rats as well. But we have fun basically taking these intestinal segments and putting them inside a bath. You know, it's a physiologic saline solution that keeps the segments alive. And so basically, when you remove the intestine out of an animal and you put it inside this bath, it's actually functioning exactly the same way as it would if it was inside the body, right? And that was one of the evidence that Baylis and Starling used to prove that there was an enteric nervous system, because if you have eliminated control from the central nervous system or the brain, and you're still seeing this very coordinated movement of, this is literally a poop pellet. My job as a PhD student was to paint it with black nail polish so we could put it inside the intestine. It was a fun job. But so when you put this pellet in right at the beginning, you can actually see the contraction that we talked about in the previous slide and relaxation right after the pellet. And that's creating that pressure gradient, moving that pellet all the way through from the beginning to the end. And then it just pops it out or poops it out. That's literally pooping, right? That's what happens when we poop. And how do we know that this is a neuronal thing? How do we know that it's not just a muscle? You know, your muscle is just smart. We know it's neuronal because when we administer tetrodotoxin, and this is the second panel below, when we put tetrodotoxin into the solution, which basically shuts down all neuronal impulses, the pellet just sits there. It can't move because there's now no instruction. So we know now that there's this little brain that's giving instructions to the pellet, to the muscle, telling it contract, you relax, and move this thing out. And it's actually really neat when you get the opportunity to do it in the lab. All right. And so just as I described, this is our setup. We love this so much. Call it a gastrointestinal motility monitor. We run it a lot. It's a lot of fun to run. If you ever are out in Idaho, hit me up and I'll have you go and watch. It's a lot of fun. All right. And so the enteric nervous system is actually composed of hundreds of thousands of millions of neurons. So it actually has about the same number of neurons as there are in the central nervous system, which is a lot, right? And they are embedded in the intestinal wall, as you can see in this cutout, and also in the vasculature or the circulatory system. So, you know, and if you look at the intestine as a tube, when we cut it, there are several different layers. The first layer is called the submucosa, right? And this submucosa has a lot of blood vessels that is taking blood in and out. If you think about the intestine, it's absorbing nutrients. And so there's a lot of blood vessels there. Sorry. We have the first part of the enteric nervous system called the submucosal plexus. And a plexus is just a network of neurons. That's literally what a plexus means, it's just a fancy word. And this particular collection of neurons controls absorption and secretion. And so it's secretion of hormones, all chemicals that the gut needs to work, and then absorption of nutrients or water or salts or minerals or that kind of thing. And then when we move a little bit up, we have the myenteric plexus, And the myenteric plexus is basically just another fancy phrase referring to a network of neurons that lies between the muscle. So there's several layers of muscle, circular and longitudinal muscle in the intestine. And their job, the neurons here, their job is to tell the muscle what to do. And so really that video you saw before, a lot of it is actually, the instructions are coming from the neurons in this layer. So it's pretty neat. And it's actually right from the esophagus all the way to the colon, like literally from the top right here, all the way down. These little balls you see are little neurons. They're parts of the enteric nervous system. Pretty, a very, very extensive network. So in our lab, another technique that we use, apart from, just putting poop inside intestines and watching it move, is also staining. So I think the previous speaker talked about immunohistochemistry. This is when we take chemical protein antibodies and we're able to tag or add on a color dye, like a fluorescent dye. And then when we stain, they're able to stain for different kinds of antibodies or proteins in the gut. And so here we have neurons from the myenteric plexus. Again, remember, these neurons are embedded between the muscle. They control the contractions and the relaxations. And so you can actually see how nicely they are located together, right? And so the red neurons here produce a very specific type of neurotransmitter called nitric oxide. The green is just a stain that stains for all types of neurons. And so using techniques like these, we've actually been able to identify about 10 different classes of neurons. All of them make very unique neurotransmitters and peptides that are able to communicate with the gut. And this is also a really cool image that is showing that apart from just neurons, just like in the brain, we have in the central nervous system, the enteric nervous system has what we call glia. So glia are these really nice, large cells that spread their arms and hug the neurons, and they give them food, they keep them happy, they clean them up, and they're actually super important cells in the central nervous system. We find them also, so this is also a picture from our lab, from Brian, actually one of our friends' labs, showing that glia are very closely, they're hugging neurons in this picture, as you can see. So there's a lot of glia in the enteric nervous system as well. All right, so when we try to put the enteric nervous system as part of the entire nervous system, it actually falls right in the middle, right? You can't actually say that enteric nervous system is parasympathetic or sympathetic. And this is more for all of you neuroscience enthusiasts who know what the parasympathetic system is, the sympathetic system is. It actually performs both functions, right? And so because of that, it's one of the few systems in the body that is able to very uniquely positioned in the middle of both systems, and it does both motor and sensory functions. It's pretty cool. I love it. All right, so the enteric nervous system communicates with the central nervous system using the vagal pathway, also the splanchnic pathway, but really a large portion of the communication is through the vagus nerve. So typically we know this because when we cut the vagus nerve off, there's no communication between the enteric nervous system and the central nervous system. Okay. All right, so that is little brain number 1. Wasn't that pretty cool to find out you have a little brain in your gut? Like, who knew, right? I didn't until I started my PhD. Now I do, and I hope you do too. So the second thing, though, that compounds our discussion of brains is that we have a lot of microbes around us everywhere. There are bacteria and yeast and fungi and viruses and phage and all kinds of fancy names, but basically little buggers all around us everywhere. As I'm talking, thousands of them are coming out. And on my computer screen, if I were to wipe and put it on a petri dish with the agar, that would be literally like different color organisms growing, right? So it's around us, and we all know this, right? And in the gut specifically, there's a gut microbiome, right? And people call that gut microbiome because it's a collection of all kinds of microbes in the gut, as well as their genes and their products. And they live in the gastrointestinal tract, perform a lot of different functions, but really the largest take-home message from the gut microbiome is that we need them to survive. They help us to absorb nutrients from food. They help our immune system to work. And so if you were to remove the gut microbiome from a mouse, for example, the mouse actually develops abnormally. Their immune system isn't able to fight off infections because they don't know what bacteria look like anymore, right? That's one of the functions that the gut microbiome performs. So it's actually super important. And they live on every exposed body surface. Like these numbers are like, I don't know how many zeros those are. That's like a lot of zeros, right? You can count. There's 14 zeros because the 10 says it. But if you were to try and count, that is a lot. Like, and there's only 7.3 billion people in the world. Okay, so that's a lot of bacteria just inside of your body. There are way more bacterial cells than there are human cells, and their genetic material is like for every human gene, there's hundreds of microbial genes. So you're actually more microbe. Like you're technically just a bag of bacteria walking around, like a bag of bacterial genes walking around. That's a joke that we make in the gut microbiome space. But the big thing is a lot of us have the concept that, you know, microbes that are bad or cause disease are so many. Like when we hear microbe, we think immediately sickness, disease, COVID, right? But not really. There's greater than 10,000 groups of microbes in the gut, or, you know, all across the body, and only 80 of them are disease causing. And, you know, the CDC has priority pathogens. Only 16 of them are that. So really the vast majority of them are what we call symbionts. They're there, they coexist happily with us. They help us to break down things we can't break down and then give us back the byproducts of that breakdown so that we can use it. So it's actually a pretty nice beneficial relationship when it's not interrupted, okay? So again, it's regarded as the forgotten organ. The microbiome mass is about 2.7 kilograms. The brain is just about 1.2 kilograms. So really ask yourself if we're going by weight, who has more power here? We'll see. All right. Okay. The gut microbiome is made up of different types of organisms, and I'm not going to spend a lot of time on this, but really the take-home message from this slide is that as you get older, right, the diversity or the different types of bacteria in the gut goes down. Well, actually, as you get older, it goes up, and then basically we all begin to have very similar types of bacteria in our body as we get older. Okay, so like, why do we care about the gut microbiome? Well, there's like literally thousands of papers that will tell you why, but I'm going to focus my attention on diet a lot of the time. Diet is like the big driver of the kinds of bacteria you have in your gut. And a lot of the research is telling us that the kind of diet you eat is shaping or determining the structure of your gut microbiome. It's a community. It's basically like a village, and there are different types of bacteria, and that balance is critical. But when it's thrown off, there's all kinds of diseases associated with it. So a lot of focus is on figuring out what kinds of things in our diet is actually changing the gut microbiome into a more unhealthy state, or a state that can contribute to us being unhealthy. Okay, so this is one of the studies that showed that if you use artificial sweeteners, it can actually induce glucose intolerance, which is basically a symptom or a precursor to type 2 diabetes. And it does this just by altering the gut microbiota. So they did this in mice, and you can actually see the blue line here up here is showing, like, this is glucose measurements, right? So you can see that this one is up, and it stays up above this control group. And that's basically what the point of this paper was, is we can cause type 2 diabetes just by changing the type of bacteria in the gut. And so it's super important because the gut microbiome, again, influences a lot of different biological processes. And I've talked about that. A lot of conditions are linked to microbiota changes. This is like a very short list. This is a super short list. The list now is addiction, smoking, alcoholism, like anything you can think of, there's a link to the gut microbiome. But since this is a neuroscience conference, I'm going to try and stick to the neurons, right? So there's actually a link between the enteric nervous system, or the little brain that we talked about before, and the gut microbiome. So this was a pretty cool study that determined the entire path of my future, because I read this paper and decided I wanted to be a microbial neuroscientist. What they did was to take a specific gut bacteria and then mix it up with physiologic saline solution and put it inside the colon. Okay, so before they put it inside the colon, you can see that the colon's contractions, so these are contractions, if you can picture that in your mind for a little bit. The colon's contractions were, longer apart, they were bigger, you can see that, right? Then they put this bacteria in and notice that immediately the contractions sped up like crazy, really, really, really fast. And they went on to actually show that the bacteria were doing this by communicating directly with neurons. Okay, so basically the take-home message here for me at that point in my career was, oh my gosh, bacteria can talk to my neurons, and then that means bacteria can make me do whatever they want. Let that sink in for a second. Like, they can literally control me, can control my gut, they can control my feelings and emotions. Can they? I was curious. So I wanted to find out more. So that's literally why I did my PhD. Okay. So that brings me to, is there really a link between the gut or the gut and the microbiome down all the way down here in the brain? Well, you might go like, well, there has to be, right? This is a lot of very few papers. There's maybe thousands of papers now out about this at this point, all of them showing that there's an association between diet quality and mood, which a lot of people never actually thought about. But a lot of the work now is showing that the reason diet has such a huge impact on how we feel and on even psych disorders like depression is because the diet is shaping your gut microbiome, and that is indirectly communicating with neurons in your gut, who then communicate with neurons in your brain up here. So it's actually important to physicians now to include a dietary program in managing disorders like depression and anxiety. Okay, and then when we move to disorders like Parkinson's or diseases like Parkinson's or neurodegenerative disorders, there's a lot of work now showing that there's actually a direct or indirect link between the type of bacteria in the gut and the development of Parkinson's. And I'll talk a little bit about that. So there's what we call the gut microbiota brain axis. That's what we've named it. And it basically just stands for this bidirectional or communication in and out of the big brain from the gut. And I'm not going to attempt to have you understand this figure because it is a lot, right? But really what I want you to take away from this is that the gut uses a lot of different methods to talk to the brain. It uses neurotransmitters, right? So neurotransmitters are chemicals that are produced in the gut or produced by the brain, and they're used to communicate directly with neurons. So it uses neurotransmitters, it uses short-chain fatty acids. Short-chain fatty acids are actually directly from microbes. Short-chain fatty acids are produced as a byproduct of microbial digestion of fiber. So the fiber that, a lot of the time they're like, eat a lot of fiber, eat more fiber, eat more fiber. This is literally what happens when you eat fiber. The bacteria break it down in your gut and uses that fiber to produce short-chain fatty acids, which then those short-chain fatty acids are able to actually communicate with the neurons that are here in the immune cells, and that actually then gets back to your brain. So it's super important. And then the third part of the gut-brain axis is the immune system. The immune system is probably one of the largest components of communication. So when the immune system is impacted in the gut, there's actually typically some kind of communication that tells the brain. That's why sometimes you feel pain here when you have an infection, right? You have a stomach bug. You can remember the last time you had a stomach bug, it was terrible. That's why they're so painful, is because there's actually these nerve endings from the vagus that is getting that information directly from immune cells, right? And so it's way more painful than typically something else that's going on. All right. So one of the pretty cool pieces of work that I wanted to share with you, not from our group, but from a different group, is that they found that gut microbiome changes. So basically, if you change the type of bacteria in your gut, you could alter human brain activity. And they did this using imaging, fMRI imaging. And so the different colors are really put in there by the researchers just to show that when they give people before and after probiotics, they're able to see a shift in the kind of brain activity, or the types of the parts of the brain that are active after some time. So basically, the insula is more active, and it goes to periodactyl gray. I'm not going to attempt to mention all of these, but the take-home message here is that they were able to show that changing the gut microbiota could alter human brain activity, and they did this in people, not in animals. So that's actually super important. And then, as a researcher whose work I love, Elaine Shao, over at, I believe, UCSF at this point, or UCLA, I can't recall. But Elaine Shao has given a very cool TED Talk on the subject. If you are interested, it's on YouTube, go check it out. And her lab basically has shown that if you take mice, right, that are either germ-free, they don't have any gut bacteria, or you change the type of bacteria in their gut using a variety of different methods, you can actually change their communicative behavior, okay? And so, for example, this is like a study that she did where she was given mice a bacterium called B. fragilis. It's a very common component of probiotics. So if you take probiotics, good for you. This is in there. And what she showed is that the control group is this, they have a call, the mice have calls to each other, like they're talking to each other, not with a mobile phone. It's not like mice have phones that they're using, but they make sounds that they are using to communicate with each other. And they were able to show that in one group that were very low vocalizing, or they didn't communicate a lot with the rest of the mice, when they gave them the fragilis, that communication went back up. And the group that was not communicative or very communicative is actually a special kind of mouse they developed to study autism spectrum disorders, right? And so the point of this study was to basically show that they could help patients with autism by just giving them this common probiotic. Okay. And then they also showed that in the same group of mice, when they gave them the same bacteria, anxiety-like behavior was actually less. And so they were able to basically treat anxiety, in quotes, in mice by giving them this bacteria. Social behavior is the same. I'm not going to spend a lot of time on that slide. Going to skip this because it's a lot. And basically just the take-home message here from Elaine Shao and a lot of her compatriots' work is that the type of bacteria in the gut can actually play a role in the development of autism spectrum disorder. And basically, a lot of people are looking for ways to treat autism at this point, autism spectrum disorders, ASD, by giving people either different types of bacteria through probiotics, or by figuring out how the bacteria are potentially contributing to ASD symptoms so that they can then help to treat it better. So I think this is a pretty cool piece of work, that relates very closely with a lot of the interests, or a lot of the things that we're concerned about recently in the world, right? And then this brings me to neurodegenerative disorders. Neurodegenerative disorders are near and dear to my own heart. My aunt and my grandmother died from neurodegenerative diseases. And so I became interested in this at the latter part of my career as a PhD scientist. And I wanted to figure out if there was really a link between the gut microbiome and the development of diseases like Parkinson's or Alzheimer's. And I realized I was late to the game because there were already like companies that were producing products for this. Like Regenx is a company that is actually actively studying this. Okay, but basically the summary of the work with Parkinson's so far is showing us that when we study the development of this disease using mice with a gut microbiome or without a gut microbiome, we're able to notice that there's a difference between disease progression in the two groups. And so the group that does not have a gut microbiome, for example, basically does not develop Parkinson's symptoms. They did this in the lab and showed that you needed gut bacteria to produce short-chain fatty acids, which then was reacting or setting off your immune system. And that was actually what was leading to a lot of the pathophysiology, or that's just a fancy word for the development of the disease using cellular models. And so this was actually really compelling in mice. But then you ask the question in humans, is that the same? Well, we don't know because the diagnosis of Parkinson's at this point is typically the disease is pretty advanced by the time it's caught. But what we do know is that one of the very first symptoms that Parkinson's patients will show is impaired gut function. They'll go to their doctor and say, hey, I can't go to the bathroom as often, or I'm going to the bathroom too often, or I have too much pain in my gut. It's a very common complaint. Over 80% of people who are later diagnosed with Parkinson's and Alzheimer's will actually go to their doctor with these symptoms. And so that tells people that, okay, maybe we need to start paying attention to what's going on in the gut when people come to us, you know, telling doctors, when people come to them and complain about these symptoms, especially past a certain age, they need to start paying attention. And that seems to be working out in some places where clinical trials are happening. And in Alzheimer's, it's pretty much the same, right? In Alzheimer's, we have what we call an imbalance. Dysbiosis is just an imbalance of the gut bacteria, which leads to the formation of the very unique plaques. Or there's a very, it's a very common symptom when you cut the brain of a Alzheimer's patient open, you're going to see what we call these tangles and plaques. They are formed by the deposition of a very specific type of protein. It happens best in the gut. So when people pass and they do autopsies and they check, they're actually able to see that there are numerous plaques of these in their guts before it actually begins to go to the brain. So there's actually a hypothesis now that amyloid plaque formation begins in the gut and then it's transported. These proteins are then transported to the brain. And that happens in Alzheimer's, but also for tau in Parkinson's disease. All right, I'm going to skip that, okay? So I did a lot of my work with mice, okay? This is just going to be like an ending take home a little thing about some of the stuff I did when I did my PhD. And what I would do was use diet. So a high fat diet. The mice were eating a 72% high fat, which is like, I believe, higher than your typical McDonald's meal. Okay, so your typical McDonald's meal is about 60%. I was using like 72%. That's a lot. And what I would do was feed both male and female mice this diet. And after 8 weeks, this is how fat they get. You can see that the ones that get the green or the healthy or the low fat diet are still pretty skinny. And as expected, the group that eats the fatty diet gets fat. Of note, this only happened in male mice. So yay for girls. I'm kidding. But the girl mice did not get fat. And it was one of those points in my career that my PI, my professor was like, are you sure you're feeding the female mice as well? I'm like, it's not like we're force feeding them. We give them the food and they eat it. So, and they were eating the food. It's not my fault that they're not getting fat. But it really was, and it ended up becoming a super cool, study on its own. And I'm not going to go in that direction. But really, the take-home message was we were able to establish that if we fed mice a high-fat diet, we got them to be fat, we got them to develop diabetes, and we actually changed their entire community in their gut. So we're able to change the type of bacteria in their gut quite significantly. Okay. And then what we noticed then in our mice was that we were in females that were not obese and they didn't have diabetes at the time that we took the samples. They actually had damage to the neurons in their gut. The neurons were dying. They were super unhealthy. And this is a video, a screenshot from one of those motility experiments I was showing you earlier. What we noticed was that the standard child mice were able to process food. The contractions were smooth and regular. You can see that's a nice contraction right there. That's another contraction. In the high fat group, that was not the case. They basically had constipation. Okay, so the food would literally be sitting in their guts, not moving at all. And this was because their neurons were basically damaged, right? And so what we found helped us to form this little framework, which I'm going to expand here, right? And it helped us to realize that when we induce diabetes, before we induce diabetes and obesity, or if you induce changes to the gut microbiome, what you actually do is cause inflammation, you cause oxidative stress, which then leads to gastrointestinal dysfunction, or, the slide I showed you before, where they have constipation pretty much. And then what you also get when you do that, I actually went on to show this, is that you have damage to neurons in your brain as well. And so that leads to a lot of these other disorders that we don't know a lot about, like Parkinson's and Alzheimer's and dementia or major depression disorders that are linked to death of neurons and damage to neurons in the brain. Basically, a lot of this, the center is this gut dysbiosis, changes to your gut microbiome. So the big thing then is, how can we use this knowledge? This is information, right? Well, first of all, try to eat healthy, right? And I know you've heard that a million times, but really it's important. What you eat really does matter. It doesn't mean that you're only gonna be eating salads and all of that. Balance. So try not to tip over to eating McDonald's every day. I hope you don't already. But try not to be on the extreme side of anything. Be smack in the middle. You want to eat McDonald's once in a while, that's okay. It's not like you're going to die eating McDonald's. That's not what I'm saying, okay? I'm saying that you need a balance, right? And in order to get that balance, you need to make sure you're eating a lot of different types of things in your diet. So that means that eat that salad, eat that smoothie, eat fruits, eat veggies, eat all that fiber that your parents after you to eat. I know I struggle with that a lot. But really on the drug development side, we have what we call microbe-based therapeutics now. There are a lot of companies who are developing different drugs that are going to help us to treat all kinds of things, from diabetes to Alzheimer's and Parkinson's. It's a very active area of research. But the most interesting microbiome-based drug came all the way from our great-great-great-great-great-great-grandfather Hippocrates, the father of modern medicine, as he's called. He said all disease begins in the gut. And what he meant by that is that you could actually use poop to treat disease. So in his time, people could actually take poop solutions and give it to animals or people, and they were able to treat different types of diseases That has come back now. So you may have heard the phrase fecal microbiome transplantation, or you may have heard the phrase FMT, fecal transplant. All of them refer to basically taking poop from a healthy person and giving it to someone that is struggling with a kind of disease. And so a lot of people have focused their attention on Clostridium difficile. I think it's Clostridiatis now. C. difficile infection. C. difficile is a very, very scary pathogen that unfortunately kills a lot of people every year. When, especially in this hospital acquired, it's resistant to antibiotics, and it comes back over and over and over and over. But what we're finding now is that if we give people poop pills, okay, poop pills from healthy individuals, it doesn't. It actually goes away and it doesn't come back, and people are cured. And so a lot of my work right now is working with companies that are working on these products, trying to get them to patients. A lot of them are in clinical trials. It's very exciting. But really, we're getting to the point where maybe 60 years, well, 60 years is too far. 10 years from now, you may be able to get a prescription of poop pills for that ailment that you may have. You may be able to get a prescription of poop pills to help lose weight, right? Because your gut bacteria is making you fat. And so you can go like, hey, can you help me? Give me poop from a skinny person. This is real. Like there's kind of clinical trials going on right this moment that are doing this. So it's very exciting times for this field. And I hope for anyone who wants to get into it, they do. Just remember that it takes a village to get anywhere. It's taking me a village to get here. It will take you a village to get where you want to go. But I want to be part of that village, right? By giving you some advice. I've already told you first, make sure you pick a good mentor, someone that will support you. And the other piece of advice I can give you is to try to support other people as well, right? So as you're pursuing your own dreams and your own goals, try to be the village for other people so that when you need a village, you're not alone. It's very important to remember that we don't exist in a vacuum and we can't just take all the time without giving. So give. Really try to give, try to serve, try to be a really proactive person when it comes to being a village to other people. And so whatever someone else around you, their dream is, their goal is, their vision is, try to encourage that, because you have your own dream and your own vision. Someone else will encourage you. In Africa, where I come from, we have a proverb that says that he who waters will himself be watered. And it basically means that when you give, you get. And I think in India, we call that karma. I don't know. I don't know much about that. But really, just remember, those are the two most important, those are the things that have helped me personally to come to this point, is to remember that I'm not alone, number one, and I need people. Like I really did need people to get where I am. But the other thing is remember that your gut feelings do matter. Don't ignore your gut symptoms. They're very important. Your gut could be trying to tell you something. So when it says I have a strong opinion, listen. Of course, in moderation. Okay. All right. So this is a puzzle. You can feel free to grab a piece of paper and a pen. I am the daughter of two teachers. I always jokingly say that, but my mom and my dad were both teachers. And I chose not to teach much anymore, even though I still have to. But really, I want to be the teacher and give you a quiz. So this is just a fun puzzle. The first clue is basically, what is the complex community of microbes in the gut called? Okay, this one. It starts with an M. It's pretty easy. And then down, the clue is the name of the little brain of the gut. It starts with E. Well, the first word is enteric, if that helps. And then the last one is the number of major networks of neurons in the gut. Remember, we talked about this? The myenteric plexus is between the circular muscle. The submucosal plexus is with the vasculature of the circulatory system, right? So how many? That's easy, right? It's just 2. All right. So that's That's the solution. I hope you got it. It's just a fun quiz. It's not really anything. I wish I had prizes. Maybe I do. Shoot me an email if you got it all right. And maybe I'll send you a prize, okay? But I just want to thank everybody, my village of people who's brought me this far. Like the individuals on this slide have changed my life in crazy ways and made me a way better person. And I'm so thankful to have come across all of them. And that's the fun part about being in science is you meet cool people and you work with cool people and you rub off on each other. And at the end of the day, you're both better at the end, hopefully, right? Hopefully, if you work with the right people, you're both better at the end of the day. So I've been I want to admit I've been very lucky. But really, I just want to close by saying thanks again to the crew of people who put this together. I'm so impressed by this entire event, the organization that went into it, the time that went into it. And Inchara was my point of contact emailing me. She's pretty awesome. Like, you guys are amazing. And I want to say I'm available over email for anything you're curious about. I love to talk about different things. I have a Clubhouse account. You can email me for it if you want, but I typically talk about science, going women in science, Black people in science. Those are two of my favorite topics to talk about, kind of the things that can help you to succeed. I also talk about mental health disorders a lot. I've struggled with my own issues in the past. I had a best friend that unfortunately, committed suicide, and literally the week before my PhD defense. And so it's one of those things that's near and dear to my heart. If you ever need someone to talk, come talk to the random stranger that just gave a talk at Simply Neuroscience. But really, like, talk to someone if you struggle with some of this stuff. And I know this is a very simplistic presentation and simplistic view. It's a very complex issue, but I just want to say that it's super important that you reach out when you need help. Connect with your village, basically, right? Talk to people. But really, thank you. I've really enjoyed giving this talk. If you have questions, please, love to hear them. Let's see. I don't know if you have questions in there.
Yes, we have one question. It says, based on the information you presented, do you support or recommend all individuals taking probiotics as a way to support good health?
Good question, Jessica. Yes, I encourage people to take probiotics. I'm not a doctor, so please, this is not medical advice. And do remember that probiotics are currently not really considered drugs. And so they don't undergo the kind of stringent controls that a lot of other drug products do go through. But as supplements, they can be pretty helpful. And I have heard some pretty cool testimonies from people that have taken probiotics and seen improvements in their health overall, right? So, I mean, it can't hurt. What I would encourage is to try to find a good company that makes reliable products with strong reviews. You can ask your doctor before you start. And ideally also support that with a diet high in fiber, or add in prebiotics. So there's prebiotic supplements you can buy in addition. So the prebiotics actually help the probiotics you're taking to basically take root in your gut and to stay. So it supports their growth. So that's typically what I recommend, is not just the probiotics, but also remember your diet as well.
And then we have one more question. Someone in the chat asked, do you think natural and herbal medicine is better than swallowing drugs? And what do you think is the best solution to people suffering from issues with the nervous system?
Yeah, that is a really complex question. What I can say is, in my lab, we do study herbal natural remedies. So my professor has a drug from Africa that he studies, and we were actually able to establish that it's very neuroprotective and it's natural, doesn't have any side effects or anything. There are a lot of similar products like that around the world. But I really hesitate to say that if you have a medically diagnosed condition and they've put you on a treatment regimen, I hesitate to say don't take the drugs and take a herbal, because there are so many herbal regimens. Which one are you going to take? Like that you don't know which one is going to work on which one isn't. And so in all of this, I encourage, again, just make sure you work with your doctor. You can work with a holistic doctor. So there are doctors who are able to work with you on more holistic therapies beyond traditional pharmaceuticals. And so they can help you if you're considering doing like drug stuff. But again, this is a super complex issue. So medical advice takes precedence over anything in a random person on the internet says, basically. Okay, so just remember that. But that being said, there are some medical doctors who tend to be more proactive about alternative therapies for their patients. And so if you want more, scientific publications in that regard, there are a couple medical doctors who have pretty good papers in that field. I can send them to you if you email me. And I see someone saying they can't wait to take some pills. Okay, Erin, I see you. I can't wait to. I think it's gonna be pretty cool when we get to the part time in our world when poop pills are normal. Yeah. Anyway, so more questions? Or are we all good? I think I got them all.
Yeah, I think that's all the questions as well. Thank you so much for presenting. I've learned so much about your field and the gut microbiome.
Yeah, so that was a lot of information. Yeah, so thank you again, guys. Thank you. Enjoy the rest of the presentations.
Yep, thank you so much, Dr. Nyavor. It was really nice having you and really enjoyed your talk.
Thanks.
Now I'll hand it over to Ryan, who will be introducing the next event, which is an LGBTQ+ roundtable.
Hi everyone, I'll just be jumping in really quick before Ryan. Thank you all for being here. Super excited for this next panel. I'm going to introduce Ryan really quickly, who is our official host for this session. Ryan Cabote goes by he, him pronouns and is currently a psychology major at Santa Barbara City College and plans to transfer to a four-year university next fall. He's the CEO and founder of Mental Health United, a student-run nonprofit mental health organization driving the culture of mental health from awareness to action through mental health resource integration, and also mainly integrating mental health first aid as mandatory training for students and faculty in higher education. As an LGBTQ+ person of color, he strives to advocate for and take action for the LGBTQ+ community and various underdeserved communities in a plethora of ways. In his free time, he enjoys reading self-help books, finding new alternative underground artists on Spotify, and journaling. Thank you so much, Ryan, for being here. And I'll go ahead and hand it over to you so you can introduce our speakers for today.
Thank you so much, Sasha, for the introduction. And I appreciate you guys for having me on today as the moderator for today's LGBTQ roundtable discussion. Today, I'm going to be introducing the amazing panelists that we have that are also part of the LGBTQ+ community and are prominent professionals in neuroscience. First, we have Dr. Priya Silverstein, who goes by the she/they pronouns. She is currently a research scientist at the Center of Open Science and a community manager at the Journal Editors Discussion Interface. As well as this, they continue to work on projects in predominantly developmental psychology and trained researchers at all career stages in open and reproducible science. So let's welcome Dr. Priya Silverstein. Next, we have Dr. Justin Matthews, who goes by the he, him pronouns, and he's an associate professor of general experimental psychology at the California State University, Monterey Bay. He teaches courses in statistics, research methodology, and experimental design. His research blends cognitive science and social psychology in the realms of spatial cognition and human depiction in popular print media. Justin serves as the Vice Chairperson and Commissioner on Monterey County's Equal Opportunity and Civil Rights Advisory Commission, representing LGBTQ county residents. He is a first-generation college graduate and was born and raised in California's central San Joaquin Valley. Our next panelist, we have Dr. Tareq Yousef, who goes by the he, him, his pronouns. He is a PhD candidate at the Medical Neuroscience at Dalhousie University in Mi'kma'ki, the territory of Mi'k people in Nova Scotia, Canada. Apologies for the pronunciation, correct me if I'm wrong. And he, Tareq's thesis investigates the mechanisms that contribute to vertebrate light adaptation with focus on the role of neuromodular-driven gap junction coupling of horizontal cells and their electrophysiology. Alongside a passion for teaching and science literacy communication, Tareq strives to create better inclusion for equity-seeking populations in academia. And finally, we have Dr. Angel Kaur, who goes by the she, her pronouns and is an assistant professor of neuroscience at UNC Asheville. She's a neurobiologist by training, and her research focuses on high impact practices and undergraduate STEM education in a liberal arts environment. She is particularly interested in the pedagogical innovation to increase student engagement and promote deep learning. She's originally from New Delhi, India, and holds a PhD in cellular and molecular neuroscience from the Scripps Research Institute and a bachelor's degree from Bryn Mawr College. Dr. Kaur is currently serving an associate faculty fellow for the UNC System Digital Learning Initiative and will also be joining the UNC Asheville Center for Teaching and Learning as a faculty fellow in July 2021. She's also an artist, graphic designer, and a mom to 6-year-old twins and a 75-pound puppy named Lynn. Given that, I would like to introduce all of our panelists, and we'll just go ahead and start with our first question, which anyone of the panelists can answer, and that is, what has your experience been as an LGBTQ plus in the STEM community? And could you just elaborate your experience on that in the work field and academia? Whoever would like to start first. You can go ahead, Dr. Matthews. Oh, you're muted.
Unmute. Oh, there we go. Okay. Hi, I'm Justin Matthews. I'm associate professor at CSU Monterey Bay. I mean, just to break it down from the beginning, I came out in grad school. So for me, it was kind of a learning experience while in grad school, earning my PhD. And I'm from California. I went to grad school in California at the University of California in Merced, which was the newest campus in that system at the time. And it was a very small campus and very supportive and racially, ethnically diverse. A lot of first generation students, a lot of students from California, but also outside of California. So for me, I was very fortunate to be in an environment that was supportive of me coming out and being very honest about myself with everyone, including my PIs, my professors, my fellow grad students, friends. So it was a very supportive experience, and I was very fortunate for that. And then transitioning into a faculty position, again, I stayed in California and I ended up at CSU Monterey Bay, which I have come to find out is also extremely supportive. So there are little blips here and there, running into certain people who might not feel as comfortable with, who you are, but for the most part, it's been fairly supportive. And because of that, I've chosen to get more involved in my community representing LGBT people at a community county level. So overall, my experience has been fairly positive, not 100%, but it's been fairly supportive.
Thank you so much for sharing, Dr. Matthews, and I'm glad to hear that your experience has been supportive overall. I did also want to mention just briefly, because I just remembered now, but happy Pride Month, everyone. Hope that everyone's Pride Month is going well and is productive. Moving forward, I guess we can hear from Dr. Silverstein about your experience.
Yeah, I had to ask for permission to unmute, so sorry for the pause. Hiya. Yeah, thanks so much for having me. I'm so excited about this panel discussion. My name's Priya, and I think my experience has been really mixed as well, because I think I've been out as kind of either bi or gay. That has like fluctuated throughout my life, but since I was a teenager, so I think that part was easy, like going through university and my PhD and workplaces. I didn't really ever seem to have any negativity about me having a wife or being with women, but I think that part was really easy. But then when I started kind of, I wouldn't even say questioning my gender identity, because I wasn't questioning anything about myself necessarily, but I was just questioning what it really, how I'd always felt, what that means, basically, if that makes sense. Like, I'd always taken it for granted that before, like, pronouns were really a thing, that I would just use she, her pronouns. But then when I really started thinking about it a bit more, then I started thinking, well, that's not something that definitely like resonates with me. It's just that I just always have, because it was the thing that was done. And so then that became not difficult, but I kind of almost didn't, I had this sense of like not wanting to make a fuss, like at work, and not wanting to make people feel uncomfortable by mentioning it and asking them to change it if I was still comfortable with like female pronouns, but it just wasn't necessarily my favorite or what I identified with the most. So I struggled a little bit with that in academia. And then when I kind of was brave enough to like kind of put my pronouns in my bio, in my email signature, and kind of explain to my colleagues what this meant, I kind of, well, my boss at the time just never emailed back when I kind of wrote this like email pouring my heart out and explaining, kind of trying to really walk them through it, because I knew that they kind of didn't know anything about this stuff. So that part was more difficult. And I found that in one of my current roles, which is like not in traditional academia, but is still in research and still in science, I found it so much easier. Like, everyone just by default has their pronouns in like, you know, in their Zoom name and things like that. And my whole team is like predominantly queer, and that is like much nicer for me. But yeah, overall it's been completely fine. It was just that kind of little bit in the middle which felt a little bit uncomfortable. Yeah.
Thank you so much for sharing about your experience, Dr. Silverstein. I would like to say as well that we should definitely try to create a comfortable environment, whether that be in the workplace or outside of the workplace. So thank you for sharing that. I guess we can go off and pass it on to Dr. Yousef to share about your experience.
Thanks so much, Ryan. And I appreciate you jumping the gun a little bit, but I haven't quite snatched the doctor thing yet, but almost there. Thanks so much for having me today. And I'm so happy to be here with other queer people talking about being queer in Pride Month. I think this is like a really good experience for me. Being able to share space and time and commune with people who have similar experiences. I would suggest to all future students to try to take advantage of that as much as possible. That's something that I haven't necessarily been able to take advantage of. During my time at Dalhousie, not that Dalhousie is a terrible place to be queer, but trying to create space for queer people can be difficult anywhere, unfortunately. And trying to make the time for that when everybody's busy with grad school and so on makes it even harder. I haven't necessarily been able to have the opportunity to engage with many queer faculty or many other queer graduate students. So I try to do that as much as possible in any extracurricular time that I have, whether that's through advocacy work or just, getting to network and getting to know people. I definitely agree that there's been a lot of support. I'm very lucky living in Canada, of course, that there is support. And even though I know a lot of queer people probably would identify with this, like we're always coming out. It doesn't really stop. You just are always coming out to people for the rest of your life. It does get easier with age, I find, but you're always coming out. And I guess like I've learned to find a way, like in a professional academic setting where I'm not even necessarily coming out anymore, but I'm through my conversation, I'm queer because I mentioned my boyfriend over the weekend. Like we went and did this kind of thing, or we went to a park or whatever, and I was with my boyfriend. And I'm very privileged to be able to have the safe space to do that. So I've been able to experience that kind of safety for sure. Yeah, thanks.
Yeah, thank you so much, Mr. Yousef. I can also relate in terms of the coming out part where you have to come out every time you do meet with someone, and it does get easier with age, but at the same time, I also feel like we should try to maybe destigmatize coming out and not normalize just not having to come out to people and just make it known, whether, like you said, oh, my boyfriend, or through casual conversations rather than just strictly saying like, I am gay, I am bi, I'm lesbian, so on and so forth. So definitely appreciate you sharing your experience with us and hope that a lot of people can also relate. And now I'm going to pass it on over to Dr. Kaur, who hopefully can share her experience.
Yeah, I'm going to echo a lot of what my fellow panelists have said. I've been lucky to be in safe spaces for most of my career. I came out in college, but I went to a Seven Sisters school where there was a solid queer spirit that welcomed you with open arms. And so I've had that community forever, right? Once it was there, those are people that walk with me every day, even if they're not anywhere nearby. And so even when there are those things that happen, as Justin said, here and there, it's easier to put them down when you have that community with you, and you know that you don't feel so isolated because they're always just there. I did have to, as you said, you have to keep coming out, and I did kind of formally come out in grad school to my PI because it just hadn't come up before, and it came up when I had an engagement ring on my hand, and I had to say, yeah, so she's a grad student, blah, blah, blah. And that was the extent of coming out. And I think that there's nothing wrong with that. I mean, that's how other people tell us about their lives when they mention their husbands or girlfriends or what have you. So we're just taking up the same space and doing it the same way. I'll be honest, when I was first engaged, I got quite a thrill out of correcting people. Like I'd go and buy some chocolates and then be like, oh, are you getting it for your fiance? He's so lucky. And I'm like, yeah, she is. And just take a little thrill of it, because it doesn't actually hurt and the person didn't actually mean anything bad by it. It's just their particular experience, and just kind of go with that. As a faculty member, I landed at UNC Asheville, which is a public liberal arts university and is in a very queer-friendly town, is itself a very queer-friendly place. I can think of so many queer faculty that are in our, and staff that are in our ranks and are in high up positions in our university's administration. And so here, again, there's been the one or two people that will say one or two things. But overall, I've felt very supported from that perspective, just because I can see the representation of queer folk across our campus.
Thank you, Dr. Kaur, for sharing your experience. And I just would like to say, I know I have noticed a pattern among our panelists that have had supportive experiences overall, but for those who don't have that supportive space or who are unable to relate, just know that there are supportive spaces out there for you guys to reach out to, whether that be through your local LGBTQ plus organization or even through your school and academic and extracurricular activities. They are out there, and we are all here to support one another in our own experiences. So with that being said, I know that we do have a lot of youth in this, this Zoom and this convention today, and was wondering if you guys, as the panelists, have any advice for the LGBTQ plus youth that are going into STEM who may be struggling with their own identities.
I mean, I'll go first on this one. It connects very well with what I was just saying before. One of the reasons I've been in safe spaces is because I have made that happen. I made the choices to pick spaces that would be comfortable. When I first moved to UNC Asheville, it was everybody knew that this person is queer and their partner is coming and all this stuff. And we were welcomed in that way, which is one of the reasons we chose to come here. And so rather than, this is why I just wear my queerness out all the time. It's like, I have so many aspects to my own, like there's a lot of minoritized aspects to my own identity. And so it's often hard to say if any sort of microaggression is happening because I'm queer, or is it because I'm a brown woman in STEM, or there's a plethora of reasons to choose from. But I completely lost my train of thought. I hate when this happens. One of the things that is true about me is I have ADHD. And so it's almost 4:30. That means my medication is starting to come down, and I lost my train of thought. Safe spaces, choosing them yourself. One of the reasons why I wear my queer identity out on my sleeve all the time is because I want to only be in a space where I can be myself. And if someone is going to push back against that, I want to know that now. And it can feel really scary, right? Like you're interviewing with the PI and you really want to work for them, but do you really want to work somewhere where you will feel belittled all the time? There's going to be another scientist out there who will be a better and more supportive mentor for you, who will still get you to the places you need to get to. And it's not worth the mental health issues that will come from not putting yourself in as safe a space as you can.
Yeah, I just love to echo that, Angel, because yeah, I think especially in academia, it can sometimes feel like you're meant to just like move wherever you can find a job, or wherever you can find the best job, wherever the best university is that will take you. And I just really want to, yeah, I think it's so important to choose. You're going to be living somewhere for however long that is, and that's really important to choose somewhere, both the institution itself, but also the place that you're living in, the city or the town, choosing somewhere that you know that you're going to be, or at least you hope you're going to be comfortable. And I think that's been really a struggle personally for me and my wife, because between us, we kind of hit a lot of intersectionalities. I am white passing, but I'm not white, and my wife is definitely not white passing, and we're both obviously queer. And so finding a place that we would want to live that kind of hits all of those boxes, like is multicultural, but also is like queer friendly, is difficult. And, so I think in turning that into kind of advice for young people, it's okay to pick somewhere to go to university, to go to college, to start a job. Like, you're allowed to take into account what that place is like, and that's completely okay, and that's not a bad thing, that's a good thing, and that will make it more likely for you to find community there, and never see that as a downside. And I think, my other advice, kind of unrelated to that, is that I think we've all seen during the pandemic, online community is so important as well. And I think that personally, even though at a lot of institutions I've been at, there haven't necessarily been that many queer people there, even though everyone has been fine and supportive. I found academic Twitter to be like the main place that I found like my queer scientist community. And yeah, that has been really, really helpful to me. So, and there's never too early a career stage to engage with academic queer Twitter in that way. So that would be another piece of advice from me.
Thank you both for sharing such valuable advice, and I hope everyone here can take in that. Mr. Yousef, do you have anything to add?
Yeah, I think what Dr. Kaur and Dr. Silverstein said, I relate a lot to it actually. And I wanted to talk a bit about building community and building relationships. For on the one hand, speaking of, being in the right lab, I do think that the supervisor relationship is a really important piece of the puzzle in deciding where you're going to end up working. And I'm sure people have heard this before, but I guess there's a nuance there. Maybe there's not necessarily a right answer. Maybe you are somebody who wants to put up with someone not being, the best supervisor, not necessarily mean, but maybe not always there to support because you're really passionate about this line of work, for example. But I found, at least in my experience, I've been in a lot of different labs, through summer projects and so on. The supervisor relationship is really important. And at the end of the day, when you put your head down on your pillow and you're not thinking about how stressed you are because of the treatment you're receiving, that's a really, really beautiful thing. It's a really great gift to have. And I would totally echo what was said about, finding a place where you are comfortable. That is very meaningful. And it doesn't mean that you're not going to get to where you're going to be, like Dr. Kaur said. In terms of making community, like I said before, it's not like, where I'm from, it's not necessarily like it's not queer friendly, and there are queer people, there is a queer community, but it's not very big. So for example, COVID was really difficult on the queer community here in Halifax. We lost our very last gay bar because it went bankrupt over the pandemic. So it's kind of like that sort of thing. Like we don't really have much infrastructure here, at least. So I also, like Dr. Silverstein, turned online. I have learned to, this is something that's kind of scary, but I've learned to kind of cold call every once in a while when I see a queer faculty member, for example, whose work I really admire or it seems really interesting. I'll just send them an email to say, I really like this. This is very cool. And I came across it in this way. And this is what I think about it. Not necessarily expecting a reply because I know how busy people are. But it's gone really positively so far. I've gotten to have little conversations online with queer people who have found success in academia, and that always feels really empowering. In terms of making queer friendships, I'm sure whether you're in high school or you're in your undergrad degree, I'm sure it feels different talking to somebody about your degree who's in the same classes as you, as it does talking to somebody who is not doing anything in your degree at all, but is the same age as you. So even though you're kind of in the same generation, there's just things that like they won't get the little idiosyncrasies about that professor, or they won't get, what it's like to be in this kind of lab course because of the kind of hours that you have to put into for writing this report, for example. So I haven't necessarily been able to have that here with a number of other queer students, but I've tried to find it online. And again, just, even though it can be stressful sometimes, social media, sometimes it can be really positive. So again, just trying your best. It doesn't always work. You might not always get a reply, but sometimes you will. And I have great friends that I've made that I talk to every day now because of that.
I think that that's a great advice to cold call other people and have never really thought about it in that way. So I'll definitely try that out for sure. And I also do believe that reaching out to the general online community has been super helpful, especially for me during COVID, because I have personally used Discord, if anyone's familiar with that, just to talk to other queer people who are interested in STEM and interested in other things that I am interested in, and really creating that community and making sure that we have that safe space. So moving forward, Dr. Matthews, do you have anything else that you would like to add?
Sure. I guess the first thing I would say is, as a queer faculty member, we are out there, but the student might have to do a little bit of detective work to find us and connect with us. If you're already on a campus and you are, deciding whether or not to come out, to seek out some mentorship or community, first of all, remember that you have Oftentimes you have community queer groups that are academic related. So oftentimes they'll be on campus that are sponsored through the university. And if those seem a little spotty or not really there, remember to also search in the community outside of the university. So you have PFLAG chapters, you have chapters, queer support groups that you can also look for. So don't just look in one area, but you might have to diversify a bit and look outside the university as well. Now, in academia, I often find that most queer professors that I know are openly queer in the classroom, and students know that we are queer. And so I have a lot of students that will approach me and talk to me about queerness and about their dreams and aspirations. And in class, I'm not like on day one telling everyone that I am queer. But I have no problem telling anyone if they ask me. And if you go to my website, there are, certain links and so forth that suggest that I also study things that have to do with the LGBT community. So I think as a student, you might have to just do a little detective work and find those faculty members and connect with them. And I have no problem doing the cold calling thing either. I have many students who will just email me saying, I'm interested in your research. Do you have any positions in your lab? And then come to find out those individuals are queer, and they also want to connect in that sense. Like they want that queer mentorship. So yeah, you might have to do a little bit of detective work to find us on campus, but we are everywhere. So you might also have to go off campus if it's not super obvious on campus. Maybe look for community resources as well.
Thank you for that advice. And I actually do have a question just out of curiosity, because I know that you guys have mentioned that it is a bit difficult to find queer faculty. Do queer faculty put, say, in their About Me bios that you explicitly say that you are queer, or is that just something that you have to actually find out by asking? Or do any of you guys have experience with queer faculty who actually state in your About Me bios or anything along the lines of that you are queer?
I can go first. If you go to my Twitter profile, it's obvious. My official university bio, I do not believe it says that I'm queer in my official university bio, but if you link to my website, there are indications there. We also, at least on my campus, there's also an LGBTQ staff and faculty support group. And so I think that listing of individuals is public somewhere on social media. So there is a list of individuals that have elected to be open on a list that students can seek out support from. Not all campuses have that, but if yours does, you might want to look into that a little bit for a quick list of people you could connect with.
Mine is exactly the same as Justin described. It's clearly listed on Twitter. It's not mentioned on the university page, mostly because I don't study anything related to queer stuff. And there are articles linked on my actual website that talk about my queerness. So
I don't have an institutional profile at the moment because I'm kind of in these like alt academia jobs. But when I did, I think I did. I did have something. It wasn't like stating that I was queer, but similarly to how we were talking about ways of coming out subtly, something like, if you have anything about your personal life at the bottom, like, in my free time I enjoy hiking with my wife or something like that, then that's a way of like being out without having to like state I am gay. Like yeah, which might feel out of place on a university bio, even though it shouldn't be, but yeah.
Mr. Yousef, do you have anything?
Oh yeah, so I'm not a faculty member, so I don't necessarily have an institutional bio in that sense. I try to I try to make it known, I guess, like for incoming students that I am queer. It feels weird to say because it's like, it's been so long, but I guess I'm a senior PhD student now in my department. So whenever new master students or new PhD students come in, I just try to make it known that I have a safe space to offer them. Yeah, so just in informal conversations, I definitely do try my best. On my social media, it's really obvious I'm queer. I talk about it there. Yeah.
Okay, great. Thank you guys for that. And moving forward with the next question is, what can LGBTQ plus professionals / people and allies do to aid in making progress in the neuroscience community and beyond? And what can LGBTQ plus persons and allies do beyond Pride Month to advocate for the community in the STEM and workplace or outside of workplace?
I hope if it's okay, I'd like to go first because I think I have a little short thing, and I think I'll gain a lot more to hear from what the other panelists are going to say. I'm looking forward to learning from all of you. The first thing that I thought of was, I think a lot of people are not as privileged maybe to be in positions where they feel as safe as I do, for example, and they're just sort of surviving. You know, a lot of members of the queer community, like trans women of color, are especially surviving sometimes in different places around the world. You know, such harsh realities, like, you know, like I always think of like Sarah Higazi in Egypt waving a pride flag. And unfortunately, you know, the end result of that is her having ended her life. You know, people still get tortured and imprisoned for being queer, et cetera, around the world. But or like I sometimes I even feel like a second-class citizen just because in Canada I still can't donate blood, which makes no sense at all. So I think my only piece of advice here is that I think people really need to take care of themselves as much as possible. Taking care of yourself and making sure you are the best person that you can be and that you're healthy and that you're mentally well will let your queerness shine through as powerfully as it can. And that in itself, I think, is a bit of advocacy when you can just be your most authentic self. And I don't think you can be your authentic self if you're not taking care of yourself. So I think, you know, there's no shame in not being actively advocating all the time if you feel like you're not in a safe space to do so. Yeah, so my little piece of advice is take care of yourself. And now I'm looking to learn what I can do moving forward from the rest of the panel.
Yeah, I'd just like to say that I completely agree with you. That was, yeah, it's a really, really important point. And I think that it ties in with what I was going to say, which is that I think I was glad that you put what LGBTQ plus people and allies can do to advocate, because I think this is really, the burden should fall on allies, like I think in all cases. And I think everyone is an ally for a group that they're not a part of. So I think that maybe my advice would be to focus I think you should participate in as much advocacy, like if it's feeding your soul, if it's making you feel happier, if it's like if you feel good about doing it, then of course do as much as you want to do for your own groups. But I think also we should really be thinking about what advocacy we can do for groups that we aren't a part of, and really recognizing intersectionality, and you know, that, you know, something like equal marriage is not the end of advocacy for the LGBTQ community, very far from it. Yeah, so yeah, I think that's it for now.
I think institutions can do a lot of things that make it very clear where they stand on advocating for certain groups. Cover trans health. Just do that. That shows us what this community means to you. And that is something that is still incredibly difficult to find coverage for, especially in state-run systems, because who runs the state systems in the United States? I happen to, again, be lucky that we chose well when we came here. At UNCA, there are so many small practices that have big messages. There are gender-inclusive bathrooms in almost all buildings that are very clearly labeled. And I believe they're also labeled on maps very clearly. Almost everyone has their pronouns in their email signatures. It is not a big deal at all. And when things change in someone's email signatures, as happened with my now husband, when he was transitioning, there's a different partner, by the way. There was a divorce in between. I feel the need to clarify that. But as he was transitioning and changing his pronouns on his email and such, there was response from the folks involved. They wanted to understand what was going on. Folks made the transition. And as things continued. Most recently, I found out when you go in to select your gender identity in our system that holds all the information on all the faculty, there are clear choices that say trans men are men, women, trans women are women. Just looking at that, I immediately called my partner over and I was like, honey, look at this. UNCA is so awesome. It's such a simple gesture, but it says so much in that very simple, inclusive gesture. And I think, in addition to that, of course, our university's done really well about letting the queer people that are on our campus shine, not because they're queer, but because they're amazing. And I think that's really where we all should come to. We all have things that make us different in one way or another. That's not really the point. The point is, what is it that makes us different, but also really good at what we do? And let's have that be the thing that we celebrate. So I never have to worry as a queer woman or a woman of color or what have you. I can see people in our administration that are like me in those various aspects. Not all the intersections, because I'm peculiar, but there are places where I can see myself going within our ranks because other people are already there who have queer identities and who have people of color identities. Is it as even as it likely should be? Probably not, but we'll get there. But just being able to see those simple things, that's a clear commitment beyond let's have another panel during Pride Month, right? That's more continuous, effective support.
I would say you have to insert yourself into these conversations and into these conversations that are going to make policy changes. So if you're a person on campus, like a graduate student or a faculty member, and there are committees that are in charge of doing X, Y, and Z, and these actions are going to influence the lives of people on your campus, and you have an opportunity to be on that committee and voice your opinion on that topic, you got to do it. Or if it's out in the community, you can have a little bit of a wider audience. So for example, for me, I felt like on our campus, we had the bases covered. We had some pretty good queer support groups, faculty, staff. In my psych department, we are extremely diverse in our staff and faculty, lesbian, gay men. We have trans women. We have a lot of people represented. And so I felt like, another voice at the table in our department or at our campus would do good, but what's the impact? So I decided to actually go out into the community and speak on behalf of queer people in the community at a county level and do that rather than just staying insular at the university. So, it took me 18 months to figure out what are the county advisory boards? What are the commissions? How do I get on them? How do I get sponsorship to be on them? What are the tasks that these committees do? And what is the upcoming legislation or policies that are going to be discussed? And can I be in on those conversations? Now, all of this stuff takes time and energy, and you have to make sure that you have the time and energy to do this without sacrificing your goals. If you're a grad student, of course, you want to make sure that you're, keeping your eye on the prize, so to speak. But if you're a faculty member, how do you work this into your tenure and promotion work? How do you get credit for doing these things? Because that's another thing. You don't want to do this as unpaid labor. How do you get this to work in your favor, both at a community level to support your fellow queer residents, but also how do you get to check a box on your promotion and tenure package? So maybe you need to talk to senior faculty members on how this works. Because I'm kind of over the unpaid labor thing. And especially for groups that are underrepresented or disenfranchised, constantly advocating for yourself and your group is very tiring. And so it's better to be able to roll that into your actual work that is being, and that you're being compensated for that. So yeah, it's not an easy thing to do, but there are people doing it, and connect with those individuals and try and figure out a game plan. But look beyond the university too. Don't forget, there's things at the state level, there's things at your county level, at your city level. Look and see if there are openings there. They always need volunteers to do stuff. So choose wisely and choose something that you can make a difference policy-wise. That will influence a lot of people's lives. So I would advocate for that.
Dr. Matthews, I just wanted to add a point there that's so like spot on with what I was thinking in terms of inserting yourself into conversations and just speaking. As a student, I feel a power imbalance when I'm in those conversations. So for students who are listening, it is really scary to be like the only brown queer person with like all the white deans at the university in a meeting. And that's often the case, unfortunately, when I'm in those meetings for various committees and so on, specifically dealing with diversity. In that case, what I found that works for me is sometimes if I have something really important to say, like about compensation, which I might be unfair or some sort of mistreatment, unethical treatment of, somebody who comes from an underserved community, I have sought out allies. Maybe they're not even in that committee. People who would understand the situation, people who know maybe some of the faculty members or the administration that are on this steering committee, for example. And I would tell them what the problem is, and I would tell them what I want to say. And they've kind of helped me figure out what the proper way of addressing these people who are in a position of authority over me, so that it doesn't just backfire in my face. That is really scary, but don't be afraid to ask for help from people who you trust in order to be able to say the right thing and say those important things that Dr. Matthews, you were talking about, because it's true that those are the ways that we need to make change.
And just really quickly, that's a great point. Oftentimes on these committees, an individual is representing a group of individuals, right? So if you're a graduate student and you are on a committee representing graduate students, right, there are probably hundreds of you on campus potentially. One way to protect yourself from this power imbalance is to get the graduate student group. Maybe it's in the University of California system, they're called GSAs, Graduate Student Associations. You can get your campus GSA to write a letter, and you can read that letter at your committee, right? And you can say, I am here representing the 238 graduate students in this college. And here is what we would like to say. And it kind of takes the pressure off of you as an individual. It also reminds people on the committee that it's not just your voice, but you are speaking on behalf of your constituents. So there are these little tricks that you can do if you feel that power imbalance is going to be a liability for you. But yes, absolutely consult with some senior people or consult with people who have done it before, and possibly use the power in numbers type of a thing. To just change the conversation slightly to remind people that it's not just you personally saying these things, but you are representing constituents.
I really appreciate that. I'm going to use that in the future.
Yeah, it's such a good idea. I'd never thought of that before.
I just want to offer a small reframe, Tareq. One of the things you said was saying it the proper way. And that makes it feel like we don't know the proper way to do something is. And we don't want to approach this from a deficit perspective, right? Like there's nothing wrong with all of you. But as with anything, like you work in a neuroscience lab for the first time, there's terminology you need to learn, right? And so here you're just using those same science communication principles in a different setting. And so you're building your audience awareness, you're learning about their specific jargon, you're collecting that information so that you can most effectively communicate with the person you want to communicate with. And I'm someone who both is very passionate about science communication, is trying to study it, and also a big fan of mindset work and the way that we talk to ourselves and how that can make a difference. So just a small reframe to make it more empowering and also fit more seamlessly, right? It fits more seamlessly with just what we do as scientists. as we figure
I
out
really
things and then,
appreciate that.
yeah, figure out ways to talk to people about it.
Thank you so much. That's very good to remind myself of that, Hugh.
All great advice, and I hope that a lot of students today can take that. But is there anything else that anyone would like to add before we move forward with our next question? All right. Well,
I'll add just one quick thing. I know we're all here because we're queer, and that makes us unique and in our togetherness here. But when we're out in the real world, there are other identities to all of you, and the people that you interact with also have other identities. So they may not be queer, but kind of to echo what I think Priya had said earlier, there may be other things about their lives that makes it hard for them. So approaching everyone with the same kind of empathy that you want them to be able to share with you, I've found that to be really powerful personally. I'm a huge also proponent of just being vulnerable, because we are all human. And there are times when we are all struggling, and there's nothing wrong with that. And the more we can normalize that, I think the greater our capacity to network beyond our communities is. There's so much power in the queer community alone. But if we could join hands with all the other communities, strength in numbers, right?
yeah, I definitely agree. And I appreciate the wise words, Dr. Kaur. As for the next question, I believe we touched a little bit upon this in our previous answers, but what are some limitations in neuro research in relation to the LGBTQ plus IA populations, and what can researchers do to make their work more inclusive and accessible to all communities? Anyone can start off.
I don't mind sharing something quickly here. If someone's listening and they like studying in an interdisciplinary way, something that I've really enjoyed reading about and that I'm just learning about myself is the field of neurofeminism. So neurofeminism is a field of neuroscience and feminist philosophy that talks about how neuroscience is interpreted from a feminist lens. So sex and gender-based research is a huge topic in neurofeminism work, and talking about how it should be more inclusive is a big part of that. So sex and gender research in neuroscience definitely still struggles with a lot of ignorance toward the reality of what sex and gender really are. So beyond that, not taking into consideration the lived experiences or privileges or other things that come into play with the various expressions or societal norms that have to do with gender, or the spectrum of chromosomal sex, which is very often looked over. So if people are really interested in learning more about that, I would say look up neurofeminism. It's very cool to learn about.
Yeah, I was actually about to say one of the big issues which is what you're talking about. Part of what you're talking about is neurosexism, which is basically the which a lot of which people are kind of trying to push against, which is amongst other things. I mean, I'm definitely not an expert, but the idea that there's like innate sex-based differences in brains, and then that explains everything about the difference between men and women, which is like just hugely, almost always hugely misogynistic, but then also transphobic because of this idea of like a female brain and a male brain in the first place. And so I know that a lot of work is being done to push against this, and it does intersect a little bit with something that I'm really interested in, which is open science and reproducibility. And I know that a lot of neurosexism seems to have come out of people just always analyzing sex differences in neuroscience research with no theoretical basis for this, just because it's an easy way to, if, for example, you didn't find what you wanted to find in your study, but then you quickly analyze the sex differences and you find something, then you have something that you can write up, basically. And so, yeah, it does definitely intersect with kind of issues of incentives in academia more generally, and kind of why might someone want to do that. It might not necessarily be because they have like any opinion on this actually, but they're just using it as an easy way out to get a publication, because publications are the currency in academia. So yeah, I think you can't really disentangle these things, but it's really important to push against it.
To piggyback off of that and come back to something I care about, more effective science communication around the neuroscience principles that relate to these aspects, I think would be really meaningful, because neuroscience is lousy with misinformation. Just because it is so easy, people can connect to what the brain is doing and what studies say to the human person. And if it's not handled correctly, it's just disastrous. So for all of us, we have the opportunity to engage in science communication in one way or another. If you can find ways to do that in a public-facing way, and something as simple as just Twitter is actually meaningful, because you might get retweeted by something and see someone else sees it, and someone may think differently about how brains work. And that's really what we all want to be able to get to.
If anyone is interested, I feel kind of shameful for doing this, but I do want people to start to have this conversation. I did write a little blog post about what I've learned so far about neurofeminism recently on my Twitter, which is just Tareq Neuro. I'd love to hear your thoughts.
You can also type it in the chat so people can follow as well, but would like to emphasize how media can affect the communicating certain stigmas and certain issues, and making sure that we do get the word out there to through various different mediums, whether that be Twitter or Instagram. So just would like to echo on what the panelists have stated. So Dr. Matthews, do you have anything else to add?
Sure. Historically, my research is on how men and women are portrayed in mainstream media, print media. Recently, I've changed to how people are displayed in print media, and going away from gendered terms. Undergraduates, so I'm at a teaching institution, which I like to call a teaching one institution, because that is our main focus. And we are primarily undergraduate, and the undergraduate students that come into my lab are interested. I'm mostly interested in how LGBT people are portrayed in popular print media, but I have students who come in that want to look at how women of color are portrayed in magazines, marketed toward women of color. So how are women portrayed on the covers of, Essence magazine and so forth? And I think for researchers, it's a reminder to be flexible in the areas in which you are comfortable studying with your students. So I am by no means an expert in how women of color are portrayed in media or Black media, but that doesn't mean that I can't support a student to do work in that area and learn and teach the methods that I use for my area to help them answer the questions that they have. So this question of what can researchers do to make their work and research more inclusive and accessible to all communities, support those undergraduate students in their goals, even if it's not, the Venn diagram of their interests and your interests. They don't have to be like this. There can be just a little bit of overlap methodologically, or the way that you handle the data, like statistically, or just a theoretical perspective. Like I think Dr. Cal said, be vulnerable and be okay with that ambiguity and say, you know what, I don't really know the history of this, but let's work on it together. Why don't you do some literature searches on these topics, and let's meet and talk about this and come up with a plan for you to follow this research question. So it's being vulnerable as a faculty member, as a mentor, and trying to support your students in answering questions that they're interested in, even if they don't have huge overlap with your area. Now, I'll say it might be harder at an R1 institution where you kind of maybe have to be more focused in your research area and not, go outside the lines too much, but at a teaching institution, it's almost required if you're going to be successful in your lab and have your students be successful as well.
Thank you for that, Dr. Matthews. And I would like to echo a bit on that as well, because I am interested in conducting research in human sexuality. And I have found a few researchers and professors and faculty that are interested in what I would like to conduct, but also not necessarily in the exact area, and would like to sort of echo that in that professors should be open to conducting research with students who are interested in not necessarily the exact same thing, but something similar, whether that be through methods and so forth. So thank you for sharing that. I do have the next question, and this is a bit of a more fun question that we have, which is, what is the most interesting research paper or research that you have conducted or / read in the field of sexuality / gender neuroscience?
I just recently read a paper. I'm so nerdy, I'm realizing, but my friends and I have our own journal club. Can you believe it? We recently read a paper about pronouns and using pronouns and how effective that is in changing a person's perspective on what kind of gender spectrum is being identified. So there's a lot of work coming out of Scandinavia about this research. They're way ahead of North America, for example, in terms of having non-binary pronouns become mainstream. It's even become a topic of public discourse with public debates happening around them. So that's not really happening here. There's a lot of work obviously being done, a lot of advocacy and activism, but it's not on the same level public facing. So this research paper, if I'm remembering correctly, it was several weeks ago now, but they found that just writing they, for example, instead of he / she, so getting rid of the binary and writing they, the people who were reading they still thought in a male-centric fashion. They would still think of men first. So one thing that they recommended was to use more than one pronoun beyond he and she. So for example, using she / they, or she / ze, ze, for example. So yeah, that was a really interesting paper I read. Yeah.
I found that really cool. I will definitely try to look into that for sure and try to read upon the literature, but does any other panelists want to hop on as well?
Sure. One of my favorite papers is actually from the early 1980s. It was a JPSP, Journal of Personality and Social Psychology paper. The lead author was Dane Archer. They were at UC Santa Cruz. And it's called Facism, 5 Studies of Sex Differences in Facial Prominence. It's a really cool paper. They were examining how men and women were portrayed in magazines and in newspapers. And what they found is that men are often portrayed with more headshot type photographs, and women are more often portrayed with more body shot or long shots, where you'll have a photo that's like from the knee up or the waist up. And they found this in a whole variety of different media outlets. And some of the research that they found that these types of patterns even went back to the 15th century. So it was a really cool paper, 1983 by Dane Archer and colleagues.
I confess I don't read about these things. The stuff I'm reading most right now is related to science literacy and science communication, because I'm working on a grant and need to finish the project description.
Yeah, I was going to say as well, like, I find that I don't read much about sexuality and gender in scientific papers. And I think partially it's just because I don't work on those topics, but I also find that, like, I've almost been put off reading anything about this, because of the only time it kind of comes across my Twitter feed, for example, is usually when like some horribly sexist, homophobic, transphobic paper has like got through peer review, and then I'm reading about like a Twitter storm about how terrible it is and just feeling despair. So I think if anything, I probably have a too harshly negative view of these fields because of some of the people who work within them, when I'm sure when there's loads of great people doing good work. But yeah, I have found myself avoiding these topics.
Well, thank you, Dr. Matthews and Mr. Yousef, for sharing papers that you were interested in, but I also appreciate Dr. Kaur and Dr. Silverstein for your transparency in this question as well. And oh yeah, I see that they are sharing the research that they found. So if anyone's interested in looking into more detail and reading them in detail, feel free to go ahead and check those out. Thank you guys. Okay, well, now I believe the next question is, what advice do you have, or do you all have, for having conversations with individuals that do not take pronouns seriously?
I guess I would say, if someone's very upfront about that in kind of an aggressive way, the first thing that I'm thinking of is I'm not doing this for you. I'm doing this to create a safe space for the person who's going to see it and understand that I'm someone who maybe understands the topic of gender and who you might feel comfortable expressing yourself to, if that's the case. Of course, publicly, it would be nice to get everybody on board with being, okay with sharing pronouns and starting conversations off that way. So it should be for everybody, of course. But if somebody's not, kind of like picking up what you're putting down, or somebody's not being kind about something that you're doing, which, is a nice thing, that's not your fault. And maybe rest easy in knowing that hopefully that's going to catch somebody's eye in the future, and that's going to really help them, even if it didn't help this one person.
I think in the context of if you are asking to have certain pronouns be used and it's your supervisor who will not use those pronouns and will continue to misgender you, in that situation, getting people with more power than you involved is probably the best bet. And what that will look like will depend on if you're in undergrad or in grad school and what the structures of power are. But as a grad student, I think you can just go to HR. And as an undergrad, I would go to the department chair or a trusted faculty member who can probably tell you where to go next. Because that's not a microaggression. That's someone ignoring your stated, like, this is my pronoun. They are ignoring it. If it is a friend or acquaintance, say goodbye and forget them. I mean, honest, I know it's a very flippant response, and I know with family it can be harder, but at some point you are having to make that decision of how much of your own mental energy and like strength do you want to give up for this human. And if they have made it clear where they stand, believe them and walk away. If they want to come back around, great. If not, it's not your job to educate everybody at the expense of yourself.
Yeah, that's great. I definitely didn't understand the question then, because that's so true. And I would say, just to add on to that, if you are a student, like totally, yeah, go to HR. I just want to advocate for doing that, because I often see so many new graduate students coming into our program and being afraid to bring up things that have hurt them, or relationships with supervisors that have been harmful. There are people whose specific jobs are to be coordinators of your graduate program, who are not going to be grading you or giving you a mark or being on your committee, for example, who they are safe to go to and to approach. And then otherwise, if the faculty way is not really helpful, I think, just like Dr. Matthews said, approach the society, the student society who's there, and they hopefully will be able to advocate for you in that very serious scenario.
I'll say, it sounds scary to have to like go up and like stand up for yourself, but the alternative is a lot of damage to your brain because of a lot of like prolonged stress, especially if this is someone in your lab who you have to function with for 4 to 5 years, if not more. And I think that's the thing that we as people that humans are not very good at, right? The delayed response, the immediate response works well with our dopamine system. Delayed stuff we don't really pay attention to, but there is a delayed yes that you're saying to. By saying no to standing up for yourself in that situation as safe, you're saying yes to a lot of damage to your brain because cortisol sucks. So don't do that to yourself. I may have lived a lot of these things.
I think also, as well as echoing everything that you both have said, I think it's important to acknowledge like the emotional baggage and labor that comes with having to like go through the bureaucratic process of reporting things and taking it higher, and that will vary depending on the institution in how much you kind of have to follow up and push and like how much support you're getting through that. So I think sometimes it can be good to have someone advocate for you, someone kind of go through that, the kind of the burden of the emailing, the following up, the like more of the kind of admin related aspects to it, so that they can kind of take on some of that burden for you. Yeah, because I can completely understand how, even though whenever I mean, I haven't been in this situation, but my wife has been in similar situations with microaggressions of racism at her institution, and she's been told this is the way that you kind of report it and this is all the processes that you go through, and has found that sometimes that can be like harder than just leaving it, which is horrible, and I'm not advocating leaving it either because of that, but I'm just saying it can be helpful to have support through that process.
I'll just echo what everyone else has said, and I don't like to use war metaphors, but you really have to learn how to pick your battles in a lot of these situations. As a graduate student, as a faculty member, I mean, even if you don't go into academia, whatever occupation you choose to pursue, battles will always be there to be fought. And you have to first think about yourself and your own health and mental health, and which ones are you going to engage with? And that's just a question you have to ask yourself, because you can't fight them all. And so the question of, if someone is refusing to respect pronouns, my first question in my head is somewhat like Dr. Kaur's question: who is this person? Is there hierarchical power at play? Is this person just a random person that's either at your level, like in the hierarchy, or not? And is it worth fighting with them? If this person is your supervisor or has some sort of power over you or your degree, then yes, absolutely, that is a fight that you probably will have to pursue at some point, and you'll want to get as much support as possible. But you got to pick those battles, because there are many to be fought, and you can't fight them all effectively. So yeah, you got to kind of choose your battles wisely.
Yeah, and I'll say I absolutely agree with you. And when I say don't take on the stress of it for the following years, there's other ways to put the stress down, right? So if there's a random one sentence that someone said to me that stung, I can put that down with a few hours of hanging out with my trusted community. And a few weeks after that, I'll forget that it ever happened. But if I don't take that time to deconstruct it, to sit with it, to understand, again, I'm a big fan of mindset work. It helps you get through some stuff. But if you don't figure that out, it's just every time something else happens, that thing will continue to eat at you and echo back. And it's just not worth it. That person does not deserve that much power over you. No. And so finding the way that it will help you to put that down, it builds greater resilience for us to be able to go further and shine our full queer brightness, as Tareq had said earlier.
Because if you have your pronouns visible, there are probably a handful of people. For every one person who's not down with it, there's a handful of others who are silently praising you for doing it. So remember that.
I can also say from like the other side, like what feels supportive to someone who like has recently changed their pronouns. I think when you do have the option, like for something like the pronouns that either you used before or that was assumed when someone looks at you / they or something else, like then it really is so meaningful when people use the one that isn't the one that they used to use / isn't the one that would be assumed by looking at you. Because even if you don't necessarily, in my case, like I don't only accept they them pronouns, but it shows me that you care if you use them some of the time or all of the time, because I know that, because something that I've been thinking about is it's interesting, like if someone uses she, I will never know whether they're using she because they are trying to be respectful of me or whether they're using she because they assume she. Whereas if they use they, I know that either they don't know what pronouns I like, but they are like taking the opportunity to use gender neutral ones because they're not assuming, which is lovely anyway, or they actively have taken into consideration what I care about. So I think it's, yeah, and that's not going to be the case for everyone. I know that some people, like if they have 2 types of pronouns, they actively want people to use both all the time. Some people just don't care at all and just, it means use any of them. So it will vary from person to person, but yeah, that is a good way. I know that wasn't the question, it's like a backwards version of the question, but that's a good way to show your support to people and their pronouns.
Yeah, and I appreciate you guys for sharing, because I actually have had to go through a little bit of pronoun disputes with a few friends of mine who did not necessarily believe in the use, or just always would question why I would say my pronouns every time I introduce myself and all of that stuff. And I have had to pick my battles, like I said, or like Dr. Matthews have said, and just have lost a few friendships here and there because of that, because of the pronoun thing, and just appreciate you guys for sharing that. And I hope that a lot of other people can relate to it as well. So thank you. All right, well, another fun question. Are there any novels or other resources that each of you recommend from both people inside and outside of the queer community that can help provide an understanding of being queer in STEM?
This is the problem with coming into STEM already feeling very secure as a queer person. I have no idea. I have not had to seek that out. And so I personally, I don't know. I'm so sorry.
This is not in STEM, but it's in academia. I read Hunger by Roxane Gay, Dr. Roxane Gay, maybe 1.5, 2 years ago now during my PhD. And in it, Dr. Gay talks not only about, her literary work and her background. It's a really great book. It dives deep into, the problems she's had with eating disorders and various other issues, being queer. But then also she talks about being queer as a professor and being a queer person of color in a small town, teaching mostly white students, etc. So that was a really great book in all kinds of ways.
The same as Angel, I don't, unfortunately don't have any recommendations of books to do with being queer in STEM specifically, but I would definitely recommend Queer: A Graphic History by Megan John Barker to anyone who hasn't read it. It's just the most amazing kind of coffee table book to have access to, and yeah, I couldn't recommend it more highly.
I don't have a STEM specific author, but I would say anything by David Sedaris to lighten the heart a little bit. I mean, not all his stories are queer related, but some of them are, and they are gems. So it's not STEM specific, but if he writes a lot of short stories, and if you need a little pick me up, oftentimes that will do it.
Also, if it's hard for you to read sarcasm, listen to him instead. I cannot read David Sedaris, but I love listening to it because his like very dry voice captures his sarcasm so much more effectively that I bought a copy and I was like, why do I have this? I should just listen to him.
Yeah, his audiobooks are very good.
Also, if you could put it in the chat as well for anyone else who's interested, that'd be great. But I will definitely take into consideration all of your guys' recommendations. They all sound quite interesting. And the last question before we open it up to the general community for Q&A is, what advocacy have you all found to be ineffective? For allies, how can we amplify meaningful messages without unintentionally being performative?
I mean, to me, performative is you say all the right things, but you don't actually do anything. So just do things. No, that's a simplistic response, but yeah, that's what I've got.
I think in general, doing stuff only during Pride Month is like the most performative thing ever. And I think obviously I'm sure we all hold the same resentment towards companies and businesses for trying to profit off Pride Month, and it gets more and more ridiculous every single year. It's almost like a parody. So yeah, but in terms of like institutions like universities, etc., I think doing any kind of advocacy, I mean, even the exact same advocacy happening before or after Pride Month would be more meaningful to me than it happening during. Like, because it just shows that you're not like just doing it because it's a gimmicky thing. And like, it can be so much more depressing to be somewhere where during Pride Month the message is we love you, we support you, blah blah blah, and then crickets, like just nothing ever again. That can be worse than having nothing, in my opinion.
I think I'll go back to my hatred of unpaid labor. And if your institution is going to do anything for any group, really, put your money where your mouth is and pay speakers from those groups to come and talk to your employees or your students. Pay them for their work. Don't just expect individuals to come and go on a speaking circuit just because it's Pride Month and to do it for free. People have jobs, people have mouths to feed, people have bills to pay. So put your money where your mouth is and actually fund these initiatives. Don't just say you're going to do a great thing and then not put any resources behind it. There needs to be resources for those things. And if you're a faculty member, be very mindful of administration wanting you to do certain things and being the token person on a committee speaking for the group, and not actually getting some sort of compensation for doing that, because that is emotional labor as well. And you need to be compensated for that.
I guess if I was to add anything, those were all amazing answers. If I was to add anything at all, I would say, try to advocate that activist work be done with people who are the most disenfranchised at the forefront, of whatever outcome that is going to be. So if there's a specific community within the queer community that at your university or whatever is being underlooked, whether it's queer refugees, for example, or queer international students, then try to make sure that they're included. Try to advocate that they be included in those conversations and they be included in, the changes that are being made.
Thank you guys. And hopping off of what Dr. Silverstein said, I do appreciate you saying how we should advocate beyond Pride Month, because I just find it so interesting how all these companies are just like pride, pride, pride during Pride Month. And then right after, like you said, crickets, you know, so definitely will push people and students to advocate for LGBTQ plus communities outside of Pride Month, and it continue doing it after June, which is actually quite close. We're closing in on Pride Month so soon. So yeah, thank you guys. And we're just gonna open it now, the questions up to anyone who has any questions, any students in the Zoom right now or on the YouTube Live that have any questions. Can type it in the chat below. Sasha did ask do you guys have any advice for high school students interested in gender psychology, specifically the struggle in finding accessible resources to learn more about the field?
I've learned a lot just from YouTube. I would say try to hit up YouTube and see what lectures have been posted publicly. I've learned a lot even in my own field in neuroscience, getting to see lectures from people who, you know, are like huge, like big celebrities in the field. So try to see what stuff already exists online in the form of a lecture if you're looking for that multimedia experience beyond just reading papers.
There are some professional organizations like the American Psychological Association that have different divisions. Those are all defined on their website, and there are a lot of different divisions. They're all numbered. Maybe if students could peruse that to see what the goals of the different divisions are, who the key players are. Each one has a president, each one has a vice president, all these different things. Most of them are faculty, not all of them are. But they are kind of the workhorse of the entire organization. And those individuals are very knowledgeable about that specific domain. And so APA is structured that way. I'm not sure if other ones are, but you could look into that and get kind of a more, not just psychology in general or neuroscience in general, but some of these organizations are divided into different divisions that take a little bit more of a focused approach to the science. So that might be useful.
Any other questions from the general audience? All right. Well, I think that would conclude the LGBTQ roundtable discussion. Do the panelists have any final comments or final words that you would like to say to our students before we end?
I just want to say thank you to everybody, obviously everybody who listened, but all the panelists and Ryan. Thanks for giving me the opportunity to be in a place where I can talk to other queer people who I can look up to in academia. So thank you so much for sharing your stories to the other panelists. I've been eagerly Googling all of you up until today.
I would say if your institution doesn't have a formal network of support for queer people, start one. Get your friends together, do it yourself. I don't know. Advocate at the university level for funding from your Office of Student Affairs to start a club, anything. people will come out of the woodwork. You would be surprised to help and to participate. So if there is not a presence in your campus or in your organization, create that presence yourself.
Yeah, I'd just like to echo Tareq's thanks to everyone for being on the panel, and also to Ryan and to all of you for attending. Yeah, I really enjoyed it. And also, in the spirit of cold calling people, I definitely am always open to Twitter DMs or emails from any queer or otherwise students who want to reach out about academia, any questions about academia, or just to reach out and have that community. So I'll put my Twitter in the chat as well.
Yeah, I'll echo again, but do it much more truncated. This was fun. Thank you, everyone. And I'll just come back to a point that we all have made earlier, just as a reminder to pace yourself, take stock of your energy and your abilities, and do what you can to allow yourself to flourish the best possible way. And sometimes that means turning off your phone and walking away from it and protecting your time that way. And even though society will tell you that that's wrong, especially in grad school, it's not wrong. Grad school is wrong.
And I'll say, Dr. Kaur, you gave a really great pitch for UNCA. I'm really excited to look up more about the university.
We are pretty great. We have some fantastic faculty and staff here, and our students are amazing. And that's really where all of my energy for this comes from, because I really enjoy teaching. And to me, it is a very collaborative process. My students are partners with me in the classroom, because there's a lot of things I know, but there's a lot of things I don't know. And I enjoy, and my ADHD brain will be happier if I am excited and interested in what's happening in the classroom, and if students have the opportunity to surprise me. And we happen to be at a place where we've got some really incredible students coming in, and they are open to learning with us. And that is just everything to me for the work side. I'm also a whole person outside of work, which is very important to me, to be a whole person outside of work. So, but it's a place where that allowed for that to happen too. So I feel lucky.
Well, thank you so much, panelists, for everything that you guys have told us, the advice, valuable advice, and just you're sharing your experience and continuing the conversation and changing the conversation, and especially during Pride Month. Happy Pride, guys. And yeah, thank you as well to Simply Neuroscience for having me as the moderator. I appreciated moderating such a amazing panelists, and we'll definitely try to contact you guys afterwards. If you guys would like to share your contact information, whether that be your email or social media, please feel free to jot that down in the chat. But again, thank you guys.
Thank you.
Thank you.
Thanks so much.
Thanks, everyone.
Thank you so much, everyone. And thank you, Ryan, for doing an incredible job hosting and kind of spearheading the conversation. This was an incredibly important conversation. And next for Neurocon, we'll be moving on to our brain breaks. So if you're here and would like to stay on for that, we'll start that in a couple of minutes. Okay, so I think we can go ahead and get started. Are there any SN folks here who would like to explain the activity and what's planned for the brain break? Some people might be running a little bit late. Okay, let me check the chat real quick. Okay, let me unmute some people.
Oh, okay. Hi guys. So I just sent Sasha, do you think you could check the Zoom chat really quick? Because I sent you the slideshow of what you could
Yeah, I see it.
screen share.
Yes, let me do that. Okay, can y'all see the screen? Okay. Okay, awesome. Siddhi, are you still able to unmute yourself? I might have to make you co-host. Okay, sorry about the technical difficulties, everyone. I'm going to make Siddhi co-host real quick.
Okay, I think it's working now. Can you hear me?
Yes, we can. Unfortunately, since I'm not the host, I can't make you co-host. So once Arvind hops back on, I'll ask him to do that. But for now, just keep your mic unmuted because I'll have to like manually unmute you as well.
Oh, okay. Okay, so hi everyone. So for the brain break right now, we're just gonna be doing a quick networking session. So Sasha, do you think you could show the last slide, I believe, because that has the questions on it? Okay, so yeah, so these are the 5 questions. We're gonna be putting you guys into like breakout rooms, and then you guys can just go over those questions, and then if we have more time, then we'll rotate the rooms. So do you think you'd be able to make the breakout rooms? Because I don't have the permission.
Yes, I think I can make the breakout rooms. Let me go ahead and if everyone wants to take a screenshot of these questions, since when we get into the breakout rooms, I'm not sure if you'll be able to see them.
I can also send them in the chat, so it's okay.
Oh, perfect. Okay, thank you so much. I'm trying to find the breakout room setting, but I believe it might only be available for the host as well, which, yeah, it seems like it's only for the host. It's a host permission. I don't know if Arvind is here. I don't think he is. I think he had to step away, but we can totally just do it in a larger setting. We only have around 18 people here, so it's totally fine if you want to do like here, let me turn on my camera. If you want to do it in a bigger group setting, if that's okay, Siddhi.
Yeah, for sure.
Sasha, this is Shreya. I managed to log onto the SN account and I made myself host.
Oh, awesome.
So what do you need me to do?
Yeah, would you mind starting breakout rooms? Thank you so much.
Sure, let me try to find where that is because I suck at Zoom. Okay, here we go.
Can you also make me co-host if possible?
Yes, I'll do
Okay,
that
thank
first.
you. Thank you, Sharia.
And you should be co-host now.
Okay, perfect. Thank you.
Let's see, breakout rooms. How many do we want?
We can do 3 breakout rooms, 6 in each.
Okay, and you should be assigned to the rooms.
Thank you.
Okay, so for everyone that attended our brain break session, thank you for attending. And now that it's about 6 p.m. EST, we have our next session for Neurocon, which is the penultimate session, and it's the Neurocon Pub Night organized by Grant and from NeuroCollege and hosted by Grant and Andrew from NeuroCollege. And so yeah, take it away, and you have the floor. Do you guys have screen share permissions?
I think Shreya right now is working on making them co-hosts, and they should be able to screen share and unmute.
Okay. In the meantime,
Got it. Oh, sorry. Okay, I just got co-host permission, so
Okay, the NeuroCollege team should now be co-host, and Ervin, I will make you sole host.
Okay, yeah. So just to introduce all the attendees to NeuroCollege, I'll just give you guys a bit of a background. So NeuroCollege unites Neurotech students across different US colleges to discuss the latest advances in neuroelectrical engineering and to share academic and industrial resources with each other. Their speaker events, roundtables, and publications act as myriad gateways to help Neurotech students gain knowledge and experience in order to succeed in the fast-growing neurotech space, especially when similar resources are sometimes inefficient at even the most well-funded universities. As a student organization, their main purpose is to create a neurotech community that helps promote interest and awareness while supporting their career development in the neurotech space. And you can visit neurocollege.org if you want more info. So again, welcome to Andrew and Grant, and take it away.
Awesome. Thank you so much. So I'm just going to share my screen and we can get right into it. All right. So yeah, first of all, thank you so much for being here. I'll introduce myself really quick. I'm Grant. I'm a rising junior at Stanford studying symbolic systems, and I'm really interested in BCI. And my other lovely co-host, Andrew, is here. If you want to introduce yourself as well.
Yeah, for sure. Sorry about the little bit of background noise or technicality and the disturbance. My name is Andrew. I'm going to be a junior at Columbia in the fall, studying neuroscience and behavior as well. I'm sorry, is the background noise okay? No. Okay. I'll try to fix that in a second. I'll be a junior studying neuroscience at Columbia as well. Yeah, really excited to talk with all of you guys about BCI and the credentials and things like that.
Awesome. So yeah, I can get into the presentation now. So the way this kind of works is just if for those of you who aren't familiar with, like, I guess the concept of pub night is just a, casual discussion. Think about it like just like a group of friends kind of talking about something, and you just share your thoughts, share, what you're thinking about, and yeah, just have a good conversation. And the way these are kind of structured is first we'll do a quick presentation about the topic and just kind of like give some information just so you can learn a bit about it. And then we'll hop into small breakout rooms. And for the remainder of the time, you'll just discuss whatever came to mind, whatever you are curious about, anything goes. So yeah, pretty chill, relaxing environment. So yeah, let's get started. So today we're going to be talking about BCIs and neuroscience. So I know that most of you are probably your background is in neuroscience. So this BCI stuff is newer. And just to introduce that, so what is a BCI? A BCI is a brain-computer interface, and it's any technology that sends and receives signals between a brain and an external device. So, some examples of these are mind-controlled prosthetics, any type of, using your mind to control something, or even using a computer to understand the mind based off of brain activity. And next, so what is neuroscience? This is pretty straightforward. Pretty much it's just studying how the brain works and investigating, the science behind it rather than applications of that science. And the way that these two concepts kind of mesh together is when BCIs can be used to understand how the brain works, because there's so much that we don't know about the brain. Like consciousness, for example, is a big one, but many different things that, you know, we just need more data for. We need more imaging methods and stuff like that. So that's why you know, BCIs can be used for that. And I want to get into a quick company spotlight. So kind of the application of BCIs towards neuroscience, I think, is best captured by Kernel. And I'll just introduce Kernel. They're a neurotech company that is, you know, really big right now. They just released like a couple of their products that they've been working on for like 10 years or something like that. And so here's the introduction. The main idea behind Kernel is that just like how mainframes kind of were, mainframes are to personal computers as, you know, current brain imaging techniques are to Kernel. So they want to take something that's super expensive, super inaccessible, and bring it into the homes of you know, everyone, and making it a lot cheaper and more accessible. So that is kind of like what the goal is, kind of to remove those boundaries that make it so hard to study the brain and get data about the brain. And a couple of their products. So the first one is Kernel Flux, and it uses OPMEG. So I'm not like an expert on this at all, but it's optically pumped magnetoencephalography. So they use these magnetometers to measure, you know, changes in magnetic fields. And as you can see in that picture, it's a little bit clunky, but you know, you could wear it in the home and office and stuff like that if you wanted to, you know, record what's going on in my brain right now. That's something that you have access to, and you can do it in real time. Versus, you know, some stuff like fMRI, where it's a whole lengthy process and very expensive. And the next one is Kernel Flow. So here's and Kernel Flow is it's TDF-near, so time domain functional near infrared spectroscopy, something like that. But pretty much it's using near infrared light to estimate blood flow in the brain. And the kind of like results they've been able to their advertising are pretty mind-blowing. So they collect 1 billion photons per per detector, thousands more than the state-of-the-art F-near systems right now. And it also has full head coverage. So and it can obtain absolute measurements of blood oxygenation. So that was like a lot of technical lingo, but it's just pretty much a very effective brain imaging technology. And I'll show this video. Oh, I don't know if there's sound. Actually, we probably don't need sound, but I'll show you this video that kind of goes over what Kernel Flow would look like if you owned it and, you know, had it in your daily life, and you can just kind of see what that looks like. So you check this out. Actually, you know what? Let me share it with sound really quick. Okay, where did that go? Okay, is that? Can you guys see that on the screen? Am I back? Am I on the slides right now?
Yeah.
Oh, okay, cool. Awesome. So, okay, here's like their little promo video for Kernel Flow. Oh, there's no. Yeah, so that was the video, and I know there was no sound, but you kind of see throughout the video like some of the ways that kernel flow could be used just throughout your day-to-day life, and that's kind of more on the application side of things, where it was like the something that I noticed was like the detecting zombie mode. I think we can all like relate to that when we're like scrolling through Instagram and you're just like in this kind of zoned out state. Like that's something that you could have insights into if you wanted to measure your brain all the time. So that's kind of the newest technologies right now in BCI are these kind of portable lower cost systems that are able to record high quality data from the brain. And some of the key points, so there's the they have a goal of like having these devices being used by regular consumers throughout our day-to-day lives, like kind of having that like one of those like in your home, but they also offer neuroscience as a service. So that's them doing, and they call it like full stack neuroscience. So they will design and execute experiments for clients using the hardware that I just showed. And because it's portable and it can be used in a lot of different environments, it would allow neuroscience to kind of move out of the restrictions of a lab setting. Like, I don't know if any of you have done like fMRI studies, but there's so many restrictions to doing that because you have to be, in this constrained environment. But with something like kernel flux, kernel flow, we could see neuroscience moving into like regular situations. And I think that a lot could provide a lot of insights into understanding the human brain because we have, the accessible methodologies for doing so. And on top of that, Kernel also talks about how these technologies will help generate makes neuroscience more accessible to more people, since it's a lot cheaper than current brain imaging techniques. So yeah, that's kind of like BCIs and neuroscience. We're starting to see that intersection right now, with the perfect example being kernel. But this is something that is going to be growing a lot more over the next upcoming years. And yeah, so that's a presentation, and hopefully you all have a lot of thoughts and ideas in your minds right now. And what we'll do is go into breakout rooms, and let's see, we have okay, let me see.
Yeah, just jump in there real quick, 1 second. The yeah, just want to jump in and add on what you were saying about the not just like personalization of it, but also like the accessibility, and the fact that like you were saying, having something like that is marketable and on a scale which people can use outside the lab also contributes to making neuroscience more accessible for groups that historically haven't had access to either neuroscience like data and not neuroscience research, or for groups where like neuroscience research wasn't necessarily done on certain demographics and certain like historical groups. So having, yeah, almost like democratizing democratizing the field that like should in theory be the most democratic, that like every single person, right, is affected by neuroscience, for like democratizing that field to make it more accessible and to make personalized data, personalized brain data, oxygen blood flow data, and personalized insights like you're talking about, open to people who aren't in major research centers or major research hubs. Is huge. Like that not only has much potential for like the industry, but has so much potential for like applicable practical purposes of neuroscience on society and on like the lives of real people on a larger scale.
Yeah, and another thing that I just thought of in relation to that is the concept of weird in psychology, which is an acronym for like Western, educated, industrialized, rich, and democratic. That's where a lot of brain data is collected from, and there's so much neurodiversity that exists that isn't captured because we're so limited in how we can record data from the brain and image from the brain. So it's super interesting to see that hopefully as this technology gets more accessible and becomes super easy to use and access, we'll be able to understand the brain in a lot more diverse people and settings. So awesome.
For mental health, that'd be huge too, just like you're saying. For like, we all know about, like, I don't know, a lot of us probably know about like the Ocean model for personality, and like that being like one of the only empirically supported personality types or personality tests. But pretty much like even among different ethnicity and different like geographical regions, there's like slight differences in tendencies. So just think about how much more so, for like personalized mental health treatment, personalized neurocognitive disease treatment, the potential for that.
Yeah, 100%. So Arvind, sorry, quick question. I don't know how to do the breakout rooms on my end. Do you think we could split, because we have 16 people right now, do you think we could split into 4 breakout rooms with 4 people each and like kind of do that just so we can have like those smaller conversations?
Yeah, I'll make those.
Thank you so much. And then for everyone else, so once you get into your breakout rooms, it's just going to be 3 other people. So it'll be a super cool conversational setting. And just like feel free to introduce yourself, get to know everyone else, and just share some of your ideas about how BCIs can be applied for neuroscience, and any thoughts that you have. Awesome. Okay, so it looks like breakout rooms are closing in 45 seconds. So we're waiting on a few people, but so yeah, I'll just wait for them and then we'll just like wrap it up. Yeah. Okay. Awesome. Well, it looks like we have everyone back now. So yeah, that was it for the pub night. Just want to say thank you all for coming. We really appreciate you taking the time. Hopefully had some good conversations and, met some new people, learned some new stuff. So yeah, thank you so much. And Andrew, do you want to say anything?
Yeah, I just want to say thank you to all of you again as well. My breakout room is amazing. I learned so much from people who have a lot more experience with neurology, with like practical neurology, so much more than I could have imagined. Like they give me insight into applications that, like you said, I hadn't even thought of, but like which seems so intuitive. Things that our conversations range from pediatrics and like regular measuring of like milestones in development, in early development, both for children as well as even adults, to like linguistics and like how learning different kinds of languages, with like Latin versus non-Latin versus like Eastern Asian languages, how that like rewires your own brain and affects your own brain, and like vice versa. So just want to encourage all of you. A lot of you guys have extremely insightful and unique and original thoughts that a lot of people, and three more in this field. Really grateful for what I've learned from you guys. And again, thanks for inspiring me with both the current benefits as well as the potentials.
Yeah, thank you so much to Andrew and Grant for coming. Really nice having you, and hope to see you in future Simply Neuroscience events as well. So thanks. Bye.
Thank you.
Thank you, Evan.
Okay, so now for our last session of Simply Neurocon 2021, I'll hand it over to Priya, who will be running the brain resource creation workshops.
Okay, great. So today we have our, as our final presenters for Simply Neurocon, we have our some team members from the Brain Resource Creation team. Let me just pull up the little bio. Okay. The Brain Resource Creation Workshop at Simply Neurocon will cover synaptic plasticity and its role in sensation, and a couple of case studies that depict the importance of plasticity in real life. There will also be a fun Kahoot game that will review the key information from the presentation. And now, without further ado, I will hand it over to our BRC team members.
Okay. Hi, everyone. Welcome to our synaptic plasticity slides. We're so happy to have you here. My name is Kristen. I'm part of the Brain Resource Creation team. and I'm in New York City.
Hi, my name is Samanvi, and I am from Northern Virginia.
And Andrea, are you there?
Okay, hi, I can speak now. Okay. All right, so yeah, hey everyone, welcome to the Brain Resource Creation Workshop as part of the Simply Neurocon event. And before we formally begin with the presentation, we are going to introduce the presenters for today. So I'm Andrea and I'm from Hong Kong. So yeah, I'm sure you guys have introduced yourselves already because I couldn't unmute, I'm sorry. So before we begin, we would like to start with a quick icebreaker question. And the question is, what is your favorite part of the brain? And feel free to put your response in the chat. Yeah, I see some pretty cool responses coming. All right, so I think that's around everyone, so we can start with the presentation.
Okay, sounds good. Okay, so before we jump into synaptic plasticity, we'll first give an overview of the brain, and that is the information that is relevant and will lead up to synaptic plasticity. So my favorite organ system by far is the nervous system because it's so sophisticated and complex, and it allows us to do everything from forming memories to learning to dreaming to walking and to playing games and much more. So there are two parts of the nervous system, and they constitute the organ system's functions. So the first part is the peripheral nervous system, and it's responsible for picking up sensory information from the external world and sending that information to the central nervous system and carrying out a voluntary or involuntary motor response ordered by the central nervous system. The other part is the central nervous system, and it's responsible for processing and integrating sensory information, and then generating a motor output in response to that sensory input. So as you can see by the diagram on the right, the peripheral nervous system is in blue, and it includes the ganglia and nerves. So the nerves of the peripheral nervous system, or the PNS, is connected to the skin and muscles throughout the body, especially in the limbs, which allows us to carry out, motor output. As you can see, the central nervous system is in red, and that includes the brain and the spinal cord. So neurons in the peripheral nervous system are connected to the brain and the spinal cord of the central nervous system. So there are three basic stages of information processing in the nervous system, and that includes sensory input, which occurs in the peripheral nervous system, integration, which occurs in the central nervous system. So that integration, like I was saying before, is information processing. And the last basic stage is motor output, which also occurs in the peripheral nervous system. So as you can see by the diagram on the left, an example of sensory input, which is picked up by the sensory neurons of the peripheral nervous system, would be like touching a thorn on a cactus. Then an example of integration, which is carried out by interneurons of the central nervous system, more specifically spinal cord if it's reflexive, would be processing the pain of the sensory input. And an example of motor output, which is carried out by the motor neurons of the peripheral nervous system, would be removing your finger from the cactus thorn, because from the central nervous system, the stage of integration, you know that the cactus is painful. Okay. So now we're going to give a brief overview of neurons. So this is a neuron, and more specifically, it's a motor neuron. There's a lot of structural diversity among sensory, interneurons, and motor neurons. So neurons are fundamental units of the nervous system, and they help transfer information throughout the body. So fun fact, there are 86 billion neurons in the human brain. So as you can see, the dendrites are the branched extensions of the neuron to the left. And the soma, or the cell body, houses the cell's neurons and other organelles, and it's directly connected to the dendrites. So together, the soma and the dendrites receive signals from other neurons. The axon is the long, single, bridge-like structure of the neuron. It's the bridge where neuronal signals travel down. The axon hillock, which is the broader base of the axon, is where the signal that travels down the axon is generated and summated. So basically what this means, and the mechanism behind that summation, is that the signals from other neurons, which were received by the dendrites and the soma, is summed up and sent down the axon. Then the myelin sheath are the insulated layers that coat some parts of the axon. So the myelin sheath are in blue. The nodes of Ranvier are the gaps of the axon, the red gaps, and they're between the myelin sheaths. So the myelin sheaths actually speed up transmission of neural signals, and action potentials, which are electrical neural impulses, can jump from node to node referring to node of Ranvier, skipping over the myelin sheath, thus expediting the process of the propagation of the neural signal. And the synapse and synaptic terminal are located at the end of a neuron, which extends from the axon. So this is where the electrical neural signal propagated down the axon is converted to a chemical signal and sent to other neurons.
So now we'll be talking about synapses. So the synapse is an extremely minuscule gap, which is roughly 20 to 40 nanometers wide, whose function is to pass messages for communication purposes. So one single neuron can contain hundreds of thousands of synapses, depending on what type of neuron and their function is. So most synapses tend to be chemical, but others can be electrical. So chemical synapses use chemical messengers to communicate, while electrical synapses use ions. So through the synaptic gap or synaptic cleft, neurotransmitters are released and they diffuse across the gap where they can now bind onto receptor sites. So the presynaptic neuron creates an action potential, which we'll go into more detail later. And that causes a signal transmission into the postsynaptic neuron. And finally, the synaptic cleft is filled with extracellular fluid. So in short, the synapse is the site of transmission for electrochemical nerve impulses between two nerve cells, and they connect the neurons in the brain to other neurons in order to propagate and send those signals.
So how do neurons communicate? So each neuron can form synapses with thousands of other neurons. And as previously mentioned, a synapse is the gap between two neurons. However, this gap can't be crossed by the electrical impulses passing through. So this is done through chemicals called neurotransmitters. So the impulses that arrive at the synapse cause the ends of the fine branches of the axon to secrete neurotransmitters. And as Samambi mentioned, the neurotransmitters diffuse across the gap and attach to the postsynaptic receptors, which starts impulses on the second cell, so the second nerve cell, and opens ion channels, allowing electrical currents to flow, and also activates an influx of chemicals called sodium ions or other ions. So like this, communication is essentially achieved. And after the neurotransmitter has passed on the message, it is then broken down by an enzyme.
So now we'll be talking about how neurons communicate more in detail, and we'll be discussing the different steps that are involved during an action potential. And that's basically how a signal is transmitted between two neurons from the presynaptic terminal to the postsynaptic terminal. So an action potential is the rapid change in membrane potential that occurs when ion channels open and close. So the resting potential, which as you can see in the graph is around -70 millivolts, and that occurs when the membrane potential of the neuron is at rest, and during this stage, only some potassium channels are open. So then during depolarization, there are a few sodium channels that are open, and these permit sodium ions to enter the neuron. And this increase in positive ions inside the cell depolarizes the membrane potential. And when it occurs during depolarization, the membrane potential becomes less negative. So that's why you can see the graph approaching the more positive values. And that brings it closer to something that's called the threshold limit. So here we can see that the threshold is around -55 millivolts. So once it reaches that threshold, an action potential is then generated. So if the depolarization reaches the threshold potential, there are additional voltage-gated sodium channels that are opened. And as positive sodium ions rush into the cell, the voltage across the membrane rapidly reverses and then reaches its most positive value. So you can see the peak of the graph is the peak of when the action potential actually occurs. So at the peak of the action potential, there are two processes that occur simultaneously. So first, many voltage-gated sodium channels begin to close. And second, we have many more potassium channels that begin to open, and these potassium channels allow more positive charges to leave the cell. And this causes the membrane potential to begin to shift back to a more negative value, and towards the resting potential. And this is again because the potassium channels are open, so the positive ions are leaving, which makes the membrane potential more negative. And then the membrane actually repolarizes beyond the resting membrane voltage. And this undershoot, which you can see the hyperpolarization in stage 4, this occurs because more potassium channels are open at this point than during the membrane's resting state. So that's why it's so negative. And this allows more positively charged potassium ions to leave the cell. Then finally, the neuron returns to its membrane resting potential, which tends to stay around -70 millivolts. And that's essentially an action potential in a nutshell. And this graph, you'll find it commonly used. And also near stage 1, you can see many failed initiations. And that's because they're failed because the action potential doesn't reach the necessary threshold and the necessary millivolts necessary to trigger the action potential. Next slide.
Okay, so that was a lot of information. So are there any questions before we move on to synaptic plasticity? You can raise your hand or type in the chat. Okay, so it looks like everyone is okay with all the information. So we'll move on to synaptic plasticity.
So now we'll be going over the history behind synaptic plasticity. So Plato, who is an ancient Greek philosopher who all of you are probably familiar with, described the brain as a wax block. And this wax block may vary in size, cleanliness, and consistency in different individuals. And this theory is still considered relatively accurate today. So to expand more on this, what this essentially means is that individuals with softer wax could learn quickly, but memories were easily erased, whereas those with harder wax learned slowly, but also had more difficulty erasing memories. So wax back then was used metaphorically to resemble what we present day call neurons, brain cells. So then in 1894, a Spanish neuroanatomist called Santiago Ramon and his partner Cahel proposed that memories were formed by strengthening synaptic existing neuronal connections. But however, before the 1950s, researchers thought that the brain would be entirely static, which means the brain would remain completely unchanged after adolescence. But this was later proved wrong in 1970, where Eric Kandel and his group first found a clear connection between the physical changes that occur at synapses and how that affects behavior, learning, and memory. So then 3 years later, in 1973, activity-dependent synapses were discovered, and we now refer to them as long-term potentiation, or LTP for short, which we will also further discuss in our presentation. So finally, in the late 20th century, Donald Hebb, who was a psychologist, suggested that neurons that fire together wire together. So this saying, what this means is that if two cells consistently fire at the same time, then the strength of the connection between them will also grow stronger. And that's the history behind the synaptic plasticity in a nutshell, and basically who contributed what. And you can also see it took several centuries for this theory to finally get a hold into our current research. And that's what we really wanted to introduce to all of you today.
So as a quick pit stop before we continue, what did Donald Hebb suggest neurons do? You can put your response in the chat. Okay, so Donald Hebb did in fact say that if two neurons fire at the same time, their connection will get stronger. So thus they become more likely to fire again in the future.
Okay, so have you ever tried something and you were bad at it at first, but you eventually improved your skills? So what about memorizing something for a test or learned a new language? That's all possible because of synaptic plasticity. So synaptic plasticity in a nutshell is the ability for the brain to continuously change. Our brain is kind of like Play-Doh. We can remodel it and remold it. It's malleable. And that was first proposed by the Canadian psychologist mentioned before, Donald Hebb, in 1949. So the mechanisms of synaptic plasticity, of course, occur at the synapse, which are junctions between the neurons that allow the neurons to communicate. So changes to the synapse, these changes particularly relating to the connection between sending and receiving neurons, occur during synaptic plasticity. However, these changes depend on how active or how frequently activated the synapses between two neurons are. So for example, if I had previously memorized that mitochondria is the powerhouse of the cell a while ago, but I have not gone over it since, the synapses of that neural pathway have thus been inactive. And a change that would occur is that the synaptic connection in the neural pathway would weaken. So likewise, if I continued to consistently memorize that mitochondria was the powerhouse of the cell, the synapses of that neural pathway have thus been very active, and the synaptic connections in that neural pathway would strengthen. So essentially, if synapses are inactive, a change that would occur would be that this connection between neurons would weaken and vice versa. So the molecular mechanism behind synaptic connections, strengthening or weakening during synaptic plasticity, has to do with the amount of neurotransmitters or chemical messengers that are transferred between neurons via their synapse. So for example, the more frequently synapses are activated, the more neurotransmitters will be transferred in the synapse, and the strengthening and connection and ultimately synaptic plasticity will occur. So another molecular mechanism that is behind synaptic plasticity is how efficient the receiving neuron can respond to those neurotransmitters via receptors on the receiving neuron, but that will be talked about in a later slide. So in the context of learning and memory, repetitions of new experiences and knowledge can strengthen the connections between the activated synapses. So don't be afraid to learn something new.
So now we'll be going over the functions of synaptic plasticity. So synaptic plasticity is essential for controlling the effectiveness of neuronal communication. So when neurons talk, they have different volumes, and you can think of these volumes as shouting or whispering. And this is known as varying synaptic strength. So how do synapses change their strength? This can change mainly because of three ways. So first, they can change through the number of neurotransmitters that are being released, the number of postsynaptic receptors available on the cell, and also the opening of ion channels in the postsynaptic neuron, which allow electrical currents to flow in or out of the cell. And these are the three methods by which a neuron can change its synaptic strength. So now synaptic plasticity can be categorized in a multitude of ways, but two ways are short-term synaptic plasticity and long-term synaptic plasticity. So in short-term synaptic plasticity, what this means is changes in synaptic strength that occurs very quickly, and long-term synaptic plasticity can last from minutes to years and usually is the dominant model in how the brain tends to store and process memories. So short-term synaptic plasticity can occur within milliseconds to a few minutes, whereas long-term plasticity can last from minutes to hours to years, and long-term plasticity does again tend to be the more dominant model.
So now that we have introduced what synaptic plasticity is, we're going to talk more about the different types of synaptic plasticity. So intrinsic or homosynaptic plasticity involves the changes in the strength of the synapse that comes from its own activity. So that is changes in synapse strength that occur only at postsynaptic targets specifically stimulated by a presynaptic target. Within that, there are two types. The first being synaptic depression, which enables cells to respond to relative changes in the neural firing rate, and the second being synaptic facilitation, which improves synaptic transmission for less than a second and plays a critical role in information transfer and neuroprocessing. Secondly, there is also extrinsic or heterosynaptic plasticity. This involves the changes in strength of the synapse that come from activity from another pathway.
Okay, moving on to the types of continuing the types of synaptic plasticity. So LTP, or long-term potentiation, is the strengthening of synaptic connections between neurons via frequent activation. So it's characterized by long-lasting increases in the efficiency of signal transmission between two neurons. So to simplify this, take, for example, I consistently solve or memorize that 12 times 11 is equal to 132. So as I keep frequently activating that neural circuit of 12 times 11 equals 132, over time, knowing the answer to 12 times 11 would be instantaneous and automatic. It would be 132. And that instantaneous activation and memory recall and that instantaneous activation and memory recall demonstrates LTP, and LTP is crucial in underlying mechanisms for the consolidation of storage of knowledge and memories. So likewise, LTD describes the weakening of synaptic connections caused by prolonged inactivation. And it's characterized by long-lasting decreases in efficiency of signal transmission in the synapse between two neurons. So long-term plasticity is the dominant model for how the brain stores information, creates and recalls memories. It lasts minutes, hours, days, and even years. And LTD and LTP are two mechanisms of long-term plasticity. Short-term plasticity is where the synaptic strength and efficacy changes very briefly, and it's over a sub-second timescale, like for a millisecond.
So here now we have a detailed diagram that accurately depicts how LTP, long-term potentiation, and LTD, long-term depression, works in more detail. So when calcium ions first enter the postsynaptic NMDA receptors, this can initiate one of two different forms of synaptic plasticity, which you just heard about on the previous slide. And these are long-term potentiation, LTP, and long-term depression, LTD. So LTP occurs when a single synapse is repeatedly stimulated, which causes a calcium-dependent cellular cascade, which leads to more AMPA receptors inserted in the postsynaptic membrane. So when glutamate is released from the presynaptic cell, it binds onto both the NMDA and the AMPA receptors, which then depolarizes the membrane more efficiently. So now let's take a look at how LTD, long-term depression, works. And this occurs when some glutamate molecules bind to NMDA receptors at the synapse, and then the calcium flows from the NMDA receptors, which results in the endocytosis of AMPA receptor. And you can clearly see this in the second neuron. And this makes the postsynaptic neuron less responsive to glutamate released from the presynaptic neuron. And in short, this is essentially the molecular mechanism and the explanation for how long-term potentiation and long-term depression affect synaptic plasticity in neurons.
Now we're going to look at the mechanisms involved in synaptic plasticity. So a quick recap on stem cells. Stem cells are found in embryos that have not yet undergone differentiation, so they have the ability to specialize into different types of cells. So in this case, stem cells can differentiate to give rise to more specialized cells, such as the neuroprogenitor cells, which can then differentiate into different types of neurons. And this is essentially the process in which new neurons are formed. So until recently, neuroscientists believed that the CNS could not actually carry out neurogenesis. However, stem cells were discovered in the adult brain in the 1990s. So since then, scientists have come to the conclusion that adult neurogenesis can in fact occur in the healthy brain. So the other mechanisms include synaptogenesis, which is similar, but as the name suggests, is the formation of synapses.
Okay, now moving on to the role of synaptic plasticity in senses. So there is another form of synaptic plasticity called cross-model plasticity, and it's when connections among sensory systems are reorganized after sensory deprivation. So sensory deprivation can be caused from birth or even throughout age, and it can be caused by brain damage or disease. So essentially in cross-model plasticity, other unaffected and non-deprived sensory systems are strengthened to compensate for deprived sensory system. So take, for example, Sally has congenital blindness from a disease. So to compensate for her visual impairment, her other sensory systems, such as her auditory system, may be enhanced and more sensitive to speech. So as you can see, synapses are really important in the context of senses, especially when it comes to rewiring or forming new connections in response to sensory deprivation, demonstrated in cross-model plasticity.
So now let's go over some ways that you can promote your own synaptic plasticity. And one way is through exercise, because exercise has shown to be one effective way of stimulating brain activity, especially within neurogenesis, which Andrea just talked about. And as one extra bonus, it also increases serotonin release. And why is serotonin important? Well, that's because serotonin is a key hormone that stabilizes our mood, feelings of well-being, and our overall happiness. And this hormone impacts your entire body because it enables neurons to essentially communicate with each other. So why is it important to promote your own synaptic plasticity? Because without this ability, any brain, not just a human brain, would be unable to develop from infancy all the way to adulthood or be able to recover from any potential brain injuries. And it's also important for memory formations, learning, and behavior development in any organism. Another way you can promote your own synaptic plasticity is through maintaining a healthy sleep schedule, because your brain consolidates information and replays memories and much more while you're asleep. So by having a good sleep cycle, by sleeping 8 to 9 hours a day, that can help strengthen those connections and synapses between neurons. And even for just a few days, you should stay consistent with your connection practices. So for example, playing brain games and mind games, working on challenging problems or projects, making sure to always challenge yourself and going above your expectations, or just practicing a skill like playing the violin or doing some math problems for your next upcoming test.
So now we're moving into the case studies. And because synaptic plasticity is the ability of the brain to change, it's really essential to learning and memory, as well as repairing brain injuries. For this case study, we're going to focus on Dr. Jill Bolt Taylor. In 1996, it was a perfectly normal day for Dr. Taylor, who at the time was a healthy 37-year-old neuroanatomist at Harvard. So she woke up and she went about her morning, but she noticed a sudden headache on her left side. In actuality, she was experiencing a massive stroke in the left hemisphere of her brain. And a stroke indicates a severe interruption of blood flow and thus oxygen flow to her brain. In a mere 4 hours, she had failed to carry out basic actions such as walking, talking, reading, writing, and recalling memories.
So over the next 8 years, synaptic plasticity actually helped her recover. So the process was very difficult, as you can imagine, due to how much damage the stroke caused, as Andrea mentioned before. It damaged her ability to create language, to place meaning on language, and her ability to create sound. So Dr. Taylor had to learn vocabulary from the beginning and relearn what emotions were and relearn how to describe how she was feeling. And sleep was very important for her healing, as Simanvi mentioned before, because a lot of memory consolidation occurs during sleep. And her firsthand experience allowed her to understand how the brain is able to recover from brain trauma and injury through neurogenesis, which is, again, the generation of some new neurons, and of course, neuroplasticity. So the end result of her rehabilitation and the growth of her new neurons and the growth of her new nervous connections may resulted in her different values. And so it resulted in like her different values because, as you can see, Dr. Taylor, after 8 years of recovery, she liked different clothing and different styles and different colors. So anyway, this is really inspiring because this demonstrates how the brain is plastic and can at times recover from brain trauma and brain injury.
So now we're moving into our next case study, which is the case of Cheryl Sleich. So Cheryl Sleich in 1997 contracted a bacterial infection after a hysterectomy and was subsequently treated with the antibiotic gentamicin. But the problem is the excessive use of this antibiotic gentamicin damages these semicircular canals. And those are basically 3 joint structures located in the inner ear. And these structures are important because they give us our sense of balance along with our orientation and how we're moving of our body during space. So in Schleichel's case, these semicircular canals were damaged so severely and the consequences were dramatic. So she awoke one morning to find that she could not stand up without falling. And even she would continue to experience that same sensation even when she had fallen to the floor. So she sought a consultation from her doctor, which is Paul Bakirita from the University of Wisconsin Medical School, who created a contraption, which is a motion sensor, as you can see in the picture, to help Cheryl. And it consists of an accelerometer attached to a construction helmet and the brainport balance device, which was a tongue strip containing of several array electrodes. And these are both connected to a computer, and together these are involved in a substitute vestibular system. And the vestibular system is basically the apparatus of the inner ear that's focused on coordination and balance. So the tongue strip, although sort of bizarre looking, contained tilt sensors that detected head movements and microprocessors that relayed this necessary information to the tongue via the electrode arrays. And this forged basically new sensory pathways that are conveyed to the vestibular system, which processes balance. And this creation or this contraption was actually successful. And Andrea will go more into that right now.
Yep, so continuing on Cheryl's story, she felt tingling sensations on her tongue that corresponded to the direction she was moving in. So as time progressed, her brain began to adapt, and she knew how to turn the feelings in her tongue into a sense of balance. So she began training, and it was after this extensive training that she could depend on the helmet less. So her brain had tuned into the tiny signals that came from the 2% that remained of her vestibular system and recruited other brain waves to help out, allowing her to successfully restore her sense of balance. And it was actually synaptic plasticity that helped her to do so.
Okay, so again, that was a lot of information to take in. So do you have any questions? You can put them in the chat or you can raise your hand again. Okay, it looks like everyone is okay with the information. So now we're gonna move on to our Kahoot. I'll just,
We'll wait a couple of minutes for players to join before we start the Kahoot game. I think we can go ahead and start the Kahoot now.
Great job, everyone.
I'm just going to quickly interject here because some people chose the green. So it's long-term synaptic plasticity that usually takes like minutes to days to months to years. And that's it's long-term plasticity that helps the brain store and process information.
It's by a point. Oh my gosh.
So I think it's LTP, which is the persistent strengthening of synapses that occurs consistently. But long-term synaptic plasticity is currently the dominant model for how to store and process information and memories.
Yeah, the two types of intrinsic synaptic plasticity are synaptic depression and synaptic facilitation. Long-term memory and short-term memory, I guess, would just be categorized as types of memory. Yep. So yeah, cross-model plasticity is when other sensory systems are strengthened to compensate for deprived sensory systems. Okay, so Dr. Jill Bolt-Taylor actually was able to recover through neurogenesis and neuroplasticity, or forming new neural connections.
I think the picture hopefully might have gave it away, but it's the tongue, if you remember the contraption that her doctors gave her that created a tingling sensation that basically strengthened her synaptic plasticity and allowed her to regain balance, which I think is the next question. So the vestibular system is those three bones located in your inner ear that help coordinate your coordination, movements, balance in space and around you.
Yay, congratulations to all the winners.
And congratulations to everyone who participated. We really value your participation and we thank you for listening to our presentation. So are there any questions about the presentation or from the Kahoot that you would like to be answered? Can we give access to the slides, Pavan?
Yeah, I think I just like copied the link, if that's okay. And again, thank you so much, everyone, for listening to our presentation. It was a lot of information to handle, but I'm sure you guys got through it. And again, thank you.
Yeah, so if you like this workshop and presentation, you can feel free to go on the Simply Neuroscience website and check out the other cool things that the Brain Resource Creation is doing at the moment.
Yeah, so thank you so much to the BRC team for running this workshop. I'm sure all the attendees found it very useful and informative. And with that, that's a wrap on Simply Neurocon 2021. I hope you all enjoyed all of the sessions we had throughout the day. And all of our speakers are extremely wonderful, as I'm pretty sure you can all agree. And for those of you that want to view the slides or the any of the presentations again, they're featured on our YouTube channel. You can just watch the live stream again and just fast forward to any point of the live stream that you want to watch again. And again, yeah, thank you all for attending. It was really nice having you all in Simply Neurocon 2021. And we hope you all enjoyed it. And before you go, Shreya, do you want to take a picture of everyone? Is Shreya here?
Sorry, for some reason I became a guest, so I couldn't unmute. Okay, there we go. So if any of you are able to turn your cameras on, I'll just take a quick picture. Let's see if I can. Okay, so I'm having a bit of issues with my computer for some reason. So, Irvin, would you mind taking it instead?
Yeah, I can do. Okay, so everyone smile. 3, 2, 1. Okay. Should I do a video as well? Okay, I'll do that. So yeah, I'll take a video as well. And so just smile and wave at the camera. Yeah, thank you all again for attending Simply Neurocon 2021. I'm sure you found it a worthy successor to last year's Simply Neurocon as well. So yeah, thank you all for attending. We hope you enjoyed. If you have any questions or would like to participate in any other of Simply Neuroscientist initiatives, Shreya, can you put the link tree in the chat? And you can just click on that link and you can access our staff application, visit our website, and also sign up for future upcoming events as well as other initiatives that our organization is putting on. So thank you again so much for attending Simply Neurocon, and we really enjoyed having you here, and we hope to see you at future Simply Neuroscience events. Bye, everyone.